of our second day of the 10th International Antarctic Conference live science session. Nice to meet all of you. I also want to thank everyone who participated in our yesterday's plenary session. It was really very busy. Informative and interesting reports were presented, and we also had interesting discussions. Thanks to everybody. So for today, we're also awaiting a busy session. We plan to have 28 oral presentations, 14 of which will be made by participants from Malaysia. And we apologize in advance for the incorrect pronunciation of the names and surnames of our colleagues from Malaysia, but we'll try to do our best. I think that all speakers are present, I hope. And so I give the floor to the chair of the live science session, Ivan Parnikoza. Ivan, welcome.
Hello, everybody. So we can start, I think. First presentation is Malaysian group of Dr. Ahmad and colleagues. Please, I invite you to presentation.
Okay, you can see my slide, right? The voice is good?
Yes.
Okay. Okay. Привет, Dobry Rano, Ukraine. Good morning, Ukraine. Good afternoon, Malaysia, and maybe good evening, Antarctica. Okay, today I will represent members of eco-remediation technology groups to present our Antarctic work entitled "The Application of Antarctic Microorganism on Bioremediation Processes." So, bioremediation techniques seem urgent and requires more attention to solve the existing environmental problems. The pattern in the number of publications published on the topic of xenobiotic remediation or hydrocarbon degradation in polar region, especially Antarctica, with the application of indigenous microbe community has been increased from 2009 to 2019 before COVID. So, to address the state of pollution, it is necessary to understand the mechanism and process of the bioremediation of xenobiotic compound. The application of various microbial community originate from the polar region, especially Antarctica, can provide a better interpretation on the mechanism of the specific microbes in bioregression process. Okay, in this slide, you can see the summary of our Antarctic bioremediation research. We focus on two microorganisms, which is bacteria and also algae. Our main target pollution is the hydrocarbon, such as diesel, engine oil, and waste cooking oil. Our second target is phenolic compound. As we already know, most of the stations in Antarctica have a clinics. Okay, they mostly use the antimicrobial solutions such as Dettol to kill the germs, and they use always every day they use Dettol in the station. The target is heavy metal. Trace of the heavy metal has been reported to be give a negative effect to the Antarctic ecosystem. Okay, in the cold place such as Antarctica, the environment and the ecosystem are more affected by the contamination as a necessary cold adapted sensitivity. Okay, to introduce myself, my name is Siti Aqlima Ahmad, and you can call me Aqlima. I am an associate professor in biochemistry department, Faculty of Biotechnology and Biomolecular Sciences, Universiti Putra, Malaysia. I am currently a head of laboratory of eco-remediation technology. my research interest in the field of bioremediation, toxicology, environmental biotechnology, and enzymology. Okay, I have went to Antarctica 3 times in 2017, Sydney Island, British Station, 2018 and 19, on Hingis Station, Chilean base. Okay, this slide shows my international collaborator. Okay, first, Associate Professor Dr. Claudio Gomez from Universidad de Magallanes, Chile. His role as a consultant and environmental engineer. Currently, he is a director of Center of Research and Antarctic Environmental Monitoring. We call it CIMA. Second, I think everybody knows the famous ecologist from BAS, Professor Peter Convey. And third, Associate Professor Dr. Azham Zulkarnain from Shibara Institute of Technology, Japan. He is in this team, he is a biochemical engineer. And then, so we also have a collaboration in our university, which is Universiti Putra, Malaysia. Okay, Dr. Azmi, he is currently a HOD, Head of Department of Biochemistry. His research interest in the plant biochemistry for in Antarctica research is more on the algae. While Dr. Shrena, research interest is in microbiology and molecular, focus on the enzyme technology. Okay, at the same time, we also have a collaborator from Universiti Sains Malaysia. Okay, Dr. Farina Marikan is expert in the microalgae taxonomy, while Dr. Japarang Lalong expert in the environmental microbiology, especially on cyanobacteria. Lastly, the last slide on the collaboration with the Dr. Wong Chuyen from International Medical University. She expert in the algal physiology, and while Dr. Kalila from Associate Professor Dr. Kalila from UiTM for University Technology Malaysia is expert in applied microbiology. Currently, our teams, Eco-Remediation Technology Lab based in UPM, Universiti Putra Malaysia, have 12 PhD and 4 master student work on the Antarctic research. Okay, main focus is diesel. Diesel is incentively used to complement our activity in Antarctica, especially scientists. Diesel oil is a petroleum product that supply fuel for the automobile, aircraft and ship, as well as generate electricity in the station, especially for the scientists to switch on the equipment. Diesel oil, most of the research station in Antarctic use a special Antarctic blend, SAB diesel, which is exclusive blend for the better performance in cold temperature. Okay. As encouraged by the Antarctic Treaty System, many Antarctic research stations are currently opting for the use of renewable energy source like solar, ocean, and wind energy. However, most of the stations in the region still require the use of the diesel to generate the power supply. Okay. Why we choose diesel? Because the increasing rate in accident involving vessel in the Southern Ocean, which is Antarctic Ocean, have highlighted the potential of fuel spilling into the Antarctic environment. Giving the human activity the Antarctic perspective to incentive and diversify the Antarctic environment, conservation is pressure. Okay, the challenge is to protect the polar environment is the effectiveness management of the hydrocarbon contamination from the oil spill. Okay, diesel pollution tend to be more persistent in sub-zero temperature compared to those of the warmer area. This is due to the increased velocity and evaporation of the low temperature. Okay, and the limitation of the nutrient that signification inhibit the metabolite activity of the hydrocarbon degrading microbes. Okay, this slide shows the toxicity effect of the diesel pollution. Okay, So policy, attitude and the awareness on the environmental protection have improved in the recent year. However, the risk of the diesel pollution remained. Okay, if we left it untreated, the toxicity of diesel will cause negative effect on the living organism in the polar regions. Okay, as you can see, several studies have reported the toxicity effect of diesel fuel on the specific location of Arctic and Antarctic, and effect on the biological, on the microorganism, and also effect the wildlife in Antarctica. Okay, especially for the penguin and Antarctic seabird. Okay, so in this study, there is a, okay, as we know that the optimum condition, the problem statement, the optimum condition of diesel degradation by microorganisms still unknown. Okay, we need to optimize the condition. And like I said, the diesel contamination water is highly toxic and rather give the negative effect to the marine organism. The contamination of the diesel from the polluted water is expensive and time consuming. Okay. Okay, this is the overall method that I use in our study. First, we will isolate the sample from the soil, and then we screen, we optimize, we identify, and then apply in to apply in bioreactor. Okay, you can see this as previously we do the sampling from the Sydney Island 2017, and then later 2018 and 2019 until last year 2020, we sampling at the Bernardo O'Higgins station, which is a Chilean base. Okay, back to the methodology. Okay, so in this study, we try to optimize the condition using the one factor at time and response service methodology. And then after we do the optimization, we identify the microbial, okay, the microbes. Okay, either there is we using the consortium, so we need to do the metagenomic analysis, and sometimes we isolate the single species, and then we just run the 16S RNA for the bacteria and 18S RNA for the algae. And then from this condition, from this result, we compare this result and then apply in the Antarctic, in the real field, which is Antarctic in the O'Higgins station. Okay. This slide shows the pictures of 2019. We try to apply in situ experiment using the bacteria. And then in the mid of 2019, we tried to develop the system, the bioreactor system. And then in 2020, last year, January, we bring all the bioreactor to Antarctic, and then you can see the left picture. There is a discussion with our collaborator, Dr. Claudio Gomez in Punta Arenas, and then we bring the bioreactor to the station. Okay, you can see that we sent two of our students to go to Antarctic to run their PhD experiment. Okay. For the details result, you can see after this, I think 8 of my students will present. Okay, first you can see Fareez, the oral, oral 5, around 7:20, he will talk about the optimization, the biodegradation of diesel using the bacteria isolated from soil. And then my student Nadira will present on the hydrocarbon biodegradation using the marine sample, which is water, seawater. Okay, and then for the poster tomorrow, my student will present on also the consortium, using the consortium bacteria to degrade the diesel. Okay, and then like I said, we also focus on others hydrocarbon like waste cooking oil. So today, my student Khadija will present about the biodegradation of waste cooking oil and pure cooking oil by Antarctic bacteria community. We get the sample from the Bernardo O'Higgins station. Okay, and then we focus we focus on the heavy metals. So my student Syazani will present about the statistical optimization of molybdenum reduction using the psychotolerance marine bacteria isolated from Antarctic. That it showed that these bacteria have a high salinity to bioremediate to reduce the molybdenum. While my student Gaia, we use the algae, try to degrade, not degrade, try to reduce the molybdenum using the algae, which is low salinity algae. It is the freshwater algae. Okay. And then we focus, like I said, we focus on the phenolic because we have a lot of, most of the station have clinics. So my student Kavi will talk about the single bacteria that develop the single and mixed bacteria that have an ability to degrade the phenol at the low temperature, the combination of the single bacteria. While my student Atira will talk about the not the consortium, the bacterial community to degrade the Antarctica. Okay, and lastly, we just tried the new method to isolate polylactic acid, PLA degrading bacteria using Antarctic soil. That's all for me. Thank you.
Thank you, Aqlima.
Is it too fast or what?
For your very interesting and very important, very brief talk. As is a good example for the next our lectures. And if we have some questions for Aqlima, we have 5 minutes for this. No question. I have question.
Okay.
Aqlima, what microalgae do you study or your colleagues study? What groups of microalgae?
Microalgae. For the microalgae, later you can talk because I'm not because I'm present of my team members. Maybe Dr. Faradina, can you switch on your mic? Because he's expert in microalgae. Because my team is in the conference right now.
Okay, no problem. We will contact. Thanks. If no question, we can go to the next speaker, Dr. Aminu and group from Malaysia too. I invite you to presentation.
Okay. Thank you.
Okay. Okay, can you hear me?
Yes, Suraj, loud and clear.
Okay. Can everybody see my slide?
Not yet.
No.
Not yet.
Yes,
Now
we can see your
presentation.
Okay. All right. Good morning, afternoon, everybody. My name is Aminu Surajo Usman, a PhD student, University of Science, Malaysia. I'm working under the supervision of Dr. Fareez American. I am happy to present my topic on the Antarctic cyanobacteria and their response to salt stress. So cyanobacteria are prokaryote photosynthetic microorganisms that are among the pioneering organisms on planet Earth. They have been in existence for over 3 billion years ago, and they are also regarded as the main primary producers of organic matter. They are also believed to be the first organism to release oxygen into the then primitive atmosphere. In terms of their distribution, cyanobacteria can be found in almost all kinds of habitats, including Antarctic lakes, thermal spring, and then tropical acidic soil. This unique distribution of cyanobacteria exposes them to various kinds of environmental stressors, including salinity stress. They They are also known to produce a range of primary and secondary metabolites, and some of these metabolites can be produced as a result of response to environmental stressors, such as cyanotoxins and compatible solid. So salinity is an important abiotic stress that can cause deleterious effect to cyanobacterial cell. All of these, cyanobacteria has to develop special mechanisms to enable them to withstand various salinity stress. One of the mechanisms include ionic regulation, and the other one include osmotic regulation. Ionic regulation include taking in more salt from the environment, while osmotic regulation include accumulation of low molecular compound known as compatible solid. And this compatible solid can be synthesized by the organisms themselves, or they can be accumulated from the environment when other organism releases them. So based on this, cyanobacteria are either called halotolerant or halophile. The halotolerants are a group of cyanobacteria that can withstand certain exposure to salinity, while halophile cyanobacteria that requires salt them to thrive. So the need for this research, it stems from the fact that in Antarctica, the cyanobacteria are exposed to various salinity fluctuation. And this salinity fluctuation may occur as a result of evaporation when the fresh water evaporates, or as a result of less precipitation, or it can occur due to summer-winter fluctuation, where during ice formation, the hypersaline brine, concentrated brine can be formed at the bottom of the lakes in most of these Arctic water bodies. Therefore, despite this fluctuation in salinity, cyanobacteria in Antarctica, little is known about how cyanobacteria respond to this salinity fluctuation within Antarctic ecosystem. Therefore, we are interested to find out whether cyanobacteria isolated from Antarctica can grow in laboratory in a range of salinity. We're also interested to find out, does the exposure to salinity influence the accumulation of compatible solid in our strains? And we're also interested to find out, does the salinity influence the production of microsystem in our strain? So for the material and method, it should be noted that we are working on a very old sample that was originally collected by Dr. Paul Brode, and the samples were both frozen for over 3 to 4 decades. After receiving the samples, we tried to establish the culture using various culturing techniques, and we managed to revive most of the species. And then after reviving them, we tried to isolate the samples and brought them into an algal collection. Isolating the individual organism and establishing an algal collection for each strain, we subject them to morphological identification using relevant identification key guides. At the same time, we also subject them into molecular analysis by amplifying their cysteine sDNA. All the strains are also screened for the presence of toxin encoding genes using relevant primers as listed here. And then the next thing is, after establishing pure culture, we grow the organism and maintain them in either BG11 or BG11-N for the growth of heterocytes forming cyanobacteria. And then from there, we developed a scale of culture that was used for the salinity tolerance experiment. And we used BG11 and its modified version by addition of sodium chloride to obtain the relevant salinity that we used during this experiment. And then we selected 3 salinities from a start: 15 ppt to represent brackish condition, 35 ppt to represent seawater condition, and 65 ppt to represent hypersaline condition. So we grow them in these salinities, to determine whether they can grow or they cannot grow. And we assess the growth by qualitatively using observation and taking some pictures, while qualitatively using wet biomass and chlorophyll A as a proxy for growth. Then the next thing is mass culture for the screening and quantification of characterization of compatible solids using nuclear magnetic resonance spectroscopy. So from our results, in this work, we managed to identify 17 morphospecies from 3 different orders. 9 from Oscillatarias, including 2 morphospecies of Formidium autumnale, 3 morphospecies of Microcoleus vaginatus, and 4 microspecies of Wilmotia murai. We also managed to identify 4 syncochials, including 2 Formidesmis frisley strain, 1 Leptolimbia aviolarum, and 1 Nodosilina nodulosa. We also managed to identify about 4 Nostecia, or 4 Nostecal morphospecies. And then this is just a representation of how the morphospecies look, a representation of the morphospecies. And then we try to corroborate our morphological information with our molecular data. We managed to amplify the 16S for all the 17 species, and then we use the BLAST to determine the closely related species to our morphospecies and gene bank. And then we downloaded relevant sequence for the phylogenetic tree construction. And then it should be noted that most of our morphospecies corroborate with our molecular information, with the exception of few strains like this one, Wilmotia murai, which matches with Formidium atrensis. It should be noted, however, that Formidium atrensis was proposed to be reclassified into Wilmotia murai. And then our Leptolimbia matches with Leptolimbia pida. And then none of our nostoc morphospecies matches with any nostoc at the gene bank, but it matches with nostoc. So probably the new species, but we need to confirm this using another molecular marker. And from the salinity experiment, the preliminary experiment where we export the strain into 3 different salinities, we managed to identify that 8 strain grew until 65 ppt, 6 strain grew at 35 ppt, and 5 strain grew at 15 ppt. 1 strain does not grow in any of the salinity. Therefore, it is regarded as sad. After establishing the fact that this strain can grow at this salinity, we further decided to subject them to more salinity to determine the maximum salinity tolerance range of each and every species. And thereafter, for those that grow until 65 ppt, we extend the the growth condition until 150 ppt to reflect the hypersaline condition, to mimic the hypersaline condition that is normally obtainable at some of the bottom layer of the thin bottom layer below the ice in some of the lakes. And we assess this data, we brought them in a test tube in a duplicate culture, and we quantitatively assess the data using chlorophyll A and wet biomass as a proxy for determining the group. So from this graph, you can see this is the wet biomass data. And if you look at like this one's graphs here, they follow the general pattern of decrease in biomass along the salinity gradient. In most of the strains, highest biomass were recorded at the control culture after a decrease is observed along the salinity growth gradient. As the salinity increases, the biomass concentration decreases. The exception is on few strains. We have 2 for our Microcoleus vaginatus here that grow best at the type salinity, and 1 Anagnostodinema pseudocatissimum that also grow at 5 salinity. The most interesting result here is this one, Omidesmis priestley, that grew until 105 salinity. Although the strain produces the highest biomass at the control, but
now we have technical break. No connection.
At the control at 45 ppt. The data for chlorophyll A concentration. The data for chlorophyll A concentration also follow the same pattern with the data for weight biomass. You can see this decrease at the saline in chlorophyll A concentration at the salinity increase, with the exception to the same species that produces highest biomass at 5 ppt for Macropoleus. They also produces highest chlorophyll A content at the same salinity. So by putting together our observation, that is qualitative data and the quantitative data, we summarize and categorize our strain into 3 categories. A low salt, a salt-sensitive strain, that is one of the Wiimotia murai that grew only at the control and it does not grow at any of the salinity. A low salt tolerant strain that grew at including 2 Microcoleus baganatus and 1 Formidum autumnale that grew at 15 ppt, and another 2 strain that grew at 25 ppt, that is for Anagnostodinema pseudocatissimum and Nodosulina nodulosa, and including another 5 strains that grew at seawater salinity, including 4 of our Wiimotia murai and 1 Formidum autumnale. And the last group are the moderate salt tolerant strain that grew at salinity above sea level, above seawater salinity, that is hypersaline condition, from 55 until 105. Notably here, the most interesting strain in this research is Formidum Formidesmis priestley, that is microcystin producer, which grew until 105 ppt and with consistent biomass production. So after this, the next thing is we are in the process of mass culture cultivation to obtain enough biomass for nuclear magnetic resonance spectroscopy for the quantification and characterization of compatible solid. And the next thing is we will select the Formidesmis priestley and subject the strain to rigorous growth analysis. We want to see the effect of salinity stress, the growth, metabolic response, and genomic potential / microcystin production in this strain that grew until 55 ppt. In conclusion, our experiment showed that cyanobacteria isolated from Antarctic lakes can adapt to varying sodium chloride concentration. Study also provides us with an opportunity to identify a compatible solid as an osmo-adaptive compound that might have been produced by cyanobacteria isolated from Antarctica for the first time. Our genomic and metabolic studies, after completed, will also add to the knowledge to address one of the scarce questions, which said how can changes in the frequency of extreme event be used to improve the biological understanding and testing. Another aspect of this research, after completion, will also help us to address one of the questions about the genomic and adaptive basis of Antarctic and Southern Ocean organisms and communities. And this will also provide us with further insight into how cyanobacteria in Antarctica respond to salinity. And it will enable us to understand how how this organism survive in this ever-changing Antarctic environment, and how they tolerate the salt level at molecular level. Thank you very much for listening.
Thank you very much, Aminu, for this very interesting presentation about cyanobacteria. And we have time for a small, short question. You can not only write your question in chat, you can ask now if you want. Okay, I will read from the chat. Dear Dr. Aminu, do you expect that diesel can be fully broken down and oxidized to carbohydrate in situ? And how would that impact the acidity?
Didn't get the question, please.
You can see
it
the
in
question
chat.
in the chat. Look, please.
Okay, let me check.
Diva Faradina, if you can answer her.
Cannot see the question.
No. Look, please, the next message from the chat. It seems to me it's your chief.
Okay.
Yes, I think this is Dr. Aqlima's presentation. She was the one who talked about the diesel, bioremediation of diesel contaminants.
Oh, yeah, I've seen the question. I think it's not for me.
Yes, and that is why I invite Faradina to answer, if she would like, of course.
Oh, okay.
Can you answer? No?
No, I think this is Aqlima, because I work just with the algae taxonomy and phylogeny, and she does the work using this algae to break down. So maybe I get her to answer that.
Okay, yes, I think yes, we can, because we already have a result on the algae degrading of diesel. Maybe later you can see there is a presentation on the algae degradation. And normally we use the green algae.
It seems to me the sense of this question is a little bit another, but it seems to me, Ivan, please let us continue.
Okay. Sorry for this small misunderstanding. Thank you, Dr. Aminu, and we now go to the next presentation to Dr. Darham and group. Please, you're invited to the presentation.
Ivan, I'm sorry, Maria Pavlovskaya wants to ask a question.
No time for the question now.
No time. Maria, please write in your chat and you will be answered.
Okay. So I guess this is my turn. Can everyone hear me? Okay.
Yes.
Thank you. I hope my audio and internet connection is okay throughout my presentation. So, okay. Greetings to everyone. My name is Syazani binti Darham, and I am not a doctor yet. I'm a postgraduate student in the University of Putra, Malaysia. And today I'm going to present my research titled Statistical Optimization of Molybdenum Reduction using Psychrotolerant Marine Bacteria isolated from Antarctica. So this is my brief presentation outline. I'm going to talk about the overall research background, and then I'll move to problem statement, objective, and then I will discuss my result, and then I will conclude my research. So moving on to the introduction.
Syazani, can you share a slide?
Sorry?
Because I cannot see
we
your
cannot
slide.
see your presentation. Please share it.
Please share.
Okay. Wait. I think my Suddenly my there's a hang in my laptop.
Probably you can tell us your presentation. No?
Syazani, do you want me to show your slide presentation?
Yes.
Okay, I'm sorry for the interruption. Okay, so I'm going to share my slide right now. Okay, can everyone see my slide?
No.
No,
No.
can't yet.
Can anyone see my slide right now?
No. No slide. No.
Uh,
Oh my God.
do you see a share screen button in Zoom?
Okay.
Oh,
Now it's loading. Okay, cool.
it's okay.
Okay, sorry. I think I should start from the beginning. Okay, so I'm going to present my research, which is the statistical optimization of molybdenum reduction using psychrotolerant marine bacteria isolated from Antarctica. Moving on to my research background, we all know that Antarctic is a very geographically isolated region, but however, it is not as pristine as we thought it would be due to the recent studies that have reported the presence of pollutants, including heavy metals in the Antarctica. So these traces of heavy metals, due to various sources, especially anthropogenic sources, have been reported to negatively affect the ecosystem. These are some of the examples of the studies that reported on the heavy metal contamination in Antarctic seawater, Antarctic sediments, as well as the Antarctic soil. So these heavy metals are causing negative effects on the Antarctic organism. This is because most Antarctic organisms have greater lipid content than tropical species, which makes them more vulnerable due to the accumulation of toxic contaminants in their fat. For example, heavy metals such as mercury has adverse effects in penguins, such as high mortality rates, reduced food intakes, which leads to weight loss, causes progressive weakness, as well as decreased fertility. So my research is mainly focused on molybdenum in Antarctic. Molybdenum is a type of heavy metal that is a rare heavy metal, and it has been the reports that it has been detected in Antarctic has been published in the recent decades. These are some of the examples of the research published. And molybdenum is actually a type of silvery white metal with various oxidations between 2+ and 6+. It is important to living organisms as it plays a critical role in several metabolic pathways as a catalytic component of certain metalloenzymes. It has ductile properties, highly resistant to corrosion, and has one of the highest melting points of all pure elements. Due to these properties, it is being widely used in many industrial products and being used such as in products such as alloying agents, used as industrial catalyst components for machines and turbines. It is being used in paints and corrosion inhibitors, as well as lubricant additives. So there are various sources of molybdenum in Antarctic. There are anthropogenic sources and natural sources. For anthropogenic sources, activities such as waste incineration, sewage disposal, and oil spill are the prime sources of these pollutants, while for natural sources, Examples are migratory animals such as birds. Due to their wide foraging habitats, there are increase of heavy metals in their excrements, and also volcanic dust that are transported and being deposited in the Antarctic ecosystem. So these pollutants can also be sourced from nearby land masses and can reach the polar regions via long-range atmospheric transport as well as the oceanic currents. So countries with active metal mining that lies relatively close to Antarctic region, such as Chile, are also one of the primary source of the pollutants of the molybdenum. So molybdenum, although it is essential for the human, for a living organism, it can cause health hazards to human, and in human it can cause symptoms such as hallucination, seizures, aching joints, and hyperuricemia, while in animal studies it induce secondary copper deficiencies in animals such as cattle and sheep, which leads to lameness and osteoporosis, while in rodents it causes growth retardation, kidney and liver histological changes, reproductive failures, bone deformities, as well as anemia. So to solve, one of the upcoming alternatives to solve the problems of heavy metal pollution is by bioremediation. So bioremediation is actually a process by which we utilize organisms such as plants, fungi, and bacteria to degrade and immobilize hazardous organic and inorganic pollutants to ecologically safe levels with the help of certain metabolic processes. For bioremediation of molybdenum, it is called the molybdenum reduction. And this is the pathway which there are the involvement of electron donor and electron acceptor, and with the help of enzyme phosphomolybdenum reductase and with the presence of phosphate, we get a desired compound called molybdenum blue. This molybdenum blue is our desired compound, as it is blue in color. It can be seen with naked eyes, and it can be measured using spectrophotometer using 865 nanometer wavelength. And it is a type of colloidal compound that can filter out from the environment. So, so far there's only been one research on the molybdenum reduction in Antarctic, and it is in the year 2013, utilizing soil bacteria. So the problem statement is that the trace of heavy metals, including molybdenum, have been found in Antarctic environment from various sources, especially anthropogenic sources, and its toxic effect towards organism has been observed. And the remediation of molybdenum in Antarctic seawater using indigenous cold adapted marine microorganism has not been studied, and its optimized conditions are still undetermined. So the aim of this study is to actually optimize molybdenum reduction using selected molybdenum reducing isolate for maximum reduction using conventional one factor at a time OFAT and statistical response surface methodology RSM approaches. So this is the overall method for optimization. For OFAT, we did a conventional one at a time optimization of various parameters such as salinity, temperature, substrate concentration, as well as the pH. While for RSM, we did using central composite design by utilizing software called Design Expert. And it is RSM using CCD is to determine the optimal values and condition, as well as to study the interaction between significant factors. So this is the general workflow. First of all, we grow and then we enrich the culture, grow it in the LPM, low phosphate media, and the parameters will be the variable will be there are variable changes in the LPM for the OFAT and RSM, and then the blue production, MO blue production will be observed and then will be measured using spectrophotometer. So the key player is bibacterial strain Marinomonas sp strain AQ589. It is an Antarctic marine bacteria. It is halotolerant and psychrotolerant. Gram-negative, it is motile and aerobic, and widely distributed in marine environment, including in polar regions. So the bacteria is isolated in the water sample in the vicinity of Base General Bernardo O'Higgins Reclamation Station, Antarctic. It is isolated in the year 2019. And moving on to my results and discussion. The first one is my OFAT result. For salinity, we can see that the bacteria can grow up to 80, more than 80 ppt, but the optimum is shown at 50 ppt. So it proves that the Marinomonas, the strain Marinomonas is halotolerant, while for the temperature, it shows optimum reduction at 15 degrees Celsius, while it actually can grow up to 25 degrees Celsius, which proves that the strain is a psychrotolerant bacterium. While for sucrose, while for carbon source, the bacteria prefer sucrose as a carbon source at the concentration of 2%. While for nitrogen source, the bacteria prefer the ammonium salt, especially the ammonium sulfate, at the concentration of 3 grams per liter. So nitrogen is essential to enhance the production of bacterial metabolites as well as the bacterial growth. While for the molybdate concentration, it is important to determine to achieve the maximal reduction as well as to ascertain the concentration that causes inhibition. And as we can see at 10 millimolar, the reduction is the highest. While for pH, we can see that the molybdenum reduction is slightly on the acidic side because phospholipid is highly unstable at neutral pH and is rapidly oxidized. Studies have shown that the optimal pH varies from 5 to 7. And as we can see, the bacterial growth increases as the pH increase due to the alkaline tendency preference of the bacterium. So for CCD, we took six parameters that are significant and obtain the response, and then we analyze it using Design Expert software. So this is the result that we obtained, ANOVA result that we obtained from the CCD. We can see that the model is highly significant and the lack of it is not significant. While the R-square is highly is satisfactory at 0.87. We can see that for the model, there are significant models for the linear and as well as the quadratic models. While for interaction, we can see there are 8 interactions that are significant: A, B, AE, BC, BE, BF, CE, DF, and EF. These are the 3D surface plot that shows the graph of the interaction between 2 different parameters. For example, for BC, we can see the temperature and the carbon source interaction producing a peak at 16 degrees Celsius and carbon concentration at 1.8%. So these are the images of the 3D surface plot of the 8 significant interaction of the parameters. So as a conclusion, we can actually compare the optimal condition determined using OFAT and RSM. For OFAT, we can see the values are not as refined as RSM because as you can see, for salinity, temperature, as well as nitrogen concentration, the RSM value is slightly more specific than OFAT and more reduced. While for the OFAT, the incubation day requires 8 days to achieve a molybdenum production OD at 2.459. While for our RSM, it took 4 days to reach OD of 2.2, which means the RSM is a method that we use to further refine the result that we obtain for OFAT. So So as a conclusion, conditions for molybdenum reduction must be optimized to ensure successful remediation, while the second one is the preliminary optimization of molybdenum reduction using conventional one factor at a time OFAT, and then refining further using the statistical RSM is the ideal approach. So that's all for me. These are my references. Thank you.
Thank you too. I think you can see question in the chat and you can answer in the chat because we have not time, and we will go. Thank you for your very interesting presentation, and we invite the next speaker, Dr. Radzi, and
Ivan, I am sorry. We had to take a photo. We promised, but
okay.
After the third, after Susan, we'll make a photo. Please, could you, friends, could you switch on your video cameras in order we can see all of you? Farah, Erisa, Hayar, I don't know how they are pronounced. So, are all of those who wish to take a photo are ready? Please smile.
Smile. And
is it taken?
One more.
One more.
Because we have 2 screens of participants. Okay.
Thank you. Thank you.
Finish.
Thank you, Helen.
Okay. Next speaker, please. You can start your presentation.
Hi, hello. Hi.
I actually don't
we hear you. Don't worry. You can start.
Oh, okay. Can everyone see my slide?
Yes, yes, we see your presentation.
All right. Very good. I think I need to click on this slide. All right. Can I start now?
Yes.
Okay. Hi, salam and greetings, everyone. My name is Ranina Radzi. You can call me Ranina. I am a graduated master's student under the supervision of Dr. Fareez American from Universiti Sains Malaysia. So today, I will talk about the diversity of terrestrial cyanobacteria from selected habitats on SygnY Island, South Orkney Islands in Antarctica. Okay, let's go. Okay, firstly, everyone knows that cyanobacteria is a very unique prokaryotic algae, which existed in 3.5 billion years ago, and it has no membrane organelle. It also has a chlorophyll A that undergo photosynthesis, and it also has the ability to survive and thrive wide range on conditions like pH, salinity, radiation, and also extreme places like Antarctica. And we also know that Antarctica is a sun-exposed ecosystem. So from there, cyanobacteria is an ancient group that can mostly be found there. From there, the confirmation of the presence of the group is from the first discovery of Shackleton expedition, which documented the presence of benthic mats from filamentous cyanobacteria way back in 1907, and as for the current study, which we conducted in SygnY Island, the floristic survey was firstly conducted by Brody in 1979, and now we wanted to see the floristic survey in the current time. And from there, so there have been numerous approaches to the taxonomy of cyanobacteria. So at present, we are using a polyphasic approach which combination of morphology, ultrastructure, and molecular. Okay, here are the project aims, which I divided into two. First, we wanted to record the description of morphospecies using selected techniques, and then from there we compare the morphospecies recorded with the previous study by Brody 1979. And from there, the description of morphospecies that we recorded, we selected three cyanobacteria, which is Nodosirina signicis, Wimmutia murai strain 1 and strain 2. From there, we see that we observe their ultrastructural analysis, and then we also investigate the closely related of morphology by using 16S rDNA for both for all three cyanobacteria, which is Nodosirina signicis and Wimmutia murai strain 1 and strain 2. As for using ITS to see the closely related morphotypes, ITS is only will be used on Nodosirina signicis. Okay, so here are the sampling sites in 1971-1974. As you can see here is Paul Brody in 1971-74, where he collected samples from 122 sites, and then he managed to record 162 morphospecies of algae, and from there only 49 morphospecies of cyanobacteria were able to record it. Okay, moving on to the current one, where the sampling the samples were collected by Dr. Japarin Lalung. So here he is, Dr. Japarin Lalung. He managed to collect 37 sites. From there, we managed to record 17 morphospecies of cyanobacteria. This was in expedition with BUS into the 2015-2016. Okay, in order to achieve the objective that I told before, these are the methods that were used. The sample collection were collected from sites that were close as possible to the original location that remain accessible, and then for the preparation of culture media, we use BBM and BG11, and also we add cycloheximide supplemented with cycloheximide. The isolation techniques, which I will explain later, and then the identification of morphospecies. So here are the sample collections and culture establishment in Antarctica. So Dr. Japarin Lalung established 27 mixed culture, and from the 27, 9 cultures were utilized in this study, as these plates only contains cyanobacteria. Okay, so moving on to the isolation techniques, where I use two methods. The first one is the most probable number, MPN, is a serial dilution techniques to obtain culture from field samples such as soil. Okay, and the streaking techniques is the standard bacterial soil isolation to obtain unialga cultures. Moving on to the identification of morphospecies, where from the culture, the characteristics were recorded by measuring the cell size, cell shape, and then in the necrotic cell, all sort of the details need to be recorded by using light microscope. And then, in order to identify, we used to compare it with Brody 1979, and also reference was made to report by Komara and Anaxidis taxonomic keys. Okay, so here are the results, where you can see the column in Brody 1979 is where Brody managed to grow in culture from 122 sites, whereas the current one, the 10 cultures were managed to 10 cultures from 37 sites were managed to grow. So Brody recorded a total 49, and current one we managed to record 17 morphospecies of cyanobacteria. Okay, moving on. Okay, here are the 70 morphospecies recorded, contains of 4 groups: Corcales, Nosocales, Oscillateralis, and Cyanococales. From the 17, 3 were successfully grown in uni-alga culture, and the remaining 14 is still in a mixed culture. Okay, here are the photo taken by me, and here are the recorded. These species are also recorded previously by Brody in 1979, and these species are also rare in sight for both present and previous records, and it also were recorded previously in Antarctica, other than SygnY Island. Okay, next. Okay, here are also these species are the dominant site found in previous record. But as for the current study, the dominant one is only the Leptolimbaya fovularum here, and the Formidesmis Roslee, and other species are rarely observed. And these species are widely distributed in Antarctica. Okay, here are the interesting part where in this study, we managed to record 7 new species in SygnY Island, which has not been recorded yet by Brody. And this probably could be occurred long time ago, but in a very low abundance, or maybe due to the environmental change that happened currently in Antarctica, and or probably other reasons could probably from the first year populations have increasing resulted with the high nutrients. Okay, now so 3 out of 7 are considered for polyphasic assessment, which is Nodosirina signensis, Wimotia murai strain 1 and Wimotia murai strain 2. Okay, so moving on with the 3 cyanobacteria, I proceeded with these methods, which to get the uni-alga culture, and then moving on to the ultrastructure analysis to see the internal structure, and molecular analysis to see the closely related morphotypes by using 16S rDNA and ITS. Okay, first, this is a Nodosirina signensis species, a new species from SygnY Island, Antarctica. Firstly, we recorded morphologically as Leptolimbaya because they have the similar characteristic and very resembles with Leptolimbaya. However, after going on with the molecular assessment, it resulted closely related with Nodosirina genus. So from there, Percussion 2011 shows that a clear separation of this Nodosirina and Leptolimbaya, and it has been described to possess a unique nodules in a low light intensity. So to date, there are 7 species of Nodosirina in Nodosirina genus were recorded. Okay, as you can see here, this is the Nodosirina signensis. From the arrow, you can see a form of like kind of a nodule, but with twisted of spiral formation. So this is the unique of the Nodosirina genus, and the rounded apical cell here, you can see here the rounded apical cell resembles the Leptolimbaya. Okay, and moving on here. Okay, the difference between the Nodosirina, the distinct between Nodosirina signensis and other 7 Nodosirina is by their cell shape, which is they are a bit, they do have isometrics and a bit longer than wide, and they have an obvious barrel shape here. You can see here a barrel shape, and then the next one, this is the only species, Nodosirina signensis, that are lack in cell granulation. And the special feature was the uniserrate trichomes lie parallel or twisted around one another within a common shape, in one shape that resemble nodules. So they form a close together and they form a nodules. Okay, this is the nodule Selenia ultrastructure that fits the description of nodule Selenia. And these are the 16S rDNA where you can see here. Okay, this is the zoom part. Okay, the nodule Selenia sinensis. Oops. Okay, nodule Selenia sinensis are well separated from the other nodule Selenia, suggesting it is a new species with a high bootstrap value. And this is the ITS results where you can see where they are similar with D1 helix structures, and the sequence of nodule Selenia differed in sequence detail from those 7 species. Okay, moving on to the 2 cyanobacteria, strain 1 and strain 2, which is Wilmotia murai. Okay, this is the first strain where you can see a bit shorter than wide here, shorter than wide, and a bit the crossword are distinct, and you can see very obvious. Compared to the strain 2, which is more longer than wide, and the crossword are a bit slightly constricted. And we know that Wilmotia genus was erected from Formidium, genus that originated from Antarctica. Previously, they were known as Formidium murai, and then they renamed it as a Wilmotia genus, Wilmotia murai, sorry. And then to date, there is 3 species were recorded in Wilmotia genus, which is Wilmotia stricta from Brazil and Wilmotia koreana from Korea. The genus currently defined has no distinct characteristic and can only be identified with molecular. Okay, the comparison between these 2 strains and other Wilmotia strains is where the cell shape, and the cross wall. And another feature that has not been recorded is the necrotic cell, where the previous Wilmotia murai recorded absence of necrotic cells. But for both strains, the current strains, they have a necrotic cell. And Stronecki 2011 has considered that a fasciculation of trichomes is a dicharacteristical characteristic. However, these features are not observed in both of the Wilmotia murai strain. Okay, these are the Wilmotia murai ultrastructure fits the division of the genus. And okay, this is the 16S rDNA, okay, where you can see it is a major clade of Wilmotia in this shaded box, and you can see there are 2 parts of it where it is noteworthy that the major clade of Wilmotia murai showed a clear separation or well separation between strain obtained from cold region. Okay, this one is the cold region where you can the Antarctic cold region places, and the tropic. And this one is the tropical regions. So both regions might be morphologically similar, but they are genetically distinct. Proposed to reevaluate and reclassify the ones that not from Antarctica to reclassify as a new name from Wilmotia genus. Okay, so the conclusion is from this study, we gained 70 morphospecies of Cyanobacteria were identified. And then 10 morphospecies of Cyanobacteria in the current study were recorded by Brody. 7 morphospecies of Cyanobacteria are new records of SIGNY. 3 were successfully isolated in alga, and 1 new species has been described as using my polyphasic approach, Nodosina signicis. And for Wilmotia, as for Wilmotia murai, only molecular assessments were able to identify the genus. I would like to thank my team members, findings, and British Antarctic Survey for this project, and thank you everyone.
Thank you, Kamina, for very interesting talk about cyanobacteria. And now we have time for the question. We can answer by your voices if you want, or write in the chat, and we all time have possibility to read in chat and answer in chat. Please only provide after for all question, okay? So No question? I have one question. If we speak about cyanobacteria, yeah, can we speak about some diversity of flora of cyanobacteria from the different parts of Antarctic Peninsula? Are some data about this? Something like Pete told us previously, probably some regional groups of species in Antarctica.
Oh, for my study, I focus more on cyanobacteria, but the previous study in Sygney Island, by Brody, he also collected algae, not only cyanobacteria.
Okay, cyanobacteria.
Cyanobacteria.
Mm-hmm.
Yeah, cyanobacteria.
Cyanophyta cyanobacteria is like similar. So
no, it's different, totally different.
So know some patterns, regional patterns of cyanobacteria in Antarctic Peninsula, or?
No, for now I only focus on Sygney Island, South Orkney Island, Antarctica, not the peninsula.
Okay.
Is that answering your question?
Okay, thank you.
All right.
If we have not a question, we can go to the next speaker, Dr. Roslee, and group of Malaysian and UK scientists. Please, I invite you to presentation.
Can everyone see my slide?
Not yet. We need more time.
Not yet.
Yes, I can see your presentation.
Unstable. All right, shall I start now?
Yes, please.
Okay. Hi, greetings everyone. My name is Fareez Ahmad Roslee, under the supervision of Dr. Siti Aqlima, currently doing my PhD. So today I'll be presenting my PhD project, which is on the growth optimization and kinetic profiling of diesel biodegradation by a coal-adapted microbial consortium isolated from Trinity Peninsula, Antarctica. To be specific, from the General Bernardo O'Higgins Station. So moving on to my research background, we know that from years and decades of studies and observation, the scientists there, or the polar scientists, are aware now that diesel have been used extensively in Antarctica. So in Antarctic, they have a special Antarctic blend variant, which is a winterized diesel formulation that contains higher volatile compounds. So its applications there includes as a fuel for transportation. It is also used as a fuel for power generation, also heat generation. And in some places, it can also be used as a fuel for waste incineration. Chemically speaking, it is a flammable liquid, and it contains a myriad of hydrocarbons, such as the polycyclic aromatic hydrocarbons, naphthalene, paraffins, and olefins. According to the reports by the ATCM in 2015, the average diesel consumption in Antarctic region can go up to 90 milliliters, and you can see in the next slide, this I will share here. So in this slide, you can see the breakdown of some of the bases in Antarctic and the amount of diesel that they consume per year. And here we can see some bases, they are very, very reliant on the use of diesel, can go up to millions of liters per year. And some basis, they use just a little amount. So given this situation, we know that the Antarctic community is not yet ready to actually move to shift to a renewable energy source, because we are still heavily reliant on this diesel consumption. And by having a lot of diesel being transported into Antarctica, it can actually lead to many catastrophic events, such as here, as you can see in the picture. Although it does not happen as often as before, the ships ran aground after colliding with glaciers or iceberg is actually still happening at small scale in Antarctica. So some of the implications are that it can cause oil slick formation in the sea area. So it can you hear me? Okay. It can cause intoxication in certain marine life, such as the penguins and seal bear. So in this next slide here, it shows the summary of some detrimental effects of hydrocarbons, not just diesel, but it applied to all hydrocarbons. For a prolonged exposure, it usually leads to multiple types of cancers, such as the melanosis, skin cancers, and other types of cancers, such as pulmonaries. And for acute effects, it can also cause mild intoxication and drowsiness, and usually seen in penguins. It can also cause oxidative stress in the cells and tissue. And sometimes, for a prolonged exposure, it also leads to infertility and teratogenic and mutagenic effects. So one way to combat this problem is through bioremediation, although there are other types of remediation, such as a physical as well as chemical. However, bioremediation is actually regarded as to be a better solution, since it is more eco-friendly and it can remove a lot of pollutants over a period of time. Here in my study, I am emphasizing on the use of bacterial consortium, because it is assumed that the synergistic effect between this soil organism, the bacteria, will actually give a better result as compared to just using a single culture. The concept is, as you can see in the slide below. Okay. So here the table summarized some of the strains that have been used in previous studies to degrade multiple types of hydrocarbon contaminants. All right, and here also in my study, I will also include metagenomic, but in this presentation, I will not actually go into the metagenomic part yet. Moving on to my problem statement, these are some of the problems that are generally encountered by the Antarctic communities in their studies. The first one is that petrogenic hydrocarbons tends to be very persistent in cool climates such as Antarctica. And then the second one is that even though there are extensive researches on the biodegradation, however, only certain studies or very little of the studies actually have succeeded in situ application of these bacteria. And the last one, yes, there is still major knowledge gap pertaining to the existence of interaction between the soil microbial consortium. And my hypothesis for my study is that the bacterial soil consortium is able to actually efficiently assimilate diesel for its carbon source. And we can see the comparison between the OFAT and RSM, where OFAT will give a better result. So in my study, I have laid out 5 objectives. However, for the sake of this presentation, I will just emphasize on the first 3, which is to evaluate the intrinsic diesel degrading capability, to optimize the growth and diesel biodegradation conditions, as well as to generate kinetic profile on the microbial growth of the consortium. Here is the experimental design consisting of sampling, screening, optimization part through 2 methods, which is the 1 factor at a time, as well as responsive methodology, followed by the model validation. And then there is also parameter estimation through kinetic profiling, incubator fabrication, and optimization, as well as metagenomic analysis. And this is the oversimplification of the methods that I use in my study, where the microbes from the soil is actually obtained from both pristine area as well as polluted areas. So those samples are then inoculated in broth, grown and tested in pH medium, supplemented with diesel. There are 2 responses that I am looking out for, which is the diesel biodegradation as well as the growth. So the degradation will be assessed through gravimetric as well as GCFID analysis. And for the growth, it will be measured through absorbance at 600 nanometer for bacterial growth. And these are the factors that I have tested in my studies, which are pH, diesel concentration, salinity, nitrogen concentration, nitrogen source, and temperature, together with the levels that I use in my studies. So this optimal range will then be confirmed and validated through one-way ANOVA followed by a 2-keys post-hoc test. So it will then be used for RSM studies. Okay, moving on to my results. The first one, which is the screening part, it is a 2-phase consisting of the first and second stage of screening. In the first part, I use a 1% of diesel concentration over a 7-day period. And I started with about 28 of soil sample. And from that, only about 6 of them actually give performance that is higher than 80% of diesel degradation as well as high growth, as you can see here. So I have narrowed down my sample, and then I proceed with a screening using a 2% to actually come out with the single consortium that I will be using for my study, which is BS24. And as I have said before, the optimization part consists of 2 methods, which is the OFET and also RSM. So I have skipped the OFET parts and present to you just the RSM. RSM consists of the placket Berman and central composite design. So in placket Berman design, it is a screening method that consists of 2 levels, and it can the range that is used in placket Berman design is from OFET analysis actually. So this the value the +1 and -1 is obtained from 1 factor at a time analysis. So the range are in this table here. Can see there are 5 of the factors that will be tested in my study. And a placket Berman design actually generates a table where it is a combination of factors, and I have to follow all of these to actually get this almost similar result compared to the OFET. And with just 12 runs, the degradation efficiency obtained is about 91.23% of diesel degradation within 7 days. And this is the ANOVA table showing that the model is significant. Some of the factors, actually the aim of doing plaque abandonment is to screen what are the factors is important to the study. So here we can see that the factor A, B, C, D, E, from that, only factor C, which is the salinity, is not significant to my study, whereas B, which is the temperature, is very, very significant. So from that, I proceed with just 4 factors in my next analysis, which is a central composite design. And this time it consists of 5 levels instead of just 2 in plaque abandonment. So here are the levels that was used in my studies. And in this time, it consists of a lot more run compared to the plaque abandonment, which is 12. Here is 27 experimental runs. And by doing so, I obtained 95.5% of diesel degradation at run number 15, which is the highest diesel degradation with the following levels of the conditions, with the following conditions. And here it shows the ANOVA table. In central composite design, it is a bit different compared to the plaque abandonment, where in central composite design, we can actually study the interactions between the factors, whereas in plaque abandonment, the factors are actually the interactions are omittable. So here, the same. What I can say is that it is how to read this table is the same. The coefficient of determination is very significant, followed by the adjusted. The adjusted R-squared is in agreement to the model. And here I list all the interactions exist in the study. As we can see that temperature exists in all 3 interactions here, from pH temperature, ammonium chloride concentration and temperature, and diesel concentration and temperature. So it shows that temperature is very cardinal in our study, in my study. All right. And followed by these, and then the model has to be validated. And here you can see that the p-value is greater than 0.05. It means that the predicted model and the experimental run has no significant difference. And there are also additional studies using the kinetic models. So here I use about 5 models, or 5 or 6 models, to actually gauge the maximum growth rate, as well as the half inhibition and half substrate concentration constants. And here you can see in the final equation, the best model is Tazier as compared to other growth model by Monod, Aiba, Haldane, and Yano and Wong. So my conclusion is that the screening using RSM method is actually best compared to just doing OFFAT. And from RSM, we can actually look upon the interactions between the factors. Okay, the optimized condition through RSM method was 1% of sodium chloride, pH 7.25, 0.75 gram of ammonium chloride at temperature 12.5 degrees Celsius, and about 1.75% of diesel concentration. And the final mineralization of diesel is about 95% over 7 days of incubation. And for kinetic modeling, as I have said before, the substrate inhibition constant Ki, the Ks, the substrate half substrate concentration, and the maximum growth rate can be observed, can be actually determined. So I guess that's all for today. Yeah.
This all. Thank you, Ahmad. I think we have time for one question. Some question in chat. No question in chat. So, Ahmad, will you plan to test your models, your results in wild Antarctic nature? Because, of course, this pollution is very dangerous and very actual problem in all stations, especially in Ukrainians. Vernadsky station, for me, this is most important.
Okay, to answer your question, actually, I have conducted my studies in General Bernardo O'Higgins Station. I have used these parameters, this model, in my study in last year during my time in Antarctica.
So you plan to continue?
However, the result is a bit progress. We need to scale up the model. Because now the result is valid for lab scale. However, for upscaling, however, the result is a bit offset from what I am expecting. So we are still in progress to actually optimize.
Okay. Okay, thank you for your presentation. And I can invite next speaker, Kenta Sato, yes, and co-authors. Please, you invite to presentation.
Can you see my slide?
Yes.
Thank you. So, good morning, good afternoon, and good evening, everyone. Now I'm going to talk about my presentation. Speaker is Kenta Sato, Shibabara Institute of Technology, Japan. In my presentation, I'm going to talk about the isolate from Antarctica soil could degrade polylactic acid, which is a kind of the plastics. First of all, what are plastics? Plastics are artificially synthesized resin with excellent properties, such as lightweight, flexibility, and it is hard to be degraded because of its chemical features. However, such plastics remain in natural environment for a long time. Plastics are not degraded easily and accumulate entire the earth. This problem is called plastic pollution. Many marine organisms are damaged by plastic pollution, especially microplastics, whose diameter is under 5 millimeters, make this universal concern more serious. Microplastics such as polypropylene and polyethylene have been found in the oceans of Antarctica, reflecting the changes in the global environment. In order to solve this plastic pollution problem, environmentally friendly bioplastics have been attracting a lot of attention. Polylactic acid, PLA, is one of the bioplastics and it's made of biomass. Through fermentation process, biomasses are converted to lactic acid. Purified lactic acid is polymerized to form PLA by bonding their hydroxy and carboxy groups to each other. In these days, PLA is used as tableware, and it is also used in packaging materials for foods, since PLA is proved as non-toxic materials for human health. PLA consists of simple chemical structure and it is degraded easier than other plastics. This table shows us many reports about microbes that could degrade PLA and other substrates. Some amycorrhaptosis species could degrade orthoprotein, for example, silk fibrin powder and casein by using proteinase. PLA is also degraded by esterase and lipase because PLA belongs to fatty acid esters, and chitinase is able to degrade PLA and other bioplastics. Chitinase is an enzyme that hydrolyzes the polymer produced by plants. However, all of these microbes are not isolated from cold environments. So the aim of this study is isolate the bacterium that could degrade PLA from Antarctica soil. Next, I'd like to talk about my methodology. Firstly, PLA degrading bacterium was isolated from Antarctic soil by enrichment culture. By using mineral salt medium, this inorganic liquid media was prepared in Erlenmeyer flasks. A composition of mineral salt medium, MSM, is shown at right table. After acidization by autoclaving the flask, pasteurized PLA granule was added as a sole carbon source. Add 0.5 grams of Antarctic soil and shaking the flask at 90 rpm, 30 degrees Celsius for 2 weeks. After 2 weeks of incubation, 2 milliliters of media was transferred to new flask and incubate them. It repeat 5 times. After enrichment culture, 0.1 milliliters of medium was spread on nutrient agar plate and incubated. 1 colony was picked up and transferred to new MSM medium containing PLA and shaking them. After 2 weeks from inoculation, 0.1 milliliters of medium was spread on nutrient agar and incubated. After incubation, many colonies were visible. It means that this isolate on the plate could grow by using PLA, and isolation is success. The isolate was named as strain N3 and used for further experiments. Secondly, to identify the isolates, gram staining, enzyme activity, molecular identification, and other tests were conducted. In molecular identification, 27F and 1492R were selected as PCR primer for 16S ribosomal DNA, and its sequence alignment was searched on the Web Basic Local Alignment Search Tool, WebBlast, from National Center for Biotechnology Information, NCBI. And phylogenetic tree was constructed from alignment data. Thirdly, to estimate PLA degradation rate, PLA film degrading test was conducted. PLA film was formed by evaporating chloroform from PLA solution. The film was sterilized by soaking 70% ethanol for 2 hours. After soaking film, it was washed by sterilized distilled water twice. From enrichment culturing, by making the same medium was incubated as pre-culture medium. 100 milliliters of MSM was sterilized by autoclaving flasks. For incubation, the film was put into the flask containing MSM and 1 milliliter of pre-culture medium was transferred to the flask. The test was conducted triplicate. These flasks were incubated at 90 rpm, 30 degrees Celsius for 4 weeks. During incubation, the number of bacteria was estimated by CFU counting. At 0.1 milliliters of the medium transferred to 0.9 milliliters of sterilized water, mixed well, and at 0.1 milliliters of this solution to another 0.9 milliliters of still water. After dilution the media, spread them on nutrient agar. One colony was formed by one bacterium. That's why the number of bacteria could be estimated. After incubation, the film and the media were separated by filtering. The filter unit used in this experiment has the ability to hold particles larger than 0.5 micrometers. The films were weighed its dry weight, and degradation rate was calculated. The concentration of extracellular protein in the media was estimated by Bradford protein assay. If the isolate could degrade PLA film, it uses PLA as nutrition and increase its number. And the concentration of extracellular enzyme will increase. Also, the degradation rate increased since the film is consumed by the isolate. Now I'm going to talk about the results. These are results of identification of strain N3. Left figure shows micrographs of the isolate. Scale bar means 10 micrometers. Strain N3 is gram-negative, rod-shaped bacterium, and it has catalase activity. By sequencing the PCR fragment and annotation, strain N3 had high similarity with the genus Nactibacter. This figure illustrates the phylogenetic tree of strain N3 and family Oxalobacteries. To construct the phylogenetic tree, the DNA data of 9 species in the family Oxalobacteries were downloaded from GenBank and CBI. For the genus Nactibacter belongs to the family Oxalobacteries. Escherichia coli was selected as outgroup. It was 98.73% that similarity between strain N3 and Nactibacter alkali tolerance. This graph shows the increase of microbial numbers. Error bar means standard deviation. And the vertical axis were plotted a logarithmic scale. The number of viable bacteria was increased from 10 to the 6th power to the 8th power in 4 weeks of incubation. And from this data, doubling time was calculated. The doubling time is the time that it takes for bacteria to double in number. In this experiment, it takes 3.27 days for strain N3 to double in its number. This graph shows the concentration of extracellular protein in control group and test group. In control group, there was a PLA film. However, inoculation was not carried out. Error means standard deviation, and asterisk mark means statistically significant in student t-test. The concentration in control group was 0.038 microgram per milliliters, whereas the test group was 1.5 microgram per milliliters. The protein concentration in test group was higher than control group significantly. This graph shows the degradation rate of PLA film in 20 days of incubation. The rate in control group was 3.4% and the test group was 6.4%. However, these results were not statistically significant. The film in test group was degraded as nearly twice as the control group. These are discussions and conclusions. In the degradation rate, the rate of control group was 3.4%. It indicates that PLA film is hydrolyzed because of long term of incubation. And strain N3 has isolated from Antarctic soil. Therefore, the degradation test in cold temperature makes the strain properties clearer. In protein assay, increasing the concentration of extracellular protein indicates that the isolate has extra enzymes that could degrade PLA. In this experiment, PLA degradable bacterium was isolated from Antarctic soil. From the results, the isolate named Naxibacter species strain N3 could grow and increase its number in MSM containing PRA. Thank you for attention.
Maria Pawlowska, please.
Yeah, thank you. Thank you for the very interesting presentation. I really enjoyed it. I just wanted to clarify some stuff about the sampling procedure. I wonder if the sampling location where the Antarctic soil was sampled, if it was under any potential anthropogenic influence that could result in this local community experiencing this PLA or any other xenobiotic contamination. Is there any station close by or something like this? Could you clarify?
Thank you for question. The sample of the soil.
Yeah. I mean the sample of the Antarctic soil, where it originally came from.
Yeah, and it is the came from British base on Sygyny Island.
Mm-hmm. So there is a base close by, and there could be any xenobiotic contamination and any such influence, yeah, on this soil.
Let me see. In my experiment, to isolate the one bacterium, I did enrichment culture. So the soil transferred to the rigid media and transfer the new flask and incubate and incubate.
Yeah, this part is what I understand. I think we can discuss it in the chat together with you, because I wonder about some local conditions, and maybe it would be just more convenient for you to discuss it in the chat. Okay?
Okay.
It's possible? Okay. Yeah, great. Thank you. Thanks a lot.
Thank you.
Okay, thank you, Maria. Thank you, Kenta. More questions? Okay, we finished this presentation and questions, and I invite Hajjak Zahri and co-authors to the presentation. You're welcome. No voice.
Please mute your microphone.
Can you hear me?
Yes. But now I see.
Okay. Can you hear me?
Yes.
Yes. Okay. Yes. So basically, I'm the PhD student from Universiti Putra, Malaysia. And before that, I would like to apologize if there is any interruption during my presentation, because my Wi-Fi is unstable right now. Okay, this is my PhD project entitled Microbial Bioremediations of Waste Canola Oil and Pure Canola Oil by Co-adapted Antarctic Bacteria Community from Sains. And this is my outline that I will present today. So for the background, as we know that cooking oil is composed of saturated and unsaturated fatty acids and glycerol and also as a medium for heat and mass transfer, also can provide energy and play a crucial role in human nutrition. And most important, see from the slide, this is the statistic on the consumptions of vegetable oil worldwide by all type, starting from 2013 until 2018. And there's estimated that about 90% of vegetable oil used for consumption. However, repeated heating cooking oil can cause several toxicity, which several chemical reactions can take place during the frying process, cause oil structure modified, and increase the toxicity. And there are several diseases that might cause from this repeated heating cooking oil, such as atherosclerosis, Alzheimer's, and also cancer. And these might cause from the major toxic produced through chemical reactions, such as aldehyde, ketones, and so on. And this is the adverse effect of repeated heating vegetable oil, especially for the canola oil, where Bautista et al. 2014 reported that potential health implications of canola oil is adipose tissue and canola oil, because canola oil is a common dietary fat widely used in most Antarctic research stations. However, there is no report on the vegetable oil pollution in Antarctic. However, the vegetable oil spill still might happen or occur during the loading and unloading of the cargo, when spillage small amounts is possible. And this oil spill may percolate and create groundwater contamination and cause damage to surface of ground soil and marine. And there also the transporting of waste of cooking oil from Antarctic via ship will increase fuel consumption of the ship, which is a waste of resources. So how this problem can be solved? It is through bioremediation, where we know that bioremediation is one of the methods use of microorganisms to break down or to biodegrade the contaminants. And we know it less cost and non-invasive to the environment. And past research mentioned that microbial consortia is more effective in degradations of to degrade the oil and grease for pure mixed and consortium culture. As you can see, the consortium culture able to degrade better cooking oil for less incubation time compared to pure and mixed. And in bioremediation, there is a responsible enzymes that play a crucial role in biodegrade. So for cooking oil biodegradation, the responsible enzyme is lipolytic enzyme, which is might be esterase or lipase, where these enzymes is responsible to degrade complex molecules of the vegetable oil into fatty acids and glycerol. And at the end, the pathway of the metabolisms, the energy will produce, and this energy will be used by the microorganisms for their metabolism. Okay, this is my problem statement. Hydrocarbon oil spills in Antarctic can cause various hazards to other organisms. And most important, there is no study reported on biodegradations of WCO and as well as PCO using bacterial community from Antarctic soil sample. And currently, there is only one publication on the biodegradations of waste canola oil using single culture, which is Rhodococcus sp from Antarctica. And this is my objective. My main objective is to study the optimum conditions of biodegradations of PCO and PCO. And then the sub-objective is first to screen the best WCO and PCO degrading bacterial community from Antarctic soil samples. Then I will optimize the biodegradations of WCO and PCO using OFAT and RSM approaches. Okay, this is the research design. First, start with the maintenance and activation bacterial community cultures, and then proceed with the screening for the microbial community, and then proceed with the optimization process through OFAT, the classical method, and statistical analysis using response surface analysis. 8 soil samples from Antarctic collected, coded by BS1 until BS28 that collected from the Bernardo O'Higgins Chile station. And my waste cooking oil also collected from this station too. Okay, this is my project workflow diagram. So basically I have 28 soil samples. I culture into 10 ml nutrient broth and then upscale into 5 ml nutrient broth. And at this stage, I make several copies of glass raw stock, then centrifuge to get the pellet, and proceed with the screening and optimization by adding 1 ml liquid that's standardized to 1 at OD 600 nanometer inside 50 ml minimum salt media. And then it is incubated on the 150 RPM orbital shaker for 5 days. And then the vector growth and degradation analysis respectively. Okay, this is the factors that I'm going to optimize the vector growth and biodegradation for OFAD. Basically, I have 7 factors: salinity, pH, type of nitrogen source, concentrations of nitrogen source, temperature, yeast extract, and substrate concentration. Okay, and then I proceed with the statistical analysis of the optimization process, where using Plackett-Bowman design and CCD, where Plackett-Bowman design is a method to identify the important factors among the large number of variables. Since I have 7 factors, so I need to screen first whether there is no significant factors in biodegrade WCO and PCO. So that's why I'm using PBD to remove all the insignificant factors, and then all the significant factors. I proceed to the CCD, the central composite design, and the model curve determination will obtain. So moving on to as you can see, the highest degradations for WCO is BS23, followed by BS12, 11, and 14. However, I select BS14 for my further experiment, because when I screen for the PCO, the bacterial community in BS14 able to degrade high compared to BS23, 11, and 12. So this BS14 I use for the OFAD, the optimization process, using 1 factor at a time for 7 days. And this is the result for salinity. And you can see both in WCO and PCO. For the left is A is WCO and B is PCO, have high degradation percentage, both at 0% salinity. And then for pH, it also shows the same, that the bacterial community BS14 same with type of nitrogen source. They preferred ammonium sulfate for them to grow and degrade the substrate. And then it's slightly different with, for the ammonium sulfate concentration, where in WCO degradation, the bacterial community need high amounts of ammonium sulfate concentration compared to the PCO. This is because of the, as I mentioned before, repeated heating cooking oil can produce several toxic product in the oil itself. So this may be this toxic compound may cause may give effect to the bacterial community, and that's why the bacterial community need large amounts of energy for them to survive or perform their activity. Okay, and then for CO degradation pivot at 10 degrees Celsius, where there is for WCO degradation, A graph, there is no significant difference between, there is significant difference between 5 and 10 degrees Celsius, also between 10 and 15 degrees Celsius. So that's why I chose 10 degrees Celsius in these factors. And there is slightly interesting here, the bacterial growth is slightly increased when the temperature is at 25 degrees Celsius. So this is because of the bacterial community, the bacterial communities able to survive at this temperature. However, the bacteria that survive in that particular condition does not have the responsible enzymes or does not cannot degrade the WCO or PCO. So that's why the degradations WCO and PCO is slightly low. That's one of my factors to optimize the WCO and PCO degradation. You can see both have high degradation at 1 gram per liter of yeast extract, and I'm using yeast extract because lots of papers report that the yeast extract might promote the degradations of hydrocarbon. So it did increase the degradations of cooking oil, because as you can see in WCO degradation, when there's no yeast extract, it only can degrade WCO at 60%, while if at 1 gram per liter of yeast extract, it can increase the degradation up to 90% degradation. So I'm choosing, so that's why I'm including yeast extract as one of my factors. Okay, then for the last factor is substrate concentration. Both have a high degradation. And of course, the bacterial growth is still increased or maintained, although the substrate concentrations increases because of the presence of yeast extract. So that's why the bacterial growth is still increased or stationary, not reduced. Okay, so this is the conclusion for the optimal degradations of a fat. So I have mentioned before, only nitrogen concentration have a different condition, 1 gram per liter for WCO and 0.5 gram per liter for PCO. And then this, I proceed with the optimization using response effect method, using PBD, where I screen first to eliminate non-significant factors. And for WCO, I found out that the model is significant and all the okay, so my model is significant and all the factors are significant with my R-square is 0.999. While for PCO, the model is significant and only the BC, E, and F are significant with R-square is 0.999 also. And all these significant factors will proceed to the central composite design. So this is my result for WCO. So the model is significant and I'm only focusing the interaction between different type of factors. So only the interaction between D and F is significant, which is between temperature and substrate concentration, which is WCO concentration. And the lack of it is not significant, and R square is 0.906. This is 3D contour model graph for significant interaction. So basically, as you can see, at low concentrations of WCO and around 12 to 13 degrees Celsius, it produces or so while for PCO, the model is significant and the interaction between AD, BC, and BD are significant. And my lack of it is not significant with my R square is 0.885. So this is the significant interaction between A and D, around 7.2 to 7.3, the pH for the pH, and around 1.1 to 1.4, it yields 70% PCO degradation, while for C and B, ammonium sulfate is around 0.7 to 0.8, and at high concentrations of yeast extract, it yields 90% PCO degradation. And lastly, for the significant interaction for PCO, between B and D also show from the previous significant interaction. So the model is validated by comparing the expected value and actual value, and the p-value is greater than 0.05, and there is no significant difference between the expected value and actual value for WCO and PCO. So therefore, the model is validated. So basically, RSM is based on the contour plot. We can see RSM, I reduced 6 days incubation time compared to OFET for 7 days. And then I use the RSM point again for 7 days and to see whether RSM is better than OFET or not. And it shows that RSM give better degradation compared to OFET, where for WCO at 1% oil concentration, OFET only can degrade around 84%, while WCO around 96%, while PCO around 84% for OFET, and RSM can increase until 91% degradation. And the ability of the bacterial community to degrade WCO and PCO in cold environments may be useful applied in bioremediation approach, as the optimum temperature for this community was between 10 to 15 degrees Celsius. Remediations of liquid-rich wastewater, which is wastewater treatment, can be used in the Antarctic station, and applications on bioremediation of waste canola oil from storage containers using bioreactor tank. So this is my publications, my references. It is all. Thank you.
Thank you, Kajak. And probably one short question.
I have a question. May I?
Yes, okay. Yes, of course.
Yes, of course. Not only for this author, for another Malaysian author too. There are many publications we could see during this session about different remediation species here in Antarctica. I have one big question to all of them. What about biodiversity and ecology impact of such species in case of use them in frame of Antarctica? What exactly do you think about this?
I'm sorry, my internet is unstable. I cannot hear the questions, the exact questions. Can you
you repeat? Can you write? Okay.
Right in the chat.
Okay, I will try. Thank you.
Okay, sorry.
It will be better in chat, and this question not only for Kajak, this question for probably you can answer on this question.
For all Malaysian research life sciences group.
Yeah.
Okay.
Thank you, Irina. I finish this presentation, and we invite next speaker, Nadira, and group with next presentation. You are welcome. No, I don't.
Switch on your microphone.
Microphone.
Nadira, unmute.
Wait. Okay, hi.
Okay.
Yes.
Okay.
My name is Nadira Zakaria. I am in my 6th semester doing my PhD. My topic of my study of interest is the biodegradation of diesel using marine bacterial community isolated from non-contaminated seawater in Antarctica. Okay. Right. So there are 10 parts of my presentation, and I thank you for joining me. God, it's not functioning well. Okay, so 200 years since the discovery of Antarctica, and it has become a forcing house of global expeditions and research. So data collections from varying interdisciplinary researchers have become valuable to the scientific community, especially because these data represent how our planet is responding to increasing pressure from human activities all around the world. Okay, why I chose diesel as my hydrocarbon of interest is during the 20 years bibliometric study that I did, it reached out to me that diesel is the scientific community in Antarctica is largely dependent on the usage and consumption of diesel in Antarctica, mainly to generate electricity and heat to sustain living and activities in Antarctica, both in the austral summer and even more so during the winter. Besides diesel, we can see that Antarctic lubricant is also another hydrocarbon of interest in Antarctica, and also Antarctic lighter crude oils. Okay. Antarctica is governed, as we all know, by the Antarctic Treaty Protocol, and according to one of the sections of the treaty, it prohibits the introduction of non-native species to Antarctica. And so with this, in fact, and also with the fact that diesel is becoming more and more used in Antarctica, this also increases the risk of hydrocarbon spills, both on land and in water. So the microbial community, it has been shown time and time again that there exists hydrocarbon degraders, both from the contaminated and non-contaminated environments, regardless wherever in any parts of the world. So in Antarctica, bioremediation is limited to the native species of that particular site of any contamination. So bioremediation should be site-specific, and bioremediation coupled with other kinds of technologies can in fact be a better solution as a solution for hydrocarbon spills that occur in the cold environments of Antarctica, specifically to study the marine environments in Antarctica. Okay. Oh my gosh. Okay, so due to the increasing scientific activity in the research stations, mostly located around the coastal regions in Antarctica, there's little known about the fate and the impacts of hydrocarbons on the Antarctic marine ecosystems. And due to the chronically low temperatures of Antarctica, it makes for a very slow process of hydrocarbons in comparison to the tropical marine environments across the world. So my objective for this study is to screen for oil degrading bacterial community isolated from marine seawater. So firstly, I would go for screening. The samples were collected from a period of 2 years during an Antarctic expedition by my supervisor, Dr. Siti Aqlima. And so these samples were screened for diesel degrading activities. And furthermore, I proceeded for the optimization of growth conditions, combining both OFAT with the responsiveness method. For the identification purposes, I opted to go for metagenomics using PacBio for the platform. And then I chose to study upon the effects of heavy metals on the diesel degrading ability of the chosen consortium before optimizing the scale-up biodegradation process. And lastly, for the application of the in-situ bioreactor of diesel in Antarctica. So for the sampling and screening purposes, about 50 milliliters of seawater from 40 plus points in and around the station, base General Bernardo O'Higgins. So 50 milliliters of the seawater was collected in sterile polycarbonate tubes and kept cryopreserved until further used, and then inoculated into nutrient broth. And I used Bushnell house media modified to suit marine bacterial community. And secondly is for the process of optimization, I chose firstly was to optimize the biodegradation ability of the bacteria by analyzing the factors one by one. And then from this one, I selected all the factors into a placket Berman design, which was chosen to eliminate the individual factors before proceeding lastly into the central design composite to see the interactions of these significant factors. So for the identification of the community, which hasn't been done yet, it starts off with DNA extraction and lastly with the smart bell library construction. I'm hoping, so the identification of the community will be done with the samples from day 0, day 3, and day 6 throughout the incubation period within the exposure of the bacterial community to diesel. So for my first objective, I had 40+ samples of from Antarctic seawater samples. And in this one, I chose my samples labeled OTB. It has high degrading ability with low microbial mass. For my second objective was the optimization of the growth conditions. So the first factor that I chose was the pH. So here I used 2 buffer systems, tris buffer and phosphate buffers. And in both buffer systems, for the pH 8, the degradation percentage was higher, with higher affinity in the phosphate buffer system, which is expected because marine microorganisms have can consume the phosphate in the buffer systems as compared to the tris buffer. Second factor is salinity. Again, here the degradation is higher in the 30 parts per thousand, which is similar to the salinity of seawaters in Antarctica, which ranges from 22 parts per thousand to 32 parts per thousand. So as for the temperature, I use 5 points due to the lack of incubation apparatus that we can actually use in Malaysia. So the diesel degradation is better in the 10 degrees. However, we can see that at 25 degrees, the bacterial growth is still the bacterial growth is still supported in higher temperatures up until 25 degrees. This is because in the bacterial community that I have chosen, there are cyclotolerants, but the key members of the diesel for the diesel degraders are actually better off in lower temperatures, 10, 5, and also 15. So 10 is the highest. So for a nitrogen source, diesel degradation was seen better in ammonium nitrate. For the concentration, it can be seen that 1 gram per liter does best for the diesel degradation, supports both bacterial growth and diesel degradation. So going on to my last factor is the diesel concentration. So with all the optimized factors above, you can see that diesel degradation is supported up until 4% diesel. And there's a significant drop of diesel degradation at 5.5%, because here probably the key members of the Antarctic bacterial consortium has already died off due to the toxicity of increasing diesel. So going on to my placket Berman, the individual factors you can see here is pH, salinity, temperature, nitrogen concentration, and diesel concentration. So it was hoped that only the significant factors would proceed onto the central composite design. As you can see here, pH is not significant, leaving only salinity, temperature, nitrogen concentration, and as also diesel concentration as the substrate of interest. So going on to my central composite design, which was initially planned to analyze the minute points to analyze the interaction of the factors. So you can see here the ANOVA table that was generated by the software between salinity and nitrogen concentration, temperature, nitrogen concentration, and also temperature and diesel concentration. These interactions are only significant. So if we can see here between nitrogen and temperature, diesel degradation occurs highest in the presence of highest nitrogen concentration at seemingly temperature range of 10 to 12. This is because nitrogen concentration has been shown time and time again that it actually supports diesel degradation, but to a point where the higher amount of nitrogen would ultimately be toxic to the members of the key degrading communities. So another one is between nitrogen and salinity. Again, at the higher points of 1.5 gram per liter of nitrogen, diesel degradation is highest. At a low salinity point of 2.5% salt. And lastly, between diesel concentration and temperature, you can see that again here with the expectation that diesel degradation occurs in the lower values of temperature. So for the model validation, model validation was done as a last point from the CCD above to verify that the model and the equation generated by the software is indeed valid, which can then be used for many applications afterwards. So then I proceeded on to using the optimized media. I applied the bioreactor as a to upscale the diesel degradation. So the working volume that I worked around with was 5 liters. So this bioreactor was actually placed, I had the opportunity of going to Antarctica in the last season before the pandemic hit us. So the bioreactor tank was placed outside of the cabin and it was exposed to real-time Antarctic climate and temperature. So daily the temperature was around 3 degrees. Highest I recorded was about 5 degrees. Okay, so then I opted to optimize the different aeration conditions of the bioreactor. So the bioreactor was installed with a 2-blade impeller. So we could control the speed of the impeller. Here, the impeller, the highest speed of the impeller was 60 hertz, equivalent to 150 RPM. Coupled the 60 hertz with a bubbling system, I was able to achieve yes, okay, almost close to 80%. I'm wrapping up. Almost close to 80% of degradation, but for this experiment, I only used 1% diesel, which is equivalent to 50 milliliters of the whole total working volume of the 5-liter bioreactor. And okay, so next one is the effect of the diesel concentration to inoculum ratio. By increasing the diesel concentration to inoculum ratio, it was seen that the amount of diesel was starting to exhibit a toxic effect to the inoculum, and the diesel degradation reduced as the concentration increased. So you can see here that the bioreactor did indeed generate a oily waste. So yeah, that's it, wrapping up. Wrapping up, this last graph of mine is a biodegradation of day-to-day in situ in Antarctica. So you can see here that at day 5, the diesel degradation have started to approach 80, going on to 60 and 70, where at day 7, the bacterial growth increases. So I would like to finish off? Yeah. And that's it from me. Thank you.
Thank you to Nadira for interesting presentation with some very clear practical aspect. We can see this reactor is really optimistic, and I wait for the questions. We have time. Maria Pavlovska?
Yeah, I just have one small technical question. Thanks a lot for the nice presentation. First of all, it was really my pleasure to listen to you. I just got a bit lost at the experimental design where you told about harvesting of the bacteria, about inoculating and so on. So one part is growing in culture, yeah, is your experiment. Another part is the one where you identify the microbes in the communities that you are studying with 16S amplicon sequencing. As far as I know, this can be a bit tricky part because a lot depends on how you harvest the bacteria from seawater. The 16S analysis can depend on this. So I was just wondering, did you harvest the cells of bacteria plankton with some specific filters, or how did you concentrate the cells of bacteria plankton for the further amplicon sequencing?
Right. Well, to answer that, Maria, my identification of the community was actually done from bacterial culture in the nutrient broth instead of the seawater. Because in doing so, with doing metagenomics, we had sure that most papers would do the harvesting of the cells directly from seawater. And most papers in fact requires more than 2 liters. Some in fact requires 5 liters of the water itself. So due to this slight difficulty, we couldn't bring 5 liters of seawater over all the way to Malaysia due to the weight, because we were bringing back all sorts of equipment. We had to in fact lose some. So I was left with only the culture, and I did a what do you call it, from day 0, day 3, day 6, the bacterial change, the dynamic shift there. That was all I worked with,
Okay, yeah, that's really great. Actually, it's already a lot. Maybe if you have any additional possibility, you could also consider harvesting this original community just for future, in case you will have any logistics possibilities, because we all know that Antarctic logistics is tricky, and we have to do the maximum of what we can. Yeah.
Yes, indeed. Thank you so much.
Yeah, thank you.
Okay, thank you, Nadira. Thank you, Maria, for interesting discussion. And now we will go to the next presentation. Syazani, yeah. Please, you invite to presentation.
Okay. Okay, everyone can hear me?
Yes.
Okay. We start now. Okay, hello everyone. My name is Syazani Zaki. I'm a master student from a School of Biological Science, University Sains, Malaysia. Okay, today I will present my research titled The First Discovery of Toxin-Producing Wilmotia murai from Cryopreserved Antarctic Cyanobacteria mats. Okay, now we move to the introduction. Okay, for introduction, what is cyanobacteria? Cyanobacteria, also known as a blue-green algae. Okay, it is the one of the earliest organism on Earth, dated 3.5 billion years ago, and it is the only gram-negative prokaryote that undergo photosynthesis. It's a primary producer in aquatic food web and have a major role in carbon and nitrogen fixation. It also have the ability to produce bioactive compound that act as antifungal, antiviral, UV protectant, and anti-aging. Okay, so we move to the cyanotoxin. Okay, what is cyanotoxin? Cyanotoxin is secondary metabolite that produced by cyanobacteria. They react with different mechanisms of toxicity and target different cell and organ. Okay, the most common cyanotoxin is hepatotoxin, consists of microcystin, nodularin, and syndrospermacin that induce liver damage and kidney failure. Second is neurotoxin, consists of anatoxin and cytotoxin that attack respiratory and skeletal. And last one is dermatotoxin that target human skin. Okay, now we move to cyanotoxin in Antarctica. Okay, cyanotoxin was commonly thought to be peculiar to tropical and temperate region. However, the discovery in 2000 by heat spell in Antarctic made cyanotoxin a worldwide phenomenon. After that, several other discoveries followed the trend, such as Jumblut in 2006, Wood in 2018, Clentac in 2013 and 2014. Then, in 2018, Jumblut discovered cyanotoxin in 100 years old cyanobacteria mat collected during Captain Scott discovery expedition. And the most recent one is in 2020 by Zaki et al. Also using another old sample from the 3 decade. Okay, all these discovery confirm that the presence of cyanotoxin is not peculiar to tropical and temperate region. Okay, for the sampling site. Okay, the sample was collected. Okay, for the sampling site, the sample was collected from Northern Victoria Land in 1984 until 1985. Okay, so this is the overview of my method. Okay, first is the analysis of the sample using light microscopic and establishment of culture for the species identification. Second is cyanotoxin screening using PCR to detect respective gene for the cyanotoxin. And then when the result is positive, I will proceed with phylogenetic analysis to further characterize the species. And then the toxic analysis to quantify the toxin. Okay, for laboratory work, the sample origin, the sample were collected by Dr. Paul Brody from University Canterbury, New Zealand in 1984 until 1985. And then proceed with the analysis of the sample. The sample was conducted using light microscopic by recorded all the detail for morphological features such as trichome color, vegetative cell and width, apical cell shape, present or absent of sheath, and reproductive of cell for 30 replicates. And then the result will be compared with the key book from Comarec and Anelostidis for species identification. Okay, next for the culture establishment. The strain will be cultured in BG11 with cycloheximide to eliminate the growth of eukaryote, and then it be incubated for 2 weeks. Okay, this is for the molecular assessment. So DNA extraction, and then proceed with PCR, and then for positive result, we will proceed to the sequencing. Okay, for the molecular assessment, for phylogenetic analysis, we will be using this 16S, and this is the primer. Okay, and then for the cyanotoxic analysis. Okay, the extracted DNA was screened for 5 cyanotoxin. Okay, for this, but only for microcystin nodularin is being using in same PCR because this one is this gene is homologous to each other. Okay, and for the PCR, I'm using an extra another extra tube for the strain that previously confirmed to be cyanotoxin producer that used as microcystin. This is the list I'm using. Okay, now proceed to the result. Okay, for this research, I got two strain of Wimotia murai. So the first one is Wimotia murai strain MVL 81. So this one the color is olive green. Okay, and then it is straight and slightly straight and slightly curved. And then you from here you can see that the cell is shorter than white to isodiametric, and then the apical cell here is rounded. And then this is the sheath, and then the sheath is colorless. Okay, next we move to the second strain, MBL 95. Okay, this is a blue-green color, but the cell you can see here, the different one is the cell is longer than white, and then the apical cell is rounded. And then, okay, so the difference from previous strain is that this one is, the strain is more longer, the cell is more longer than white. Okay, this is the comparison table. Okay, for the reproduction, both of my strain is absent of necrotic cell. Same with the true Wimotia murai. Okay, you can see here. Okay, now move to the Wimotia murai phylogenetic tree. Okay. Partial 16S RNA sequence of 1,200 base pair were amplified from both strains. And then from here you can see that our two strains are well supported monophyletic clade within major cluster of alder Wimotia murai. And then from here you can see that our strain is clearly separated from Wimotia stricta, and then another one is Wimotia koreana here. Okay, then we proceed to Wimotia phylogenetic tree on microcystin gene. Okay, for the PCR result, we got a positive result for HEP PCR that indicate that our strain contain either microcystin or nodularin gene. So we send it for the sequencing, and the sequence obtained, the sequence result was compared with available sequence in BLAST, and then we got the result that we confirmed the result that present of microcystin gene and microcystin production were evaluated for both strains. So I make these three. So from here, you can see that the tree reveal that both of our strains were identical to those of uncultured cyanobacteria clone and then the result also show the higher bootstrap value for both ML and BI analysis here. So you can see. Okay, the last. Okay, for microcystin analysis to quantify the microcystin concentration, the standard curve was established by calculating the mean absorbance value for each of the standard, and then the analysis of the extract from Wimotia murai confirmed the presence of microcystin at the concentration of 1.6 microgram per liter for strain MVL 0.81 and 0. Okay, that's my mistake. It's supposed to be for 0.19 microgram per liter for strain MBS 95 strain. Sorry for that. Okay, so for the conclusion, Vimotia murai strain show positive in microcystin biosynthesis and contain MCYE gene. And then analysis of the extract from both strain by ELISA confirmed the presence of microcystin LR at concentration of 0.1 microgram per liter and 1.6 microgram per liter respectively. And then species education was confirmed as Vimotia murai using 16S rRNA molecular phylogenetic approach. And last conclusion is that new microcystin producing cyanobacteria strain were discovered in another old sample. So this is my reference. This is the analyzement, and then that's all for me. Thank you.
Thank you, Susanna, for very interesting and brief talk. It's really very actual topic, and in Antarctic especially. Some questions. I need to check chat. No question in chat.
Is there any question?
Okay. If no question, we can go. We can continue, and I invite to presentation. Next speaker. I cannot see in the mosaic of speakers. Dr. Subramanyam.
Okay,
yeah,
thank you.
You are welcome.
So I hope you all can see my slides.
Yes, please go on.
Okay, all right. All right. Greetings to all. My name is Kavilashni, from postgraduate student from Universiti Putra, Malaysia. So my study is on development of defined mixed culture system for effective remediation of phenol at low temperature. So this is the outline of my presentation. Okay, before moving on to the main topic, let me introduce on what is phenol and why it is a matter of concern. Phenol is naturally obtained from coal tar or as a degradation product of benzene. It also can be produced synthetically. Phenol is an important industrial precursor to many materials. For instance, phenol is used in manufacturing of many household products like detergents, disinfectants, insecticides, also in cosmetics and pharmaceuticals products. It is also a major component in fuel oil. Evidently, phenol is detected in various industrial effluents such as oil refining, petrochemicals, pharmaceuticals, resin manufacturing, plastics, and also municipal wastewater. Phenol is a colorless and crystalline solid with low melting point, and it has the deliquescent properties where it becomes liquid when exposed to air. It is soluble in most organic solvent and sparingly soluble in water. It is widely used in pharmaceutical products and household products due to its antimicrobial properties. Okay, phenol has been classified as highly toxic priority pollutants by the US Environmental Protection Agency and also European Community. In human, phenol causes eye and skin burning due to its corrosive nature. Short-term inhalation of phenol vapors causes headache, And long-term inhalation results in coughing, cyanosis, and also lung edema. Consumption of liquid with high phenolic contents for long-term causes serious diseases which affect endocrine, cardiovascular, and also muscular systems. Though reports on phenol toxicity on Antarctic animals are scarce, studies are reporting that phenol generally tends to cause respiratory distress in fish and also affect reproduction and fetal development in mammals. Similarly, phenol generally affect the chloroplast in plants, which cause them to lose their chlorophyll and affect their photosynthetic efficiency. Now, we know that Antarctica has extremely harsh and cold climate with 70% of Earth's fresh water. The extreme climate of Antarctica becomes a major challenge for natural processes involved in remediating pollution. As a result, pollutants can bioaccumulate in Antarctica's simple ecosystem and their food web. So these are some of the examples of occurrence of phenol pollution in Antarctica. All right. Phenol can be removed by physical and chemical means as shown here. However, biological remediation is preferable due to its cost effectiveness, eco-friendly nature, applicability, and also it usually generates non-toxic end products. Okay. In recent years, studies on mixed microbial cultures over single species have attracted research interest due to their advantage of being far more effective. Mixed culture has a greater metabolic capacity, which enables it to show higher degradation rate, and their synergistic interaction among members results in greater metabolic capacity. So these are some of the examples on mixed microbial culture method that use mesophiles. However, this method on Antarctic strains are still scarce. So this is the aerobic pathway of phenol degradation by microorganisms, where phenol is broken down into catechol by phenol hydroxylase enzyme, and this catechol is further break down into substrates for TCA cycle, either using meta-cleavage pathway or auto-cleavage pathway, depending on the enzyme they have. So the problem statements of my studies are: phenol is a major pollutant that could be found in Antarctica. Natural biodegradation of phenol in soil under cold climate conditions remained a challenge. Most phenol bioremediation were achieved using pure culture, and mixed microbial culture methods are still lacking. Hence, this study may provide the knowledge gap in regard to the phenol degradation studies by Antarctic bacterial isolates. Objectives of my studies are to identify potential defined mixed culture of indigenous bacterial strains with phenol degrading capacity from the Antarctic soil, and to determine the optimum conditions for phenol degradation by defined mixed culture using statistical analysis. So this is the workflow summary of my study. I used 3 different Antarctic strains that have already been isolated and identified as phenol degraders. These bacterial strains then mixed in both equal and unequal ratio to see their interaction, and inoculated in phenol media with 0.5 gram per liter of phenol as initial substrate. And this phenol is the only sole carbon and energy source in this medium. The samples were then analyzed spectrophotometrically to detect the residual phenol concentration. During the end of degradation process, PCR restriction fragment length polymorphism analysis was performed to confirm the presence of all strains in the consortium. The DNA of both pure and mixed culture was extracted and amplified using PCR. Then these purified PCR products were then digested using 2 different restriction enzymes, and the end products were analyzed by electrophoresis. Then the phenol degradation process was optimized using statistical approach. Total of 30 runs with different conditions were generated following the central composite design to identify optimum response and to obtain a predictive model. Then different initial phenol concentration was tested to identify the possible highest concentration this culture can degrade at optimum condition. So this is the result from the preliminary screening. Different ratios were used because inoculum ratios plays a crucial role in biodegradation, as the response could be attributed to the interactions among bacterial species. So this mixture performed effectively when presented in equal ratio, and not so equal ratio shows a slight delay in the degradation process. As you can see from the table, invariably these three strains can degrade initial phenol concentration of 0.5 gram per liter within 120 hours. But when these three strains were combined, they could degrade phenol effectively within 60 hours. So this is the PCR RFLP analysis to confirm the presence of the strain at the end of degradation process to ensure the presence of these strains. So this bands indicates DNA with different molecular sizes, showing that the same size for the pure culture with the same size of band present in the mixed culture, showing that this strain presents in the consortium, showing that all these three strains present in the consortium at the end of degradation. So this is the statistical optimization study result. This table shows the response obtained in different conditions generated by central composite design. From the result, it can be observed that optimum phenol degrading process achieved when ammonium sulfate is at 0.4 gram per liter, which I've highlighted in red color, and salinity at 0.13 gram per liter, pH at 7.25, and also temperature at 12.5 degrees Celsius. This is the summary of the analysis of variance for the result obtained. This ANOVA result shows that the model is well supported with significant model F value and insignificant lack of weight. And these models predicted that ammonium sulfate concentration, NaCl concentration, and temperature being the significant factors. So this is the 3D surface plot generated from the statistical study. This 3D surface plot demonstrates the mutual dependence between significant interactions while maintaining other factors at constant level. The first interaction shows maximum degradation can be achieved when ammonium sulfate concentration is between 0.35 to 0.5 gram per liter, when the pH is at 7.13 to 7.38. And the second interaction shows that maximum phenol degradation achieved when the pH is at 7.13 to 7.38 and the NaCl concentration is between 0.11 to 0.15 gram per liter. So at optimum condition, different initial phenol concentrations were given as a sole carbon source, and this shows that this mixture of strains could degrade 1.9 gram per liter of phenol completely within 8 days. And when the concentration increased to 2.1, the degradation ability decreased, and in the study, bacterial growth depleted as well, showing that 2.1 gram per liter of phenol act as a become inhibitive. So that's all for my study. The conclusion is defined mixed culture of indigenous bacterial strain proven to be effective in phenol bioremediation in laboratory scale compared to pure culture. And the optimization study shows that this defined mixed culture is cyclotolerant, prefer near neutral conditions, and also low salinity. And this DMC can degrade phenol concentration up to 1.9 gram per liter, which is for phenol, it's considered high for now. For now. So these are the references and acknowledgements. Thank you.
So this is end of your presentation, I understand, right? Oh, okay.
Yep.
Okay. Thank you very much, Ka Vilashni. And yes, I really impressed that you have such big spectrum of bioremediation work with very different pollutants. Yeah, it is absolutely very important in Antarctica because a lot of pollution with modern technique level, yeah, and we need to work with this. And we have one question in the chat. Probably for you, more better to see chat now. Dear Dr. Kavyoshny, from your experience, what would be the complication of phenol remediation in the wild? The compound is rather volatile.
Oh yeah, my focus is basically for bioreactor as well, because most of the phenol compounds present in the household wastewaters. So before releasing into the environment, we would like to remediate the contamination in the wastewater before releasing it to the wild. So I think the phenol is already soluble in the wastewater. So for the volatile properties, I don't think so it become a matter of concern.
Okay, and I have probably Maria. Okay, go ahead.
Yeah, I just have a very short question. Yeah, thanks a lot for your presentation. I just wanted to clarify. So you are doing the experimental community, and your main task is to make like the bioreactor in order to process the waste, so that phenol does not enter the environment afterwards. Yeah.
Yes.
Okay, because when I looked at your data on showing that this community is not tolerant to salinity, I just thought that if it could be efficient in the ocean, for example, but if you're working with the purpose to prevent its release in the ocean, then it's perfectly clear. Yeah.
Because these isolates were extracted from soil, so they show low salinity. Yeah.
And was it coastal soil, or how far was it from the ocean?
It's not from a coastal soil, but it's near to research stations.
I see. I see. Okay. Yeah. Thank you. It's clear.
Okay, and I have probably another questions. Please launch and ask if you want. Okay, I have general question or some opinion. Will you plan to use your experience in preparing of some Antarctic treaty protocols, some instructions to prevent pollutions, to fight against pollutions, because you have a lot of experience? Will you work with this? Probably this question to Aqlima. Or you can ask, there's no problem. Okay, I understand you. Aqlima, are you here? Okay, more later. So.
Hello again. You're calling my name, is it?
Yes.
Okay, what is question?
Yes.
Sorry.
I asked about one very important thing. You have a lot of experience with work with pollution, yeah? Will you plan to work with some practical documents of Antarctic treaty system to produce something new to prevent pollution, yes, and fight against pollution?
Yes, we plan it in the future. Because of that, we try to, if you see right now, most of my students just focus on the single bacteria or community bacteria or single algae or community algae. Maybe later we want to join, do the combination of the algae and the bacteria, and then maybe we can prevent the Antarctic. In the future, yes, we have a plan. And then we have maybe if the Ukrainians want to join the collaboration, yes, please text me. Maybe we can talk more, because because for Malaysia, actually, we don't have a station. It's a little bit difficult for us. But luckily, we have a good collaboration from British and also and Chile. So we get the opportunity to go there and do the remuneration there.
Okay.
Hopefully, we can go again.
Okay, thank you for comment. And if we have not questions, I think Dr. Muhammad Fauzi wait for his presentation. Go ahead.
Hello. Let me share my screen. Give me a second.
I'm sorry. I'm sorry. In my plan is the next speaker is one moment, please.
It's okay. The 11th speaker is Nuradilla Muhammad Fauzi. It is she. Yes. Do you hear me, Ivan?
Nuradilla Muhammad Fauzi. Okay.
Yes.
Thank you.
That's me.
Sorry, this is my phone.
No, it's okay. Maybe you thought I was a man. That's okay. Because that's my father's name, actually. Let me share my screen real quick. Okay, can everyone see my screen and can everyone hear me?
Not yet.
All good? Okay. Hi, everybody. Thank you for the invitation. It's really a pleasure to be here with everybody, to be able to share my research with all of you. My name is Nuradilla Muhammad Fauzi, as you've heard, but you can call me Adila for short. It's a very long name. And I'll be talking about the transcriptional response in polar pseudogenaescus soil fungi following thermal challenge. All right, so just a little bit about fungi in polar soils. If you were here yesterday, when Dr. Pete Convey was presenting, he talked a lot about the terrestrial ecosystems and how soil is a huge part of the Antarctic terrestrial ecosystem. And in polar soils, there are fungi as well. And we all know that microbial processes are very crucial to the soil ecosystem. And soil fungi actually constitute quite a significant proportion of the soil microbial biomass. And what we know about the Antarctic and Arctic soil fungi is that they do perform a wide range of ecological roles, and many of them have been shown to perform saprotrophic ecological roles whereby they actually can degrade organic material in the soil. And there was a paper that basically said that soil fungi actually may play a more extensive role than bacteria in the redistribution of carbon and nutrients over spatial scales. So it is quite important to study fungi in the soil, but unfortunately in polar systems and compared to bacteria, fungi do lack comprehensive characterization and also functional studies. So if we don't have these information, this sort of information, we cannot link community composition to what those individual counterparts actually function within the ecosystem. Okay, and it is said that even within, as it is equally important to study community composition as well as species-specific physiological functions, so that we can actually take into account how, at the species level, what kind of changes to ecological roles that may be translated into nutrient cycling within the soil. And towards the end, we were interested in the genus of fungi known as Pseudogenaescus. And if you've been around in the fungi world for a while, they used to be known as geomyces. They're known psychotolerant. They can grow in a wide range of temperature, and they're actually commonly isolated from soil. in both the Arctic and Antarctic. Even very recent papers from the past few years where they've done more comprehensive diversity studies using metabarcoding, they have found Pseudogynascus to be the most dominant, say, in the soils of Deception Island. And this particular genus is actually quite well distributed in other substrates as well, including lake sediments, glacial ice, and also permafrost. And because they exist both in both poles, in the Arctic and Antarctic, they could serve as a model for you to make comparisons between the poles, if you like. And based on studies, including studies done by our lab, they're also known to secrete high levels of extracellular hydrolase enzymes. Therefore, we stipulate that they may play an important role in soil biogeochemical cycling as decomposers. And the relative activity of these enzymes have been shown to be temperature dependent, but the molecular basis of that is unknown. So there might be some implication for soil warming due to climate change, knowing all of these bits of information. And thankfully, compared to other environmentally isolated species that are not well studied, there is some sequence availability in Pseudogynascus because they're quite ubiquitous, and some species are actually known to be pathogenic. I'm sure some of you may have heard of the white nose syndrome in bats that are occurring, and actually they're decimating a lot of bat populations in the Northern America especially. And there is some sequence information. The genomes of some strains have been sequenced but poorly annotated. But nevertheless, we have protein predictions. And because of that, we can use protein blast with known protein sequences from the model fungus Saccharomyces cerevisiae, which we all know as the budding yeast. So we can use these tools to potentially identify molecular markers that we know are involved in heat stress. Okay, and that's HSP70 and HSP90. So we're interested, okay, if we want to know how climate change associated warming might modulate the physiology of Pseudogynascus, we have to characterize how they respond to temperature. And of course, as I've said, HSP70 and HSP90 are known molecular markers for heat stress. They are protein chaperones. They help disaggregate and they help fold proteins that get misfolded due to heat stress. But they also occur they also occur in response to multiple stresses, not just heat. And they also do a lot of important functions within the cell in both stress and non-stress conditions. But what's most important is these are very conserved proteins. They are equivalent genes, or what we call orthologs, that exist across species, even as humans and even in bacteria. So, so we're going to look at HSP and HSP90. We decided to look at two strains from either both the Antarctic and the Arctic. So we have strains coming from King George Island from the Antarctic and Horsen in the Arctic. And what we did was we subjected them into a transient heat experiment, whereby we cultured them in liquid broth, and then at day 5, which is the log phase, we transferred them to 25 degrees Celsius for 2 hours. So it is a transient treatment. 2 hours is not very long. The reason why we chose 25 degrees Celsius is because based on previous studies done in our lab looking at morphological data, growth rate, and energetics, signs of stress have been seen occurring at 25 degrees. So we decided to compare the controls that remained at 15 degrees and the treatment group that was exposed to the heat stress temperature for 2 hours at 25 degrees Celsius. And then when we looked at the RNA and the gene expression occurring within these samples that have been heat treated, we saw that yes, there actually was a difference in the expression of HSP70 and HSP90. just looking at gene expression analysis through quantitative RT-PCR. So this way we know that, hey, heat stress is occurring, okay? And if we look at there is some minute differences, but the differences between the Antarctic and Arctic strains are actually not significant. So this basically allowed us to think that, hey, maybe we should do RNA sequencing to have a more global look at the pathways that are involved in response to thermal stress, because HSP70 and HSP90 are just a couple of genes, but what's actually happening within those cells, those fungal cells. So what we did was we chose one of the strains, and we chose the Arctic strain, and then we submitted 3 replicate samples for the treated and control. We submitted them for RNA sequencing using the Illumina HiSeq platform, and then assembled the transcriptome de novo, and then did some bioinformatics analysis to the transcriptome. And what we found that, looking at the transcripts, there are actually 52,000 transcripts representing 23,000 unique genes that were found. So that's actually a lot. Now let's look at what they're actually doing. So we looked at the transcripts, and if we looked at the functional annotation of these unigenes, all 23,000 of them, we can see that all of these genes actually involve many processes, but if they're actually represented by a greater extent by processes involved in energy production, translation, ribosomal structure, and biogenesis. So those are processes involved in proteins, okay, as well as post-translational modification, protein turnover, and chaperone. So these are also protein-related processes, and also some general functions involving biochemical activities. And you can see that metabolism is also well represented here. So we kind of have, from here we can get a sense of what these genes may actually be doing during the heat treatment. But these are actually talking about the genes, the transcripts that are there within the cells. But out of those 23,000, almost 3,000 of them are actually differentially expressed. So what that means is if we look at the difference between the heat treated and control samples, 3,000 of these genes are actually differentially expressed. Now this is a heat map. If you've seen publications showing you RNA-seq data, they will show you a heat map. If the color is green, that means the gene is downregulated. If it's red, it means it's upregulated. So out of these 3,000 genes, about 1,500 are downregulated and 1,400 are upregulated when we compare the treated and the control. But again, because even 3,000 is a lot, and basically if you can't see it from here, but every horizontal line represents a gene. So this is stuffing 3,000 genes within one figure. So it's really hard to kind of figure out just from this, like what's going on, but at least we can see that between the replicates, they're all showing the same things. Now we look a little bit deeper and look at what pathways those differentially expressed genes are actually involved. We can see that the pathways that are represented are pathways involved in protein translation, as well as pathways involving metabolism of amino acid, carbon, and lipid. Okay, but again, like I said, 3,000 genes is a lot, and then we were interested in, how many of these genes are actually involved in environmental stress response. Because environmental stress response is actually a network of genes that respond to all kinds of environmental stresses that have been well described in budding yeast, Saccharomyces cerevisiae. We know that in yeast, every time ESR happens, about 300 genes are upregulated or induced. And these are genes involved in metabolism, cell wall modification, DNA damage repair. So you can see that these are all processes that seem to be helping the cell prime itself and respond to the stress. And then in return, about 600 of these genes are actually repressed. They're actually down-regulated during stress response. And these are genes known to be involved in, again, protein synthesis, ribosomal genes, cell growth, and also RNA metabolism. And the landmark paper that actually described ESR in yeast is actually this 2000 paper by Audrey Gash. And in later years, it was found that if they looked at ESR between different yeasts, so different fungi, the differences that people saw were actually consistent with lifestyles that are actually species specific. So so basically looking at differences in ESR may actually tell us a little bit about how different species of fungi actually behave in response to stress, and that's in relation to their lifestyles. So when we looked deeper and basically tried to see, okay, among the 3,000 differentially expressed genes, how many of these genes are actually part of ESR as has been described in yeast. In those, basically we looked at two different yeasts, Saccharomyces cerevisiae and Clybury. And we found that about 175 out of the 3,000 genes that were differentially expressed in our samples are actually genes that are involved in environmental stress response. Okay, and out of these 169 genes, about 111 are down-regulated and 64 are up-regulated. So over here, you can see the heat map a little bit clearer. The colors, so green is, again, green is down-regulation, red is up-regulation, and each horizontal line represents a gene. So you can kind of see the different gradations of color based on the intensity of changes in gene expression, but you can see that between replicates, they're all relatively equivalent. And if we looked at the genes that are actually down-regulated, there are ESR genes that, again, just like I've mentioned and what we've already known, there are genes involved in ribosomes and translation. So basically what that tells us is during heat stress in Pseudogymnoscus, we know that protein synthesis is also inhibited, just like what has been described in not just other fungi, but also other organisms. If we looked closer at all the individual genes that make up the ribosome, you can see that many of these individual genes are actually down-regulated. So you can see that many of these boxes, which represent different genes that make up the ribosome, are highlighted in green. That means they have been down-regulated in response to the heat treatment. And what's up-regulated? Well, there are genes involved in metabolism of amino acids, carbohydrates, and lipids. So it is the same story. So we now know that in Pseudogymnoscus, truly during heat stress, cellular processes that are required to generate more metabolic energy are actually increased. And now this, these observations are actually also consistent with what has been described at the protein level in various fungi in response to temperature stress. And this has been nicely reviewed by a colleague of ours, Norliza Abubakar. I think her poster will be up tomorrow. So if you're interested in looking at this observations at the proteome level, you can check out her poster tomorrow. And the reason why these processes are upregulated and downregulated is because it's a trade-off, right? The fungi needs to compensate for the amount of energy that they need.
You have time to finish?
Okay, I'm almost finished, yes. Sorry. So basically they need to compensate for whatever extra energy that they need to deliver the stress response. And just to really quickly finish, if I looked, if I picked genes that were at the top of basically the most differentially expressed, these are genes that are known to be induced in response to other stresses in yeast. And these are also, one particular gene is known to be involved in breaking down xylose and arabinose. And these are also sugars, polysaccharides that are known to be utilized by many Antarctic microbes. So that, begs the question, are these sort of sugars available in Antarctic soils as well? And as we know, Antarctic soils are not very nutrient rich, but perhaps it makes us think, like with climate change and everything and changing distributions of plants, that might be something that will change. And we also know that one of those genes are involved in spore wall assembly in ascomycete fungi. So that gives us a glimpse of, hey, perhaps there is some mechanism that's involved in adaptation as well. And one of these genes that's downregulated. This gene is implicated in translation termination, so terminating protein synthesis, and it's also conserved in humans as well. So this is the same thing that we see in humans. So there is a fair amount of conservation. And the reason why studying ESR is important is because it is the same network of response and the same genes that are involved in responding to not just heat shock, but also other stresses as well. So they're all connected. And there are, I won't go through this because I'm running out of time. So in conclusion, our findings show that short-term exposure to thermal stress in polar pseudogenescus, so basically, sorry, I wrote something wrong there, but basically we're showing short-term exposure to thermal stress do induce heat stress in polar pseudogenescus. That's what I wanted to say. And it has been shown that even in polar soils, even in Antarctic soils during midsummer, temperatures do fluctuate and do near or exceed 20 degrees Celsius. So that, again, it raises more big questions for us. It is preliminary evidence of a similar ESR program in pseudogenescus. So this might be translatable to other Antarctic soil fungi as well. And again, bringing up more possibilities of future studies, perhaps community level transcriptomics or looking at long-term stress. So I'll finish up. These are all my lab mates. I want to thank Dr. Goh Ho Han because he helped out with the bioinformatics. And thank you. Sorry I took a long time.
Okay, thank you very much. Maria, I know that you have questions.
Yeah.
Probably very short.
Yeah, that's true. I have a very short question. Thank you for the inspiring work. It was really interesting to listen. I wonder just, so you made your analysis on the samples that were subjected to heat stress, and then immediately they were snap frozen, yeah? Immediately after the heat shock. And did you think if there will be any sense to analyze these samples after the heat shock, for example, to observe this culture for some time after the heat shock and to observe the control structure at the same time to see the resilience of these communities and to see how they revive after the heat shock, if they behave differently and when this protein expression will come to some normal, to the same as in control group.
Right.
Did you think
so
about this? Does it make some sense?
Yes, it does make sense. So I think what you're referring to is stress acclimatization.
Mm-hmm.
Yeah, so that's a really great question. And that was actually the next thing that I wanted to look at, because what we're showing here is just the transient. So the reason why I wanted to look at the immediate response is within the transcript level responses occur really quick. But if that's acclimatization, because we don't know anything about these fungi, so we actually don't know how long they take to acclimatize. But actually, the work done by Nurliza Abubakar, so the one that I mentioned, she did look at these many of the same strains of the same genus, Pseudogenescus. She looked at the differences in their proteome when they were cultured for 5 days at the stress temperature compared to 5 days at the control temperature. and she did see differences as well. But it is not, to answer your question by not answering it, it's not known how long they will take to acclimatize, and that's sort of one of the things that needs to be done in order to characterize that.
Yeah, it could be really, really interesting in terms of the adaptations of these ecosystems to climate change to see what is next, actually. Now we know what is happening, and it's interesting to see what is next.
Right. Right, like do they acclimatize? Does it become worse? Does it go back down? And the other thing we're interested in to see is how these gene networks actually connect to the genes that are involved in decomposition processes, because we do see that there are differences in activities of extracellular hydrolases, but in terms of the biochemical pathways, we don't yet know how they are connected. But yeah, that's a good thing to point out. Thank you.
Yeah, thank you. Thank you.
Okay, thank you, colleagues, for this discussion. And I will invite for the next speaker about next speaker, Rizman Edith, present?
Yes.
Oh, you're welcome.
See, hold on. Thank you, Ivan. Can everybody hear me?
Yes, yes.
Yes? Yes. All right. Okay, I'll try to share my screen. Share. Okay, I hope it's in a big screen. All right. All right. Thank you, everyone, and especially for the organizers for allowing me to give a talk. And basically, my talk is part of our previous presenter, because we came from the same lab, and I was more in charge of identifying the species. So basically, this talk is going to be about the molecular identification of Pseudogymnoscus species that were isolated from Arctic and Antarctic regions. And this is actually a collaboration between my institute, which is Institute of Ocean and Earth Sciences from University of Malaya, and with National Antarctic Research Center, and also British Antarctic Survey, and also Institute of Nature Conservation in Poland. And basically, in this sort of project of ours, we are trying to look into the diversity of soil microfungi and from the polar regions. So therefore, we did go and collect from Arctic and Antarctic as well. So as an introduction, again, I think it was pretty much well covered by our previous speaker, that soils from both regions tend to have almost similar soil fungi communities. And actually, about 60% of the operational taxonomic units that has been reported from both regions actually come from the class Lithiomycota. And among the most common lithiomycetes, member of the genus Pseudogymnoscus are frequently reported in different substrates in both in Antarctic and Arctic, and also actually in some temperate regions, and they are all mostly found in soils. And as what Adila did mention, when it comes to Pseudogymnoscus, I think later on I'll tell that there are some which are pathogenic as well. Now back to some work that has been done on fungi, and I'll try to focus more of what we as a group from University of Malaya and also from Malaysia, basically has done on soil fungi in the polar regions. So in Antarctic, we know that not much has been done. Particularly, most of the work that has been done is more on the continental rather than the maritime Antarctic. And one of the reasons is perhaps because of difficulty or challenges to get access to such areas. And nevertheless, there are many reports describing the occurrence of fungi, and a lot of it is associated with human habitats and also other human artifacts that has been studied extensively. And again, most of them also belong, about 80% belong to the Ascomycota, class Ascomycota, mostly perithecial with few discomycetes. So here are some examples of species that are commonly associated with the polar regions, such as Mochirella, Antarctomyces, Murachia. You've got several types of yeast, Oreobasidium, and Tilibulus species. And when it comes to polar fungi, usually you classify them either as psychrophiles, psychrotolerant, or mesophiles, according to their ability to grow on the different temperatures. So if they are able to be growing at 4 degrees Celsius, then we call them psychrophiles. If they are able to grow in both incubation temperatures, which is higher, then they can be psychrotolerant, and usually isolates that grow at 25 degrees Celsius are called mesophiles. And when it comes to Arctic fungi, I think, again, compared to a lot of tropical and other parts of the world, both of the polar fungi are still many that have yet to be discovered. But nevertheless, a lot of progress has been made, particularly some works by Singh et al. They have managed to find 14 genera from Nielsen in Spitsbergen soils. There has been interesting marine fungi that has been also isolated from wood debris, which are collected in Longyearbyen, done by Pang et al. And also some samples of ornithogenic influence soils from Harrington, which shows a lot of diversity, especially in terms of Mochirella, Cudonella, Perisporium, Bouveria, Geomyces, Penicillium, and so many others. And for our talk, or for my talk today, I'll be focusing on Pseudogymnoascus. And actually, Pseudogymnoascus was commonly perhaps identified as geomycete. So some of the identification has actually been changed. So there is actually geomyces, and there is this new species or new genus called Pseudogymnoascus. And as far as the common species that we associate with Pseudogymnoascus, there's this 10 species that has been described morphologically. There is Pseudogymnoascus roseus, vinaceus, bati, caucasus, alpinus, appendiculatus, verrucosus, destructans, panorum, and Pseudogymnoascus carnis. So these are the more, I think, more I would say more well-known. So in terms of Pseudogymnoascus, they are psychrotolerant and psychrophiles. And actually, for most of our lab experiments, we found out that they can actually grow at 10 to 20 degrees Celsius. And they are actually commonly found in soils, from temperate up to polar regions, and some of them, could also be found in caves. And as what Dr. Adila showed earlier on, those are the ones that has evolved and became pathogens to bats and caused the white nose disease. And they produce actually various extracellular hydrolytic enzymes, such as amylase, cellulase, keratinase, lipase, kinase, and urease. So a lot of our work in our lab has been using Pseudogymnoascus to look into various questions in terms of adaptation and response to various temperatures or ultraviolet or any stress response, and looking into their enzyme production and so forth. And nevertheless, I think it's quite important to know that we are using the right species, and I think identification of a lot of our fungi is quite important before we proceed further with any experimental work. And usually for barcoding, we tend to use the ITS or internal transcribed spaces because it is able to amplify the broadest range of fungi, and there is a lot of data that we are able to compare with. Nevertheless, there are several other genetic markers, such as the 28S nucleoribosomal large subunit RNA gene, or commonly known as LSU, the 18S nucleoribosomal small subunit RNA gene, SSU, subunits of RNA polymerase, RPB1 or RPB2, and also mini chromosome maintenance protein, MCM7. So if just like want to show that some of our work that we have done in terms of fungal isolates, and some of these actually isolates we previously tried to identify by molecular, just to help out in identifying and also organizing our culture collection, which we keep in the National Antarctic Research Center. So we do have some selection of Arctic isolates and Antarctic isolates, and for this particular talk, we are more interested in those that was previously identified by S-geomycetes. So basically, we try to identify these species, which now we know most of them actually most probably belong to the Pseudogymnoascus strains. And just for this particular talk, I'll be focusing on our application of ITS, LSU, and MCM7 to identify those selected species. And for this study, we have used 4 strains of Pseudogymnoascus, 2 that were obtained from Antarctic, from King George Island, and these were, as I said, previously isolated by our students, and those other 2 which are also isolated, but actually it's from Ponson, coming from Arctic. And these are the isolates that we have kept in the culture collection of the National Antarctic Research Center, University of Malaya, and some of the students that are working on this will have to revive them. So here, a lot of our samples, we depend on our collaboration to have access, and this is Mikla, is our collaborator from Poland that helped us to get access to samples in Hornsund. And in terms of methodology, the fungal isolates are basically isolated from the soil using the walk-up soil plating technique. This is also done basically from our previous lab assistants and students who worked on it. So once they are incubated at 4 or 25 degrees, depending on whatever conditions that we wanted. But in our particular work now, we are trying to grow them in ZPEC DOCs or CDA plates as individual isolates and at 4 degrees for 8 days. And from these isolate, we try to amplify, extract the genome, do the PCR, do the DNA sequencing, do the blast searches and subsequent phylogenetic analysis. So this one is just to show that previously, most of our work, we did it on potato dextrose agar. Now we found out that they seem to grow better in CDA agar, possibly because this medium contains a lot of sodium nitrate, sucrose, and other sources of essential ions, which seem to be beneficial for the cultivation of fungi. So when it comes to polymerase chain reaction, these are the markers that we have decided, ITS, LSU, and NCF7. And these are the primer sequence that we have used. And these are the conditions that we used to amplify those 3 markers. Now, when we send the products to a company for sequencing, and once we got the sequences, when we analyze them, we find that some of the sequences best match with geomycetes from the database, from GenBank. But later on, with several analyses, and also because there has been some reclassification, we have decided that they are actually, in fact, pseudogymnoascus, especially following by references by Minis and Linda, 2013. And looking at those 3 markers, we find that ITS and MCM7 are the most phylogenetically informative, because you will have about 15% of parsimony informative sites for the ITS, and MCM7 will obtain about 35% of informative sites. So at this, for that length of sequence, you get a substantial amount of signal, phylogenetic signal. So with the phylogenetic analysis, so in this, I'm showing you the Bayesian phylogram, and we have about 67 taxa that comprise of pseudogymnoascus, geomycetes, and leuconeurospora that was used to root the tree. And these basically phylogram, and we also put a lot of different analysis, the bootstrap values of different analysis, because we did table joining, we also did maximum parsimony, maximum likelihood, and the Bayesian analysis. So what we found is that in general, when using ITS alone, the phylogram failed to group the taxa like those in Minis and Linda. Nevertheless, you can find the 6 pseudogymnoascus clades from A to F, but at some points the clades are not well supported. And again, we find, like for example, like clade B, you have different species falling in that same clade. For example, P destructans and P verrucosus. In particular, of our interest, if we look at clade C and E, you will see that a lot of the species are unresolved. Some of the branches are not resolved. There's a lot of polytomy there, and many undescribed pseudogymnoascus species. And unfortunately, our strains also fell into that clade E, so it was hard for us to determine what species they are. And out of all of that, clade D and F resolved well resolved and well supported.
Much time.
Yep. And for this combination tree, yeah, ITSNL, CUNMS, CEM7, we find that a better resolution was obtained, and we had a lot more clades that resemble those in Minis and Linda. And unfortunately, our samples came at clade B, and unfortunately, we couldn't identify what they are because most of the references also were not identifiable. Yeah. So again, when it comes to pseudogymnostic species, there's a lot of them which are undescribed in GenBank. And so I don't think I need to show this because basically that is the reason for A, B, and C. Those clades seems to have difficulty because the barcode gaps is not as evident as compared to the other clades. So in terms of discussion, I think ITS alone provided very poor resolution for pseudogymnosticus, and we find that a lot of the species are scattered throughout the trees, and some of these patterns are found in Linda as well, Minis and Linda, when you use ITS alone. So therefore, there is a need definitely to use a combined markers, Now, we only use 3 markers, but when we compared to Linda, we didn't include the RPB2 and TEF. So therefore, we believe that by adding those 2 markers, you might actually improve the resolution. And again, there's new papers that came up by Zhang et al. That there has been like 3 new pseudogymnostic species that probably we should consider to include in our future analysis. And I think, yes, again, there's evidence to support the suggestion of including the elongation factor and RPB2 to enhance the resolution. So again, conclusion is that the resolution of the tree will rely on many genetic markers. We need to incorporate probably newly described pseudogymnostic species in future analysis. And again, there is a lot of potential to find new species or pseudogymnostic species from the polar environment, especially when not many are available in GenBank. So with that, thank you very much for your patience, and I hope I've done justice to this. Okay.
Thank you for you, Muhammad. I have not question in chat, and only very small remarks that we're probably interested in more wide spectrum of terrestrial fungi. And do you interested in an identification of some macro fungi or different group of micro fungi in terrestrial in Antarctic terrestrial communities?
Well, most of our group actually we are focusing on soil, microfungi basically, a lot of it, because our setup is pretty much into microfungi. And again, it's much more simpler to sample. And also, most of our work has been isolating from soils, sometimes from ice. So I think depending on those kind of substrates. So I think it's very rare that you would find a macrofungi in the polar regions.
Okay.
Temperate maybe.
We'll be in contact. Thank you.
Yes.
So I invite the next speaker, Mr. Wong, Mrs. Wong, Miss Wong. So presentation about molecular chaperones.
I apologize. Could you switch on your microphone, please?
Okay, can you hear me?
Yes.
Please go on.
Yes.
All right. So I shall start. So hello, everybody. My name is Michael. I'm from the Biotech Research Institute. So today I'm going to share a bit, just a bit more about adaptation to thermal stress, focusing on yeast cells rather than fungus. We have heard about fungus. Let's look about a single cell, which is a Gliciosima antarctica. Actually, we want to focus a bit more about the molecular chaperones and how it helps the yeast to cope with the heat stress. Okay, I'll just give you an outline. So briefly, I will talk about thermal adaptation in general and some of our studies on cold adaptation, especially when the yeast is exposed to freezing temperatures at 0 degree and minus 12 degrees Celsius below the freezing point. All right, and then the main part of the talk will really talk about how the yeast Gliciosima antarctica will cope with the heat stress at 16 degrees Celsius and 20 degrees Celsius. So we have both the cold adaptation and heat adaptation, but I would rather, we'll focus more on the molecular chaperones, how it helps the yeast to cope with the heat stress. Okay, just a bit of background about this yeast. Yeast are basically very commonly found in polar regions. I think Dr. Siti and also Dr. Usman has already presented some of the data about different fungus as well as yeast in the Antarctic. So basically we will want to focus really on the Gliciosima antarctica, PI12, that was isolated some years ago by Malaysians from the sea ice, all right? But Gliciosima basically is found in various polar regions as well as other regions as well. And most commonly, they are isolated from sea ice, fresh water, even the peat bog from Russia, and the marine water samples. And various reported, the first one reported was since 1969. And in recent years, the more and more were isolated from various different geographical locations. It seems that this particular yeast cope very well with thermal stress, regardless of whether it's very cold or sometimes a slightly warmer climate. So let's look at where they are found. Okay. So they are found with different geographical locations, such as the King George Islands and the Weddell Sea, as well as on the terrestrial environment in Davis Stations and Victoria Land. And the PI12 that we are studying is basically collected from the sea ice next to, very near to the KC station. And if you look across the board, you can find that the King George Island is basically slightly warmer regions compared to the other part of the places. So this makes this yeast quite an ideal one. It's scattered everywhere. It is sometimes found in the deep sea of 100 meters below the sea. And at the same time, it's found in sea ice as well. So they are quite robust in the sense of survival. So just a bit more information about Gliciosima antarctica. It was previously known as Leucosporidium antarcticum. Since the incorporations of molecular techniques for identification, they have been reclassified, and now it's called the Gliciosima antarctica. And like I say, it was isolated in various locations in the Antarctic, and this particular PI12 strain was isolated from sea ice. Okay, now a bit more about the temperature that they can grow is, and thank you, Dr. Usman, for giving a very good overview about what is psychrophiles and what is psychotolerant. So Gliciosima antarctica is basically a psychrophile. It goes best at 12 degrees Celsius, all right? It can go maximum at 19 degrees Celsius. At 20 degrees Celsius, it is not growing anymore. Basically, they just die off at 20 degrees Celsius. So you see, it's quite a wide range, but yet it cannot go beyond 19 degrees Celsius. Okay, so our interest is really to look at its thermal adaptation strategies. And interestingly, in one of our studies at incubation at -12°C, we found that the Basiosima antarctica is remain active at -12°C. This is something rather interesting. Okay, the whole genome of this yeast Basiosima has been completely sequenced by our colleague and it's been published recently. So basically, it has a estimated genome size of 20 megabase, and total predicted the gene that is expressed is 7,857 genes in total. Unfortunately, our colleagues is not be able to assemble them into specific chromosomes, but it rather end up with 21 scaffold. That was using the old technology that is 454 and the Solexa. I mean, that was about 10 years ago. But in recent years, I mean, if we can get another grant, we might want to sequence it using PacBio to get a more better assembly with the long reads. Yeah, but we are quite happy with this since we are looking at the transcriptome. This is used as a backbone for our mapping. Okay, what do we want to look at is basically thermal stress exposure. All right, like I say, Basiosima antarctica actually grow best at 12°C. So the green the temperature I'm showing is that the best growth conditions. But we are exposing it to 0°C and -12°C as it's a rapid move from 12 degree, and we transfer it to 0 degree and leave it to freeze within 6 hours and 24 hours. Then we did the extraction of the mRNA and then do the sequencing. And on the other side, we actually exposing it to heat shock at 16°C and another temperature which is 20°C, which I did not show here. So those are the things that we are looking at. So what is the fate of Basiosima antarctica exposed to this thermal stress? It's either if the population, some of them will not be able to cope, especially the older cells they are facing, going to die, all right? Especially those exposed to 20°C heat shock. And some that can withstand the temperature, acclimatize within the 6 hours. You know, there are different sets of genes that is expressed within the 6 hours. And then if you expose it longer, up to 24 hours, then you find out a different sets of genes that is expressed, basically to adapt to the new environment and probably continue to survive and grow. All right, so those are the things that we are looking at in Basiosima antarctica. Okay, so basically it's overview. I think Dr. Nuradila already explained quite well. First, we expose them to different temperature, then we do a total RNA extractions. Then we sequence using Illumina MiSeq PET-N sequencing, carry out various bioinformatic analysis, basically to look for genes that is differentially expressed. And once the differentially gene that has been expressed, we look at the identity of genes. And some of them we can identify, then some of them actually map to hypothetical genes or gene of unknown function. This is quite common. So I'll just give you an overview about the cold shock first. We look at the cold shock is that the total putative gene that is estimated coded by this is basically 7,857. How many has been, how many are expressed? The 7,733 that is expressed, and we found that there's 124 genes that was not expressed. So either they are hypothetical, means not a real gene, or they are not expressed under these conditions. And what we found that is the differentially expressed genes is 682. When we say differentially expressed, means that the fall of change within the control, which is 12°C, and the change is really more than twofold. Then we consider, and with a significant value of 0.05. Okay, so out of all the differentially expressed genes, we found that actually 33 are actually molecular chaperones, upregulated and down-regulated. Okay, so we move back to talk a bit about the co-adaptations first, and just now it's about the molecular chaperones. So basically the total sequencing, we got 325 million reads generated using the RNA-seq, and say 168 genes were differentially expressed when exposed to cold shock at 0 degree and -12°C. And out of that, we found that the differential 168, the fall of change, which is more than 2, okay? And if you look at it, out of the 168, only 73 that we can find certain function to it. The others are really mapped to hypothetical genes of unknown function. And the 73, we be able to separate them into their biological functions, which are presented here. Is mainly involved in the energy and metabolism, cellular transport and transcription and translation. There's a group of 4 genes which is very important. I'm going to discuss in more detail. It's really the stress response genes. I mean, there's quite a bit of data that we're not going to go through one by one. I'd rather just focus on some of the interesting findings that we see from our result. I really want to draw your attention to the antifreeze protein. There are about 10 antifreeze protein here, which have not changed in terms of expression. Only the antifreeze protein 2, 6, and 4 has been differentially expressed, and yet it's not all the time, but rather at different temperatures. First, we go from optimal temperature of 12 degrees Celsius, and we immediately shift it to 0 degrees Celsius. And after 6 hours of incubation, we call it acclimation temperature, we found that the antifreeze protein 2 is expressed, all right? But when we leave it longer, up to 24 hours, we found that it's no longer expressed, meaning that this has gone back to normal. And then we found another antifreeze protein, that is antifreeze protein number 4, that is now taking over the place of the antifreeze protein 2. So what we conclude is that probably there is a difference in terms of role. The first acclimatization involves antifreeze protein 2, but when it goes to 24 hours, which is the adaptations, then the antifreeze protein 4 will take over the role. But if you look at a more extreme cold, when we move from 12 degrees Celsius to minus 12 degrees Celsius, we found the antifreeze protein 2, again, immediately within the 6 hours is expressed, all right? But if you leave it longer than 24 hours at minus 12 degrees Celsius, you found that it is still retained, means that the role is important, probably because the temperature is too extreme. And on top of that, even within 6 hours, you find that another antifreeze protein, which was not expressed previously at 0 degrees Celsius, now is expressed, upregulated during the first 6 hours at minus 12 degrees Celsius, and cease to express, upregulate after 24 hours. Means that the antifreeze protein 2 seems to be an important component in acclimatization and also adaptations to extremely cold temperature. And if you look at other yeast cells such as Saccharomyces cerevisiae and Markia, we found that the other 2 yeasts are more of mesophilic yeasts. They do not have any antifreeze protein in them. And probably this is the advantage that Glycosima antarctica has by having all these antifreeze protein. In fact, there are more than 10 antifreeze protein. I think the others will be expressed at different temperature. Means that if you put it at minus 5 degrees, minus 6, it might be a different group of antifreeze protein that is expressed. This is something interesting that we found. And the next thing is really the cold adaptations at minus 12 degrees Celsius. We also found that nitrate reductase is upregulated up to 2.6 and 10.9 fold at 0 degree and minus 12 degrees Celsius. So we foresee that at these 2 temperatures, basically the yeast is frozen in the ice. Means that oxygen is really lacking at the time. Then the yeast, if they want to continue to grow, they need an alternative terminal acceptor of electron. So when oxygen level is low, they have to use nitrate as the alternative. So that is something interesting that we found in this particular yeast. Okay, now we move on to the molecular chaperones that I talked about when you expose the yeast cells to heat stress at 16 degrees and 20 degrees Celsius. And we are trying to compare how the Glycosima fare compared to a Saccharomyces, which is a mesophilic yeast. Okay, again, I just reiterate that the differentially expressed genes are 682 with 33, which are molecular chaperones, and 23 were upregulated and 10 were downregulated. I just summarize it into this chart here. That is, if you look at it, there are basically 1, 2, 3, 4, 5, 6 molecular chaperone that is differentially expressed. First, we look at the heat shock protein 70 and 90, which I think Dr. Aliza mentioned as well. We show that this plays a these 2 components plays a very important role in the adaptations to heat. And you can see that all the heat shock proteins is upregulated 16 and 20 degrees Celsius. You can see that it's not much difference in terms of the expression. It's all upregulated. Heat shock 19, there's only 1 transcript there. Is highly upregulated to show the importance of this gene. And the other group of genes, molecular chaperone that is upregulated is really the trig gene. All right, the trig is like TCP ring complex. There are 5 of them, all of them are upregulated. And the others which is up and down regulated is really the heat shock factor, the J domain containing proteins, and also the AATPase. So I'll just quickly go through what are their roles in terms of adaptations. And basically, all the heat shock protein 70, 90, and trig genes were upregulated. Whereas for the heat shock factor J domain containing protein, the AATPase, some were upregulated and some were downregulated. And the heat shock protein basically play a very important role. Like when you give the heat shock, the yeast, there is a haywire in terms of the protein refolding. There's a lot of misfolded or denatured protein inside the cell. So basically, heat shock 70 protein is either helping the yeast to try to refold the proteins, or actually they are trying to translocate the protein so that it's degraded and removed from the cells later. So those are the roles of the heat shock protein 70. And we also find the homolog within the Saccharomyces cerevisiae. Saccharomyces cerevisiae actually have 4 heat shock protein 70 genes, and which are either constitutively expressed or only expressed during stress. But in Viscosima antarctica, we only find the homolog SSA1. That is similar to Saccharomyces cerevisiae. But apart from that, they also have other heat shock protein which is variant and probably the alternative to SSA1, 2, 3, 4 of Saccharomyces cerevisiae. So that is heat shock protein 70. So heat shock protein 90, there was only one that is upregulated in Viscosima antarctica, and basically that is a sort of heat shock 90 and heat shock 70 works very closely together. In fact, the heat shock protein 90 actually works towards the final step of protein maturation. It means that they give it the final touch after all the refolding done by the heat shock protein 70. And the heat shock factor basically is a transcriptional activator. When the yeast is exposed to a different temperature, means a higher temperature, different sets of genes needs to be upregulated or expressed to take over the role so that they can try to adapt to the new temperature. All right, and so that is why the heat shock factor, some are upregulated, some are downregulated. So the downregulated one, probably those are making gene expression, controlling gene expression at normal temperature. And when the temperature change to a higher level, then some other groups of heat shock factor will take over so that different sets of genes are upregulated. And then we move on to the TCP ring complex. All right, this is a chaperonin that is played in the central role in cellular homeostasis. Means that when there's a certain change of temperature, they try to make the yeast to continue to grow by normalizing the situations. Right, it's like a gatekeeper of cellular homeostasis. And it has a high similarity of amino acids with other mesophilic yeast, such as Saccharomyces cerevisiae. So basically, the roles played by them are more or less the same in terms of making the cell go back to normal cellular processes. And the J domain, actually it binds to non-native protein. Means it binds to the denatured protein and basically deliver it to the heat shock proteins. It's like a delivery boy. It brings the heat shock protein, bring the denatured protein to heat shock protein 70 so that it can be refolded, at least attempt to refold it. And it's localized in various compartments, which is mitochondria, the ER lumens, and ER membrane. So they basically just to transport it to the heat shock protein 70 and 90. Okay, I'll summarize soon. All right, thank you. So the ATPase is basically to deliver all the energies that is needed, because when the protein is destroying the protein or denatured protein, we need energy, and to make new proteins, we need energy. So that the AATPase basically to provide the energy. So I just want to summarize this part, that is the 6 major molecular chaperone that was differentially expressed at 16 degrees Celsius and 20 degrees Celsius. So in general, their roles are to stabilize, to maintain unfolded or partially folded protein in solubilized states until properly folded, and the cell can continue to grow normally, and to remove misfolded protein, or to translocate or move the denatured protein to the engine where they can be refolded. And of course, the AATPase to provide energy. So basically, the strategy is very similar in certain aspects to Saccharomyces cerevisiae, but there are also others that is very, very different from Saccharomyces cerevisiae. So that leads me to acknowledge the funder, which is the Ministry of Science, Technology and Innovations, and also the group members that is listed here. Thank you very much for your attention.
Thank you, Michael, for a very detailed presentation about chaperones. I'm not specialist in chaperones, so probably we have questions. Maria Pavlovska in chat. Maria Pavlovska, this is for all.
Hello, yes, can I ask a question?
Okay, welcome.
All right, hi Michael, Ashab, I hope you remember me.
Yeah, I do remember you.
Yes, it's interesting to see difference in expression for specific or targeted genes, but have you checked whether, when you change the temperature, how is it affecting at the macro level, such as whether the growth speed is different or the cell reproduction is different? Because it's also interesting to put in context as to what's happening in the molecular, at the molecular level and also at the macro level.
That's right. So, okay, just to give an idea is that the one cycle at optimal temperature is about 90 minutes, means it's 1.5 hours per generation. So that is long compared to Isochromous cerevisiae is about 30 minutes per cycle. Okay, if you look at the temperature, like if you put a higher temperature at heat stress, even the morphological, you look under the microscope, is quite different. All right, so that is a heat stress. But heat stress, I found the strategy is pretty much the same as the mesophilic yeast, because the engine involved the heat shock protein 70, 90, and the other 3 or 4 genes, you can find the same one. But moving to the cold or freeze stress at -12 and 0 degrees Celsius is totally a different ball game altogether. Now we are talking about the whole yeast freezing encapsulated in the ice. There's no room at all for them to expand. Even if they want to duplicate, I don't think they can. See? At the same time, oxygen is really depleting very fast when metabolism continues. So that's why they are using a different mechanisms. Unfortunately, we are not be able to look at the morphology at -12 degrees Celsius. So what we are doing now, hopefully we can see the cell, is that we are trying to use a lot of sodium chloride, so that even at -0 degrees Celsius, the formation of ice did not occur. But then the temperature is maintained at 0 degrees Celsius. That is something that we really wish to do, and to look at how the cell behave morphologically and otherwise. Yeah.
Okay, thank you very much.
You're welcome.
Nuradila, go ahead with your question.
Yes. Hi, Dr. Michael. Thank you.
Hi, Dr. Adila.
I just wanted to comment. It's very interesting that you saw some HSF factors going down and some going up. My question is, I was wondering, how were you able to resolve the different SSA proteins in your yeast? Was there enough sequence similarity to Saccharomyces cerevisiae that you were able to see, like, oh, this is SSA1 versus, and I remember you saying that SSA2 or 2, 3, 4 you can't find it in your yeast. I was just curious because for the fungi that I was working on, I was not able to, I wasn't using bioinformatics to do it, but that was just by manually. I wasn't able to resolve the different SSA proteins, but I did find SSA and SSB.
Sure.
I was just calling them different orthologs. Yeah.
All right. Yeah, like I say, we only find one homolog to SSA1. The other 2, 3, 4 we can't find, or they are different version of it. We do have other like protein 70, which have very low homology to all the other SSA. But I have not presented, but you can see in my papers that they are very similar to other heat shock protein 70 that is found in Saccharomyces cerevisiae. So to answer the question, yes, the homology is quite high. In a sense, I couldn't remember offhand, but it's definitely mentioned in our paper to see. It's basically analyzed using the blast to go, homology search. That's the fastest. Although there are various mechanisms that we can do the same thing. Yeah.
All right. Thank you so much.
You're welcome.
Questions in chat? Okay, if no, we thanks Michael for presentation. And I invite the next. Who is the present? Berezkina? Berezkina is present.
Hello.
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 You can start.
Yes. 1 minute. I couldn't put my video. Did you see? Okay. I will start demonstrate.
Anna, did you send your presentation to our technical support?
I couldn't just put my video, my face. Maybe I.
Okay,
Yes,
we can see.
Okay.
Do you see your presentation? Is it possible for you?
What's your presentation? Did you see my presentation?
Yes,
Yeah, we see, but I ask about you. You can see? Can you see this
Yes.
presentation? Okay, let's go.
Let's go. Hello, dear colleagues. The theme of my report is phylogenetic relationships on the genus Pseudalteromonas, Psychromonas, and Okeanobacillus in the polar regions. Supervisor, Associate Professor Andrei Utevsky, Department of Zoology and Animal Ecology, Karazin Kharkiv National University. As we all know, that Antarctic has highest proportion of endemic and new species, and the priority area for international Antarctic community is increasing the scientific knowledge of Antarctic knowledge. So the interesting research area of modern biology is study the symbiotic organisms and its relationships between organisms. In the literature, there are a lot of information about symbiotic microorganisms that are associated with benthic organisms, and we know that these microorganisms could be involved in protections the host organisms against infections, synthesize antibiotics, and also help in digestion, synthesize enzymes. Both host organism and microorganisms also can secrete nutrients that are mutually consumed by each other, and in turn the host organism can provide the refuge for microorganisms, and also could be involved in selection of symbiotic microorganisms. For example, sponge could produce metabolites such as N-acetylhomoserine lactones that are directly select symbiotic microbiota. So on the slide you could see a few examples. These symbiotic microorganisms, it's Pseudomonas aeruginosa proteobacteria associated with the sponge Isodictia sativifera, Gamma proteobacteria, flavobacteria, and actinobacteria associated with the sea urchin Strichina pneumoniae. And another one example is Pseudomonas Psychrobacter and Chivanella, Psychroserpens and Algariphagus, Carinibacterium are associated with the soft coral Alcyonium antarcticum. There are many reports about symbionts that associated with mollusks. There are a few examples. It's bacterial symbionts on the mantle epithelium associated with Lapillina antarctica, bacterial symbionts on Ctenidia associated with the T-Zira falklandica, and gamma proteobacteria associated with the Gigantopelta chisoia. Some symbionts could produce enzymes that are characterized by high level of flexibility in protein structure, high efficiency in catalytic efficiency at low temperature, and also needed lower energy of activation. The few examples is Flavobacterium, Pseudomonas psychrobacter, are gut-associated with the Oligohetagrania annelida. Symbionts produce extracellular proteases, esterases, amylases, cellulases, and agarases. Thalassospira that belong to alpha proteobacteria associated with the Arctic sea fan, Paramuricea placomus, and this Thalassospira is a producer of esterase. Cryptococcus laurenti and Rhodotorula mucilaginosa associated with the Antarctic limpet Nacella cancina, and this mollusk-associated yeast are producers of lipases, proteases, and xylanases. But interesting that no any data on bacterial associates of Antarctic limpet. So this is a gap in polar science, and we try to shed lights on this topic. Such objective is to study the bacterial biodiversity of the Antarctic limpet Nacella cancina from the water area of Argentine Islands, West Antarctica, and its potential practical applications as enzyme producers. Object of the study: Nacella cancina associated bacteria and bacteria from bottom sediments. The underwater research was conducted near Ukrainian Antarctic Station Academic Vernetsky. Nacella cancina is the most common successful numerous species of local fauna, and the shell size up to 6 centimeters. But usually deep water species are usually smaller. The depth range varies from intertidal zone down to some 100 meters. We isolated 103 pure bacterial culture from soft tissue, intestinal tube, and bottom sediments by serial dilution method. Microorganisms are represented mostly gram-negative rod-shaped bacteria, and Kochi on average 10 in 5th colony-forming unit per gram. Microflora is halophilic, mesotolerant, and psychrophilic. Pure culture have oxidase activity. Some have high agarlytic activity, as well as proteolytic and glycolytic activity. For more detailed study cell morphology, we used confocal microscopy. On this slide, you could see a few micrographs, Antarctic strains that associated with soft tissue of mollusks. Red fluorescence is a capsule, cell wall, and membrane, a lipid-specific nil red dye, and the blue fluorescence is a nucleoid, DNA-specific staining with DAPI dye. The next stage of our research was barcoding of 29 strains. We using amplification 16S rRNA gene with using standard bacterial primers 16S BAC27F and 1492R. According to preliminary results of DNA barcoding, mollusk microbiota belong to the marine proteobacteria, such as Pseudoalteromonas, Psychrobacter, Chivanella, Cobetia, Psychromonas, Bacteroidetes, Bisionia, and Firmicutes, Okeanobacillus. A maximum likelihood 3 demonstrated that some Pseudotelomonas, Psychromonas, and Okeanobacillus strains are phylogenetically close to species occurring in the Arctic Ocean and north part of Pacific Ocean, which suggests their bipolar distribution. The next slide, I would like to show you some examples of these relations with polar microorganisms. For example, strains AA11, A17, and AA19 clustering with the strain of Pseudoperomonas arctica, which was isolated from seawater samples collected from Spitsbergen in the Arctic. Strain 9B1 combined with halophilic strain Okeanobacillus picturea that was collected from glacial moraine in Kvanad, Greenland. Strain 8B2 was related to Psychromonas arctica that was isolated from cold fjords of Spitsbergen, and strain 16C2 belonging to the genus Bisionia, clustering with the strain of Bisionia barichis that was isolated from fish, Splendin alpha-leucina, you could see on the slide, and that fish was collected from the North Pacific Ocean. And the last one example is strain 5C2, Formatoclade viscobatia crustatorum, which was isolated from jet gull. It's traditional Korean fermented seafood. And Chromohalobacter canadensis was taken as outgroup. Some isolated pure culture have keratinolytic and xenolytic activity. 76.4% of microorganisms showed keratinolytic activity from 1 to 4 units. The highest xenolytic activity, 0.13 unit per milliliter, was detected from strain A8A2 and isolated from bottom sediments of the Squaw Creek Strait 10 meters. For more detailed research, this keratinolytic activity, we used 4 medium and 2 temperature of cultivation. We used medium with maltose and gelatin, with addition of feathers, with maltose, gelatin, and sodium chloride, and also with additions of feathers and sodium chloride. So most cultures synthesized enzymes with keratinolytic activity at 28 degrees Celsius from 1 to 9 units per milliliter, but at 19 degrees Celsius, the level of this activity was higher, up to 15 units per milliliter. On this slide, you could see total proteolytic activity. The same, we used 4 medium and 2 temperature cultivation, and it's shown that only 5 bacterial isolates, when grown at 28 degrees Celsius, showed xenolytic activity at the level from 0.011 to 0.074 unit per milliliter, while a large number of cultures had this activity at 19 degrees Celsius, up to 0.082 unit per milliliter. Some strains have alpha L rhamnosidase activity. 23.5% of strains that isolated from mollusks showed alpha L rhamnosidase activity. Maximum glycolytic activity was detected in the culture fluid of strains 4 and 6. Up to 0.11 unit per milligram protein, which were isolated from the washout of mollusk shells in the water area near Uruguay Island, 16-meter depth. So, conclusions. A maximum likelihood phylogenetic tree demonstrated close relations of Nacella-Cancino-associated strains and bacteria inhabiting the North Polar region. Some species have not only a bipolar distribution, but are also probably cosmopolitan. And the producer of proteolytic and glycolytic enzymes were isolated from the gastropod mollusk Lancina from the water area of the Argentine islands. And most cultures synthesized enzymes with keratinolytic activity, but at 19 degrees Celsius, the level of this activity was higher. And as well as a large number of cultures had caseinolytic and glycolytic activity also at 19 degrees Celsius. So thank you for attention. If you have some question.
Thank you to you, Anna. I have one wishes to you to analyze the mollusks from North Pole, yeah, for correct conclusion, because the second conclusion of your presentation has not really results. It's only your such special discussion only, because you haven't data which can tell us about really this situation, okay?
Mm-hmm.
Okay, let's go to chat. Svalbard, limbs, pepper. No, it seems to me our participations continue to discuss some aspects of previous work. Have you any questions? No? If no, thank you very much, Anna, for the interesting presentation. And let's go. Tatiana Tisokina, welcome.
Thank you. Just a moment. My presentation. Can you see it? Well, due to the circumstances, I was delegated to present the report. The team is functional.
My specs just too big that I can send through email.
Functional features of biogeochemical migration system of toxic chemical elements in terrestrial ecosystem of Paradise Island. Toxic chemical inputs.
Dear Tatiana, I am terribly sorry. I asked Hidai, can you switch your mic? Hidai? Our technical support, help us.
Yes,
because two voices in the same time, it's very hard to analyzing.
It's okay now.
Okay, thank you.
Toxic chemical inputs to terrestrial ecosystem in Antarctica are at risk to their sustainability due to the slow biogeochemical cycle flow and the linearity of the food web. On the other hand, chemical elements flows can be the markers of natural processes in ecosystem, reflecting their structure and sustainability. There is a large data set on toxic metal content of terrestrial ecosystems in Antarctica that needs to be generalized through biogeochemical cycle modeling with the possibility of assessing both anthropogenic impacts and the local specific factors, such as geological processes or ornithogenic factors. Further intensification of human presence in Antarctica and climate change may have been harmful to polar ecosystem. While the adverse effects of anthropogenic pollution of Antarctica are well understood, the effects of climate change is difficult to predict. And therefore, it is necessary to assess of national processes of migration of toxic chemical elements until the moment when they are disrupted by human activities. The aim of our research is to establish a generalized model of the biogeochemical system of migration of toxic chemical elements in terrestrial ecosystems of Antarctica, the Andes Island and Argentina Islands. According to the aim, the main tasks are assess the specificity of the chemical composition of the abiotic and biotic components of the terrestrial ecosystems of Andes Island, estimate stocks of toxic chemical elements in the main components of the Galindez ecosystem, identify the main migration pathways of toxic chemical elements, and assess toxic chemical fluxes in terrestrial ecosystems. The content of lead, cadmium, chromium, nickel, cobalt, copper, and zinc in samples of soil, melted water, and vegetation were collected from 6 model sites. The results. Comparison of the observed sources of fresh water with the toxic metals content of Galindez revealed a high concentration of all chemical elements except chromium in the water of the pond at meteor site. We don't have, I noticed that we don't have an official name to the site, so we name them according to location or other essentials. Cupola, Colobanthus, Meteor, and D12 and VLF. The high concentration of chemical elements in the water in this reservoir, which characterized by different sources of inputs and migration routes, can be considered an indication of a common source of pollution. The presence of an elevated lead concentration in the water, according to the most researchers, is mainly due to anthropogenic activities in the Antarctic region, indicating contaminations of water body as a result of economic activity. The different distribution patterns are relatively low water concentration of nickel and chromium, indicate other sources of these toxic metals in terrestrial ecosystem. In particular, weathering products and their atmospheric transport are a significant source of chromium. This is confirmed by the maximum concentration of chromium found in the water pores at the Colobanthus site. You can see. Unfortunately, the lack of meltwater streams at the meteorological station, whose rocks are enriched with these toxic metals, makes it impossible to draw a definitive conclusion on the role of parent rock in the formation of iron zinc. Analysis of the spatial dynamics of toxic metals in soils of Moses under Moses found similarities in the distribution of chromium and nickel that low accumulations were found for VLF and the D12 sites. The cupola site was characterized by maximum metal content in the soil, where the metal concentration was from 2.5 to 5 times higher. The soils of the Colobanthus and meteorological station also found a high concentration of chromium and nickel, and not significantly different from cupola site. The distribution of cobalt, zinc, and copper was characterized by their characteristics, which included a gradual increase in the chemical element content in VLF, D12, then Colobanthus and meteorological station, and the high with cupola. In all cases, high toxic metals levels were found in soils at cupola, Colobanthus, and meteor station site. At the same time, the lowest concentration were found in the VLF site for almost all heavy metals. The distribution of lead and cadmium accumulation differed from other chemical elements. The levels of accumulation of these toxic elements did not differ significantly at all sites except VLF. By distribution, the chemical elements can be grouped into 3 groups: chromium and nickel, cobalt and zinc, and copper. A comparative analysis of toxic metal concentration in Moses was carried out for all study sites. The spatial distribution of concentration of toxic elements of Moses differed significantly from soil and was highly diverse. In the soil, a concentration of chromium and nickel were characterized by similar dynamics, but they differed somewhat in their accumulation characteristics. The lowest values of these elements were found at VLF, B12, and Colbanto site in the range of 1.2 to 1.4 milligram per kilogram for nickel, and for chromium from 1 to 1.5 milligram. Little conclusion. The first, special distribution of nickel chromium in water of water-cautious soils and vegetation compared to other toxic metals shows an independent source of the input into ecosystem of Galindez Island. However, the high content of nickel and chromium in the streams collecting water from the main part of Galindez Island does not depend on chemical composition of the rocks, which are significantly lower than the concentration. Conclusion number 2. The spatial distribution of lead concentration in Murchison soil is not consistent with any other analyzed toxic metal concentration. A correlation analysis of published data from other researchers also did not reveal the relationship of lead accumulation in the components of Antarctic terrestrial ecosystems to other toxic elements. And on the slide, we can see table with the correlation matrix of toxic metal content and their comparison. And the third conclusion refers that the area of the terrestrial ecosystem of Galindez Island, where the diesel power plant of academic Vernadsky station is located, is characterized by a clear sign of contaminations of all components: lead, cadmium, copper, cobalt, and zinc. You can see this is very high. But there is no significant increase in content of chromium and nickel. Next part of our research referred to the biomass. And in order to estimate the stocks of toxic metals accumulated by plants, group, and their formed soil, vegetation, distribution mapping was carried out to assess the area of the island. An estimate of total area occupied by plants group of February 2020 gave us this value. The area of most fields and individual banks was 12% of total area of the island. At the same time, on Marina Point, there are particularly no moss fields. The area is only 20 meters squared. When estimating the area of total vegetation, on it is almost 35% of the area of vegetation groups on the island. As a result, the largest reverse for lead was found. Yeah, it's almost almost 3.6 kilograms. And the main depot of this metal on the island in the soil under most fields on the Galindez Island, containing almost 1 kilogram of metal, and this is 29% of the total reserve. Although 42% of the lead stock was concentrated in the vegetation and soil of Marina Point, the main depot of this metal on the island in the soil of Galindez Island was 29% of the total stock. Although the temperature in the western region of the Antarctic Peninsula was decreased after a long period of warming that occurred at the end of the 20th century, the area of the Antarctic Peninsula glacier is declining. The largest changes in ice cover are observed in the northern and western parts of the peninsula. Estimates for period 2014-2019 indicate that the most significant changes in ice cover are observed in the northern and western parts of the peninsula. The glacier on Galapagos Islands lost about 2.5% of its volume. That's why this value we choose to estimate the flow of water from the island's surface. In fact, the volume of the water runoff is much higher because the flow of melted water from seasonal rainfall is not taken into account. But on the other hand, part of the loss of the glacier is possible through sublimation process. The calculation of amount of emission from the combustion of petrol and diesel delivered to the academic Vernadsky station in the beginning of 2020 was also carried out. Its value is 3,000 liters of petrol and 140,000 liters of diesel fuel. Maximum toxic metal contents for modern fuel was used to assess the chemical composition of emission from internal combustion engines. Thus, despite the substantial removal of lead from the territory of Galapagos Island, the continuation of operation of Ukrainian Antarctic station would lead to the accumulation of the given toxic metal in terrestrial ecosystems. You can see in the table there, in a year that twice as many lead enters the environmental as is carried with water courses. The information obtained of total stocks of toxic metal and their anthropogenic input and water discharge was used as a basis for a conceptual model, as you can see on this slide. This is a model of the migration of toxic chemical elements in terrestrial ecosystem of the Galapagos region, which was based on the balance method. In order to assess the relative contribution of anthropogenic sources and toxic metal fluxes, a pollution factor for the terrestrial ecosystem has been calculated representing the ratio of input and removal of the chemical element. Analysis, also I would like to add some information that the analysis of possible sources of modern toxic metal inputs suggested the existence of large-scale transport processes in the Antarctic region, and volcanic activity may be such a source. So not only anthropogenic. The northern part of Antarctic Peninsula is characterized by high tectonic activity, accompanied by a large number of active volcanoes. These 9 have been active for the last 150 years, such as Bristol, Deception, Penguin. Most of the volcanoes are stratovolcans, whose eruptions are accompanied by an explosive character with a released a large number of tephra. And given the prevailing wind direction from the north-northeast, the products of the volcanic activities of the Antarctic Peninsula will be transported mainly in the direction of the Galapagos Island. So this can be also one of the source of toxic metal. Let me not to read the results in order to save the time. Thank you for your attention. Gladly answer your questions.
Thank you to you, Tatiana, for the interesting presentation. We have one question in the chat. Can you find it? Can you?
Yeah, yes.
Chat. Yeah. Look, please at these questions.
The last questions I see about the 3D models. Yeah, it will be one of our ideas that come into life because of some circumstances, but I think we need more data. So this model is very rough, so I don't think now it can show real results. So I think in the future maybe it would be a good idea to do some 3D models. And question.
Yes, the first question to you.
Thank you for the literature, first of all. You know, the lead is for us, it was a really enigma because of it, because of its distribution in all components of ecosystem, not only in water or soil, but and in vegetation. And I think it is really because of specific of how it is bonded in environmental. And thank you for the literature. I gladly read about it.
Okay, dear colleagues, any questions? If no, we will try to. Okay, and now I would like to invite Alexander Gromyko from Lviv University. Is he here or not?
Hello, everyone. Ivan Roman will report for our group.
Okay, thank you. Welcome him.
Hello to everyone. So I start. You see my presentation? Can you see my presentation?
Yes, I see.
Okay. I would like to present you our report about soil actinobacteria from Gelindes island. How you can see its name, we a little change a name of our presentation because we a little supplement our data. So I hope it's not problem. For start, I must to say that background of this slide is like real cosmos, and it's a real cosmos of actinobacteria that we studying. So let's say about this little creature a little more. For starting, I must to say that actinobacteria is a gram-positive prokaryotic organism that can form mycelium. Actinobacteria take place in many important biological processes, including soil formation, and they produce a many number of biological activity compounds, such antibiotic. Also, they can be plants and animal symbiotes. Our goal of this work is assessment the diversity, properties, and biotechnological potential of actinobacteria associated with Antarctic plants of Gelindes Island. To isolate actinobacteria, we use 3 different medium. Is simple inoculation, treatment with phenol solution, and preheat of samples. And after that, we inoculate our samples at 4 different medium. And in first part of my report, I will say about actinobacteria associated with polytrichum, and in second part, I will say about actinobacteria from our previous data, and they associate with Darham Antarctica. After isolation from samples polytrichum, we have 23 isolates with actinobacteria-like morphology. And the most number of isolate we take after the treatment with phenol. In this photo, you see the ice dish with samples. The phylogenetic analysis based on 16S rRNA shows that all bacteria that we isolate is Actinobacteria, and they representatives of 4 different genera. It's Micromonospora, Streptomyces, Cribella, and Micrococcus. The first stage of our research was understand cultural properties of Actinobacteria. And we detect that 8 isolates can grow when temperature of air is low. It's Micromonospora 67.1, some Streptomyces, Cribella, and Micrococcus. And some Streptomyces 67.13 F15 can grow in the wide range of temperature. Also, this strain can grow in the wide range of acidity. Then we detect the some isolates like Micromonospora 1612, sorry, and some Streptomyces that can grow with high level of salinity, up to 7.5%, and 2 stains, Streptomyces 6727 and 6728, that can produce melanoid pigments. Next stage is to understand what enzymes produce our isolates, and in this work we compare our previous data from isolates of the Darham rhizosphere. And how you can see in this diagram, the more number of isolates of Polytrichum can produce lytic enzymes. It's may because of a great number of fungi in rhizosphere of Polytrichum, and they have some concurrence about substrate. Another stage of our research is antibacterial activity of stains, and we see that most number of isolate from the Darhamsia Antarctica have activities against bacteria, such Bacillus subtilis, Staphylococcus, and another. And in another hand, we see that isolate from Polytrichum have antifungal activity. You can see in this part of presentation. So it's maybe because of similar life cycles of filamentous fungi and Actinobacteria. To more and clear understanding of what nature of this actinobacterial activity, we do some PCR screening, and we screen some genes of biosynthesis like polyketide synthase 1 and 2, non-ribosomal peptide synthase, polyene-specific hydroxylase, aminoglycoside phosphorylase, and 3-hydroxy-3-methyl coenzyme A reductase, and glycopeptide monooxidase B. It's only one strain that have this gene. It's Cribella 67.2. Also, we must say that we have some bacteria, some Micromonospora that have any of searching genes, but they have antibacterial activity against fungi. And the most interesting strain was 6719 that have activity against most of studying bacteria and fungi, and we tried to do chemical analysis of this strain. So after the replication analysis of crude extract of this strain, we see the presence of surrogametes D and A, as well as antimycin A3, A10, and A1. Also, we detect 5 potential new compounds that have no similar mass in database, so it's maybe can be new compounds. Okay, in this words, I finished first part of presentation and going to another. It's actinobacteria of the genus Umezawa associated with the Shams Antarctica. When we complement our data, we see that only one genus present in the Shamsia and don't present in Polytrichum. It's genus of Umezawa. It's quite a rare genus of actinobacteria, and for now it's only 2 species in this genus. We we do phylogenetic analysis of all stains that we can take information about, and see that almost all Umezawa that we isolate, and we isolate 11 stains. So maybe all of them Umezawa endophytica, and one have no similar to any of Umezawa. And we can we think it may be a new species of this genus. So we sequenced a genome not only of Umezawa 62.2 and for all, but today we talk only about Umezawa 62.2. So after genome annotation, we saw that it's a very large genome, and it's very large number of encoding gene. Very interesting is gene for resistant to heavy metal, including mercury, and gene for the composition of aromatic compounds. Another part of our work is searching secondary metabolites potential biosynthesis gen clusters, and we use anti-smash software, and we can find 49 potential clusters, and this in this diagram you can see the type of this cluster. Among the potential synthesis cluster, we find cluster with a high homology. For example, we find probable cluster of indigoidine synthesis. It's blue pigment that show antioxidant activity and may be involved in protection against UV radiation. Also, we detect probable cluster of isoryanitren synthesis. It's carotenoid pigment that can also be involved in UV protection against UV radiation. And in last, we detect a cluster of macrothermicin synthesis, and it's macroreclam antibiotic that have activity against gram-negative bacteria and fungi. So we find a large number of probable cluster, and we decide to do chemical analysis. And after the replication analysis of crude extract of Umezawa sp 6202, we find just 2 secondary metabolites. It's fluoridectin C2 and fluoridectin C1. Before this compounds found in spongy, and they have anti-malarial properties. Maybe we think that maybe our cluster have its silence cluster. So another work, we try to activate this cluster, and maybe we see more secondary metabolites. So to conclusion, I must say that actinobacteria from Antarctic plants is very interesting and important source of new bioactive compounds. And due to geographic isolation and adverse environment factors, in this area can evolve an interesting new species and genus, and of course studying of mechanism to avoid these conditions can help us develop a new methods in agriculture and etc. It's our publication. One article is right now published and two accepted. For ending, I want to say thanks for our team, as well as I want to say very grateful to Ivan Parnikoza for collecting and delivering samples of rhizosphere mosses and Deschamsia, as well as Andriy Luzhetsky that can help us with chemical analysis of strains, and Yuri Rebets and Explogen for sequencing 16S rRNA genes and genomes of Umezavas. Thank you for your attention.
Thank you to you. Any questions? Unfortunately, I haven't any questions in the chat. Maybe.
Hello.
Yeah, you have? Okay, please.
Yeah, I do have one quick question. Yeah, I
Okay.
want a wonderful talk. I mean, congratulations to be able to find some interesting compound from there. I just wonder from your genome, do you see a lot of antibiotic resistant genes such as beta-lactamase? Because we have sequenced some bacterial genome from the Antarctic. We do see a lot of beta-lactamases in there. Do you see the same trend?
Thank you for your question. Yes, in all genomes of Umezavas, it's beta-lactamases, genomes of beta-lactamases, and our biotest for resistance for beta-lactam antibiotic is positive, that our stains grows on presence of penicillinum and another antibiotic of this group.
Okay, for the two active compound, have you resolved the structure?
Maybe can you repeat?
Have you resolved the chemical structure of the compounds that you have found?
Not ready yet. Be in processing.
Okay, so all the best to you. Very interesting. Yeah.
Thank you.
Thank you for the questions. And other questions? It seems to me that's all. Thank you very much to you for the interesting presentation, Ivan. And we will go to Popov, yeah? The Shina.
Beforehand.
Shina, but before. Sorry. Valeria, thank you for the help. Sorry, it's now will be a little break for the photos. And that is why I invite everybody to switch your cameras and preparing for photos.
Taking photos,
okay?
Yeah.
Let us. Wow.
Are you ready?
I hope.
Okay. So smart. Oh, yes. More people. So, are you ready? So, smile. Smile. One more time. Next screen. Smile. Okay.
Okay, thank you, our technical support and for everybody for photos. And now we can go with the next presentation. This presentation should be not alive, but in the film. Yeah.
We have a video.
Yeah.
Yes, with presentation. So I will share it.
Okay, thank you.
Good day, dear conference attendees. Good day, dear conference attendees. Thank you so much for the opportunity to present the results of our research to such a respected audience. Over the last decade, a team of scientists from Karazhan University has conducted interdisciplinary research to study Bantu groups and the impact on their condition of various natural and anthropogenic factors. Our huge team includes biologists, geographers and cartographers, physicists and chemists. Last year we were able to pay attention to the study of the chemical composition of bottom sediments. In this report we will consider the support of these studies by GIS and cartography. So let's start it. On this slide you can see an overview of the study area where the bottom sediment samples were taken for further analysis. In particular, areas were selected close to the location of scientific station with significant anthropogenic impact, as well as areas far from such impact. Samples of sediments were taken at different depths of the water areas. The plots were laid out in such a way as to cover the transects of previous research years, which had previously been used to study the state of Bantu groups as well as chemical composition of primitive land soils. While diving, the material was collected directly from the soil surface to a depth of 40 millimeters in airtight containers with a volume of 120 milliliters. Later, in the Institute of Chemistry of Karazhan University, the content of chemical elements was analyzed. List of that elements you can see on the slide. The main data on the analysis of the chemical elements content were injected into the geodatabase. Mapping of the distribution indicators of the studied elements in space was performed in the ArcGIS environment using the results of acoustic survey of the bottom of the Argentine Islands water area in the season 20 and 19. The use of this data also allowed the use of three-dimensional visualization of the Galindez Island area. The nature of the elements distribution is determined by local biological and geochemical processes. The obtained results show that in the control areas outside the constant anthropogenic impact, the indicators of the content of biogenic compounds are higher, and the content of heavy metals is lower. The overall situation is not so straightforward, of course. It was detected no exceedance of the maximum allowable concentration for manganese. However, using GIS and maps, we can observe an uneven distribution of its content across the territory. As you can see, the concentration of this element are lower in areas outside the anthropogenic impact. It was detected exceedance of the maximum allowable concentration in some locations, for zinc, copper, plumbum, mercury, and for all points, for sulfur and hydrogen sulfide. It was detected a local significant excess of the maximum allowable concentration in the Stella Creek Strait for plumbum and mercury. The indicators of the elements' content in their spatial distribution show a negative correlation between the most toxic elements, plumbum, copper, mercury, and biogenic elements, phosphorus and nitrogen. At the same time, there is a positive correlation between biogenic elements, phosphorus and nitrogen. In the next stages of our research, it is planned to carry out more detailed geospatial analysis of the relationship between land and water areas. One of the tasks is to analyze the content of chemical elements in primitive soils and in bottom sediments. Is it worth noting that research and mapping of these indicators was carried out by us in 2017. On this slide you can see an example of separate maps for one element, nitrogen, for the land of the islands on the left and the bottom of the water areas on the right. Also important for the further comprehensive study of chemical elements content with considering the structure of the bottom relief of water areas, underwater currents and characteristic of bentos. Undoubtedly, cartographic and geoinformation models, including three-dimensional ones, will make it possible to demonstrate the identified geospatial patterns more clearly. So we are moving to conclusions. Next action were done based on research in the composition of bottom sediments in 2020. Firstly, geodatabase was supplemented with a new adjectives layer. That data includes sampling points of bottom sediments of the region with 14 attributive data of the elements content. Secondly, we create 14 maps for that represent of the separate elements content by methods of round diagrams. Third, our team systemized data and scaled for the possibility of comparing results of bentos research, analysis of chemical composition of bottom sediments and primitive land soils for further identify geochemical trajectories of influence in the places of interaction, land water and environment bottom. Generalized indicators modeling for bottom sediments using extrapolation and interpolation methods cannot be performed now. The application of such methods requires a much larger number of sampling points in the study area. In our opinion, is it more expedient to monitor the areas of potential human impact and its activities in the near water area? The monitoring of that type should be implemented through an irregular network of checkpoints which has yet to be substantiated in the future. We are planning to publish all summary results of the research soon in the form of a public web map using ArcGIS Online with additional information for each study point of bentos, primitive land soils and bottom sediments of the Argentine Islands water area. So we are very grateful for your attention. Also, if you have any questions, we will be glad to answer them. Thank you so much.
I'm sorry. I'm sorry. Okay, thank you for presentation, this person who absent, really. Yeah. But I invite to everybody to put question if you have them. Okay, It seems to me, one moment I find in the chat. No, it's a question for previously participant. If you haven't questions, we will go and ask Natalia Raksha. Have we Natalia Raksha or not? In a link. Maybe Tatiana Galimova, Tatiana Volk, Alexei Savchuk, no? If no, I invite Tatiana Kuzmina to present her results. Tatiana Kuzmina. One moment.
Her presentation. If she's here, I will show it. Tatiana, are you here? She's in Zoom meeting.
It's excellent. Okay.
Yes, but
yes, I remember this problem. Okay. Can we start? Ми вас чекаємо. Ви з нами? Окей, так, давайте. Угу. Ну, немає двох, тому ну, давайте працюйте.
oh. Tatiana?
One moment, yeah. Okay,
One
sorry.
moment. I'm sorry, really, I didn't expect so many people will be out. So, and I try to share my screen.
Ah, okay.
May I try
sorry.
to share my screen?
Okay, yes. Okay.
If you have problems, yes, my screen. Oops. Okay. Oh, my screen. Out. Yes. Can you see?
No, not now.
My screen. I expected something wrong. Okay, would you please help me?
Okay.
Okay, I will use your screen. Okay.
Do you see your presentation?
No, 1 second, not yet. No, now I can see letter, message from. No.
Будь пару хвилин, зараз включиться. Okay. It seems to me it's okay. We can continue.
Yes, I can see.
Okay, thank you, Tatiana. Welcome.
Sorry for such inconvenience. Dear colleague, my name is Tatiana Kuzmina. I'm working in Schmalgausen Institute of Zoology, and I'm parasitologist. And now I would like to present our data on species diversity of the parasite community of bony fishes from Argentine island, West Antarctica. These studies were performed last year using material collected during 2014, 2015, and 2019-20 during 19 and 24 Antarctic expedition. So, a couple of words about the background of my study. This study was our study. This study was performed as part of future, I hope, long-term monitoring study of ecological state of marine ecosystems in West Antarctica. Why it is important? Next slide, please. May I ask next slide? may I ask, yes, environmental changes caused by global warming and anthropogenic factors, previous one, yes, previous one, sorry, yes. So environmental changes which are caused by global warming and anthropogenic factors are observed in terrestrial and marine ecosystems around the world. In polar regions, in Arctic and in Antarctic, the recent rapid climate changes are the most pronounced. First of all, these environmental changes are manifested as a rapid reduction of the ice cover, collapse of ice sheet, reduction of ozone layer, ocean acidification, and as a result, as changes in the biodiversity of terrestrial and marine ecosystems. Next slide, please. Next. May I ask next slide? Yes. Parasites of various taxonomic, previous one. Parasites of various taxonomic groups, nematodes, ops previous. I should come to Antarctic Center. Yes, sorry. Metazoan parasites of various taxonomic groups, nematodes, trematodes, Anthocephalus ancestors, are known as one of the most sensitive indicators of state of ecosystems, especially in marine environment. First of all, it is connected with complex life cycles of these parasites, which include various group of various group of invertebrates and vertebrates like intermediate and paratenic hosts. Here on these three diagrams, you can see how many organisms are included in life cycles of anisakid nematodes. You can see crustacea, different species of fishes, and marine mammals. In life cycle of Diphyllobatriid cestodes or Acanthocephalans. And marine fishes are key component of these life cycles because they are first, second, intermediate host or paratenic host for all group of metazoan parasites. As most of marine mammals and birds in Antarctica are under protection, it is impossible to descend them and collect parasites for ecological studies. So researchers have to pay their specific attention to Antarctic fish and their parasites to study environmental changes. Next slide, please. Olena, may I ask next slide? Yes, that's why aim of our study was to analyze the species diversity of the parasite community of main teleos fish species near the Ukrainian Antarctic station. Oops, we missed one slide. In previous slide, it was information that fish fauna near Antarctica is pretty diverse. And yes, and 77% of fish diversity in Antarctica, in Southern Ocean near Antarctica is presented by Natatenoidae, fishes from this suborder. They are endemic of Antarctic and subantarctic region, and they are uniquely adapted to cold environment, and they have very specific parasite fauna. In the area near Ukrainian Antarctic station, there are 33 species of fishes. Most of them are also not Natatenoidae, and dominant species of teleos fish are Natatenia coriceps, Natatenia rossi, two species of genus Trematomus, Henocephalus aceratus, and Parachorhynchus charcoti. And next slide, please. And that's why aim of our study was to analyze species diversity of the parasite community of this main teleos fish species near the Ukrainian Antarctic station. I hope in future we will use this data for long-term monitoring studies. So next slide, please. Next slide. Yes. As I told before, collection of parasites was carried out during the previous one, during the 19th and 24th Ukrainian Antarctic expedition in the water area near Ukrainian Antarctic station Academic Radzi. I think everybody knows where this station is, and much better knows than I do. So this area was very well studied before, because West Antarctic is area where many parasitological studies were performed in 1970s, 1980s. And next slide, please. Next one. Yes, during this two Antarctic expedition, 156 fish specimens of 6 species were collected. All fish species were collected using fishing rod in the area near Antarctic station in the depth of 10-30 meters. All fish species were measured and dissected using standard parasitological technique. Totally more than 20,000 specimens of helminths were collected from 6 fish species. Among them, more than 7,000 nematodes, 2,000 cestodes, almost 7,000 trematodes, appendocephalans, and about 200 specimens of ectoparasites. In our studies, we studied only ectoparasitic helminths. All leeches and ectoparasitic crustaceans were excluded from our study. So all helminths which were collected manually were fixed in ethanol, transported to Ukraine, and here in Kiev, in Institute of Zoology, all helminths were identified separately under light microscope using morphological criteria. Second slide, please. Next slide. Next slide, please. As a result, we would see that all fishes, all previous one, may I ask previous one? All 100% of fishes which were examined in our studies were infected with helminths. From 3 to 21 species, and from 9 to more than 1,400 helminth specimens were collected per one host. Here on this table, you can see how many specimens of every fish species and how many helminths and helminth specimens and helminth species were collected in our study. Next slide, please. Next slide, please. In our study, previous one. Previous one. Yes, 30 species of helminths from 5 taxonomic groups were found in our study. 1 species of ectoparasitic monogenia, 11 species of acanthocephalans, 5 species of nematodes, 9 species of trematodes, and 4 species of cestodes. So it's pretty high species diversity. Next slide, please. May I ask next slide? Ectoparasites, yes. Only 1 species of ectoparasitic helminths from class Monogenia was found in Antarctic fishes, Pseudobenedenia natatenia. It is a pretty widespread ectoparasitic helminths. In Natatenia coriceps, it was found in more than 36% of fishes, and in Natatenia rossi, in 33.3% of fishes examined. But only these 2 fish species were infected with this ectoparasite. Yeah, next slide, please. Next slide, please, Lena. 5 species of nematodes were found in all fish species examined nematodes, if it's possible, 1 back. Yes. The most widespread group, dominant group of parasitic nematodes were anisakid nematodes, which normally parasites are of marine mammals, seals or whales, and they parasitize fish on larval stages. 3 genera, Pseudoterranova, Contracepum, and Anisakis, were detected in teleos fish near Ukrainian Antarctic station. All anisakid nematodes were found in liver or in body cavity. Here on the picture on the bottom of our slide, you can see how heavily infected can be liver of fish sometimes, especially for predaceous fishes. And two species of nematodes, Scarophis natatenia and Diphyllina fraseri, are specific parasites of fishes, and they were found in fish stomach and fish intestine. Next slide, please. Next slide. Yes, cestodes of 4 species of cestodes were found in 4 of 6 fish species examined in our study. Cestodes also are parasites of marine mammals, and they use fish like their paratenic or second intermediate hosts. Unfortunately, we couldn't identify larval stages of cestodes to species level. That's why we identify them like Diphyllobotrium sp or the Tetraphilidean metacestode ABC, but we pretty sure that they are separate species. We only need molecular studies to approve their species status. Larval stages of Diphyllobotrium sp, you can see this nice larvae on upper slide. They were found in 4 of 6 fish species examined in rather high prevalence. They are parasites of seals, and what we could see comparing our data with data previously published, number of Diphyllobotride cestodes near Ukrainian Antarctic station is increasing. Maybe it is connected with increasing of population of seals. Now we can't tell exactly. Larval stages of Tetraphilidean cestodes with nice 4 botrias, you can see them below, were found in 3 fish species, in Natatenia coryceps, Parachenectes charpoti, in Hinocephalus aceratus. All Tetraphilidean cestodes are parasites of sharks, which teleos species are included in their life cycle like intermediate hosts. Next slide, please. Digenean trematodes, very nice and delicate creature, which you can see on this picture. They are specific parasites of fish. In our study, we found 9 species of trematodes, which parasitize every fish species examined. And I can tell that this is a very rich group of helminths in Antarctic fishes. And the most prevalent species were Macvicaria georgiana, Echidnalinea boerwersi, and Elytrophaloides oatesi. You can see them on this picture, and they were found in 4 or 5 fish species. So they are not host-specific parasites, and they were found in almost in every fish, with prevalence up to 93%. Next slide, please. Acanthocephalans were found in 11 species of acanthocephalans from 4 genera were found in our study. The most widespread group of acanthocephalans were species from the genus Corinazoma. They dominated fish Antarctic fish. They were found almost in every fish, but Corinazoma, they are parasites of marine mammals, seals, and birds. That's why we could find them only on larval stages. So all these parasites were juvenile, and most of them were encysted in body cavity. So but their identification to species level was possible. And in our study, we found 5 species from the genus Corinazoma, what is pretty high species diversity. Three other genera, Asperzentis, Echinarynchus, and Metacantacephalus, all Acanthocephalus from this genera are typical parasites of fishes, of marine fishes. And we found 6 species from these 3 genera, and all of them were specific for Antarctic fish from Southern Ocean. Next slide, please. In our study, we had opportunity to examine parasite species diversity depending on host species. The richest species diversity was observed in Antarctic rock cod Notothenia corycips. 26 species were found. And in Antarctic dragonfish, Parachaniftes charpoti. Even now we believe that we can separate one species in Parachaniftes, and we will have even 27 species of parasites. All other species of fishes had less diverse parasite fauna, but we had no opportunity to make normal comparison because samples of other group of species, Henocephalus oceratus, Trematobus bernaci, Notothenia rossi, and especially Harpachyphora antarcticus, all samples were very small. Unfortunately, these 4 species are not so widespread in the area near Antarctic station like Notothenia corycips, so we need more material to perform statistically significant analysis of species diversity. But what we could see, we could analyze host specificity. And we came to conclusion that for all helminth species which were found in our study, there were no host specificity. All of them were found more than in 1 host, usually in 2, 3, or up to 5 different fish species were infected with this helminth. Next slide, please. We have opportunity to analyze structure of the parasite community only for 1 fish species, for Notothenia corycips, because sample which was examined in our study was pretty high, 125 specimens. And we could separate 5 groups of helminths according to their prevalence. Dominant parasites which had prevalence more than 80%, subdominant, background, rare, and occasional species. Dominant and subdominant, both these groups are also very abundant. Together, these 6 species composed more than 80% of the total helminth number found in our study. Next slide, please. Next slide. And we had an opportunity to compare our current data with data of Professor Zhitevetsky and Professor Laskovsky, our Polish colleagues, who studied Notothenia corycips and parasite fauna of Notothenia corycips near Ukrainian Antarctic station 2 decades ago, in 2002. So we could see difference in number of species found, and what is the most important, we could see significant differences in prevalence and abundance of several helminth species. Even we have only preliminary data, we need more information, but we can consider these 6 species, maybe a couple of more other species, as potential indicator species to monitor ecological state of parasite community and for future complex monitoring study of ecological changes in Antarctic ecosystem in general. Next slide, please. Next slide, please. And to conclude our work, I can tell, first, we found that helminth species diversity of main 6 teleos species near Ukrainian Antarctic station is rather high. We found 30 species. It's a very rich helminth fauna. Also, we first documented the significant changes in the parasite community of Notothenia corycips during last decades. Even our data are only initial. We only started this work, But we can tell that the results of our work provide initial information on the state of the parasite community of bonyfish in the Argentine island area and can be used for future monitoring study on ecological changes in marine ecosystem of West Antarctica. Of course, future studies are necessary. So next slide, and thank you very much for your attention. Yes.
Thank you very much, Tatiana, for your interesting presentation. And I invite you to look in chat and find questions for you.
Would you mention parasites of shark? Are there
any specific feature common in parasites of shark who feed rarely?
A few moments. It is show metabolism compared to shark most active feeders. It's difficult to say. Not many publications of species composition of shark parasites are around West Antarctic, unfortunately. I don't know why. Sharks were dissected pretty often for morphological studies, but not too many publications on shark. But what I know that tetrabotryid cestodes in Southern Ocean are specific parasites of sharks. Most of studies were done in Eastern Antarctic near Australia by Australian colleagues. We hope to get tetrabotryid cestode fixed in pure ethanol to make molecular studies and make more conclusions about their connections with sharks, with special species of sharks. On this stage, we only could perform morphological studies. So I have only general information about cestode and shark.
Okay, thank you very much. Tatiana, any questions? Okay. Thank you very much, Tatiana, once more. For your
thank you very much. Thank you very much, and I'm terribly sorry for such inconvenience. I expected I had have problems with.
Okay, for many of us, it's very seldom opportunity to training to catch our experience in such conditions. I wish everybody healthy and healthy once more, and hope to meet each other in the next future conference. Tatiana, I ask you to look for seminar, which yesterday our Malaysian colleagues invited us. And maybe it will be interesting for you too. Okay?
Yes.
Thank you. Thank you. Bye. And we go to the next participant, Andrei Utevsky. We have Andrei Utevsky right now.
Yeah.
Yeah, okay. Please.
I'm here. Nice to hear all of you. Can you see my presentation?
Yes.
Yeah. Let me introduce my research. This concern with the studies of phylogeny of the different ectoparasites from polar regions of our Earth. What is a bipolar distribution? The occurrence of bipolar distribution is caused by conditions that prevailed during the Ice Age. When temperature in the tropic decreased and northern organisms were able to spread to the tropics and reach the southern hemisphere. Later, when the temperature increased, this organism became extinct in the tropics, resulting discontinuous habitat. This explanation of bipolar distribution was suggested by Lev Berg in his monograph Climate и жизнь, Climate and Life, which was published in 19 47. Now more than 200 tags of different invertebrates and vertebrates, such as foraminiferous, priapulids, crustaceans, pycnogonids, mollusks, holoturians, bryozoans, have bipolar distribution. On this picture, you can see amazing theropod mollusk, Cleona limacina from Arctic Ocean and Cleona limacina Antarctica from Antarctic seas. That bipolar divergences was shown for fossilized bivalvian mollusks from family Buchida, which was found in late Jurassic and early Cretaceous period. This is a period. In this year, a new deep sea fish leech of bipolar genus Pteropdelina, which parasiting on Antarctic toothfish Disastichus mawsoni, were published in collaboration with Roman Solat from Institute of Fisheries and Marine Ecology from Berdyansk. All specimens of the new species were collected on the Antarctic toothfish Disastichus mawsoni. You can see the image of this fish. At the period from 2007 2019, at the depths near 1,700 meters. According to communication of collectors from Institute of Fisheries and Marine Ecology, a few specimens were also found at the depths near 2,600 meters. Previously, some species of fish leeches from polar regions was found on large depths, such as Ambelobdella shandikovi on the depths 1,400 meters, Galateabdella bruni from the depths near 4,000 meters, and Iogansonia extrema from the depths near 8 kilometers from North Pacific, from Kamchatka Trench. And this species was found at the depths near 2,600 meters. 23 lots and near 150 leech specimens deposited in the collection of the Department of Zoology and Animal Ecology of the Karazin University. On this picture, you can see these leeches in the mouth of Disastichus mawsoni, and the image of leech with natural coloration. During the very long period, the described specimens was found in 5 seas in Antarctica, and the places of collection you can see on this map. This is a Cosmonaut Sea, Cooperation Sea, Mawson Sea, Ross Sea, and Amundsen Sea. This is a great part of the Antarctica where the new species Pteropdelina vernadsky was found. On this picture, you can see the holotype of new species with amazing external features of this leech. You can see the zigzag sculpture on the dorsal surface of the leech. This is a unique characters of this species, which wasn't found on any species of fish leeches. No, and on this slide, you can see some mid-body samites with zigzag sculptures and projections on the dorsal surface of this leech. And the suckers of the leech with marginal flanges as a part of all unique sculpture of this leech. This new species very resembles the Pterobdelina jenseni, the piscicolate leech from North Pacific, which was found in North Pacific, especially in Okhotsk Sea and near Spitsbergen or Svalbard, at the Barents Sea on the depths near 1,200 meters at the period from 1961-2002. Totally was during this period was found only 5 specimens. New species was found in at the number near 200 specimens, and Pterobdelina jenseni present in collections in the small number of specimens. We conduct the phylogenetic analysis of 2 species which have some similar features of the external and internal structure. We use 3 gene markers for genetic analysis. This is a mitochondrial genes, and we used 60 tags of piscicolate leeches and 8 taxa of the other leech families for phylogenetic analysis. For outgroup, we used some Arinhabdeli species such as Gerudo orientalis, Erpabdella monostriata. It's a very known our freshwater species, and 6 Rhinhabdelid species from family Glossophonia, very common species Glossophonia complanata, Hemiclepsis marginata, and Pteramizon tessulatum, which parasiting on the birds, and Azobranchus branchiatus, Azobranchus yacianus, which parasiting on the marine turtles. It's a very representative outgroup for this analyzing. Phylogenetic sequences were aligned using the online version of MAFT with after options. The best fit models for each partitions were identified with a Bayesian information criterion using KQSAN for software. Phylogenetic relationships were assessed by Bayesian inference using software Mr. Bias as implemented in the Cypress Science Gateway. This is an online service. Searches of the consensus phylogenetic tree were performed in 2 parallel runs with 1 cold and 7 hot chains, each for 10 millions generation for calculating more representative bootstrap of the clades of the consensus phylogenetic tree. On this slide, you can see the consensus phylogenetic tree, which was prepared using Bayesian inference with 60 tags of piscicalid leaches and 8 non-piscicalid leaches for outgroup. You can see 3 clades, but picture not large. This is a Pterodelina verna new species and Pterodelina jenseni. This is from Antarctica and this is from North Pacific. And undescribed species from Antarctica. This is a new species which we now preparing for new publication. You can see the good bootstrap one. This is a very good bootstrap which combines the Pterodelina jenseni and undescribed species of Pterodelina from Antarctica and the bootstrap 064, which combined Pterodelina vernadsky and Pterodelina jenseni. Totally, you can see the group of Pterodelina species, 2 species from Antarctica and 1 species from Arctica, which combined in one clade. And second clade, this Limnotrachelobdela ake from North Pacific and Ambulobdela shandikovi. This is a deep water species from the depths near 1,400 meters, which was found and described from Roslee. This is the second bipolar clade. And a third bipolar clade, this is a Antarctic endemic genus Murabdelina, genus Murabdelina, and Arctic and North Atlantic species Oxytonostoma typica, which combined in third bipolar clade. On this picture, you can see this species which combined in bipolar clades, Ambulobdela shandikovi, which was described in 2018, and Limnotrachelobdela ake from North Pacific, which was described at 1924. And members of third bipolar clade, Murabdelina biannulata, which was described from which parasite on pycnogonids, sea spiders, and Oxytonostoma typica from North Atlantic and Arctic, which parasiting on polar skates. Currently, our knowledge does not allow clarify which events led to the observed distribution pattern in the Pterodelina and other bipolar clades of fish leeches. But we assume that all bipolar clades originate from the southern hemisphere. More explorations should be undertaken in polar regions, and further phylogenetic analysis are needed to outline the whole picture of the evolution of the leech fauna and maybe other ectoparasites. Thank you for attention. You can see the leeches of new species. The image was made in this year by Pavel Zabroda, which took part in the expedition in Antarctica. Thank you again.
Thank you very much to you. Andrei, any questions, dear colleagues?
Sorry,
I understand. Sorry.
I cannot open the window.
You need not window, open the chat. But as I can realize, we have a question to you in the chat. Dear colleagues, I understand all of us are very tired from morning to the second part of a day. But let's try to be attentively and put questions. Okay. We have, it seems to me, once more. No, only thank you very much for the presentation from your colleague.
Thank you again.
You have question from Pete Conway.
The range. Now we are planning next research, and we are planning to include some core genes in our research for calculating the molecular clocks to estimate the time of divergence. For my opinion, there is some divergences during the evolution of the fish leeches, and we can see that on the phylogenetic tree. Maybe first divergences was at the maybe 400 million years ago, when the first pisciculate leeches divergented from Antarctic region, and this conclusion was made at some publications, especially our idea based on the phylogenetic analysis of the Megalobdella shidati, which combined with a very genetically old species of leech which parasiting on sharks. This is the first step of divergence. And maybe this species divergented from Antarctica maybe in Cretaceous period.
Fascinating. Okay, any questions more? No? Thank you very much to Andrei. And
thank you very much
to the next presentation. Komplikevich, our technical,
yes, I'm here.
Okay, nice to meet you there. Please start your presentation. Okay, thank you.
Is it on the full screen or not?
Yes, it is.
Thanks. Dear participants, let me introduce the results of our research on the topic bacteria of genus Pseudomonas isolated from Antarctic biotopes. Biotopes of Antarctica are characterized by unique conditions: low temperatures, dryness, and low content of nutrient substances. Evaporation and sublimation of water from reservoirs leads to the concentration of salts, especially sodium chloride, which helps to reduce the freezing point of water. The unique conditions are the reason for the presence of a number of adaptations in the microorganisms of these biotopes: ability to spore formation, synthesis of proteins and carbohydrates that prevent freezing, chaperone synthesis, proline isomerization, synthesis of cold-resistant proteins and enzymes, and exopolysaccharides. Among these properties, almost the main is the formation of enzymes that are active at low temperatures and easily inactivated by increasing temperature. Due to their unique properties, Antarctic microorganisms are more attractive for practical use to biodegrade hydrocarbons, phenols, bioremediation of the environment from heavy metal compounds. Another application of microorganisms isolated from Antarctic habitats is shown on the slide. We investigated the presence of hemo-organoheterotrophic microorganisms in 22 samples of moss soil, lichens, and the presence of organisms that exhibit lipolytic, proteolytic, cellulolytic, or amylolytic activities. It was found that all samples contained hemo-organoheterotrophic microorganisms. The largest number of the colony-forming units were in the sample 9.9A 2019 from Cape Rasmussen Antarctic Peninsula. Heavy metal ions are common in Antarctic habitats, as shown by many scientists. Concentrations of heavy metal ions in substrates increase over the years, which I will discuss later in the report. Romaniuk et al. Established relationships among the content of heavy metal ions in substrates and the prevalence of metal-resistant microorganisms in these regions, and the presence or absence of gene homologies that provide such stability. As a result, it was shown that most metal-resistant strains were resistant to copper, arsenate, and arsenite. Co-resistance to at least 2 toxic ions was also noted. No correlation was found between the presence of genes for resistance to certain metals in bacteria and the corresponding phenotype. From 12 samples, 3 or more morphotypes were isolated. The largest number of multi-resistant isolates was in samples 89A 2019, Demshansi Antarctica from Galindes Island, and 89A 2019 moss soil from Irizar Island. In general, 33 isolates of microorganisms were selected that were resistant to cadmium chloride, manganese chloride, cobalt chloride, potassium bichromate, ferrous sulfate, copper chloride at a concentration that exceeds the maximum permissible for the corresponding heavy metal ions by 100 times. The aim of the work was to conduct phylogenetical analysis and investigate the physiological and biochemical properties of metal-resistant Antarctic isolates from different substrates. According to the result of sequencing of 16S rRNA gene, 4 strains of bacteria of the genus Pseudomonas were isolated. The map shows the point from which samples of substrates were taken from which these strains were isolated. Species of the genus Pseudomonas are common in various biotopes, including extreme ones. Due to their metabolic unpretentiousness, ability to use a large number of organic compounds as carbon and energy source, and production of biologically active compounds. Bacteria of this genus are producers of exopolysaccharides, polyhydroxyalkanoates, and bacterial lipases, capable to degrade the aromatic hydrocarbons as benzene and toluene, and bacteria of this genus are characterized by multiple resistances to antibiotics and heavy metal ions. Among the selected strains, 2 belong to the species of Pseudomonas yamanorum and to another 2 Pseudomonas arsenicoxidans. The nucleotide sequences of these strains are deposited in GenBank database according to the number indicated on the phylogenetic tree. Strain Pseudomonas yamanorum IMV B7916 is deposited in the depository of the Bolotny Institute of Microbiology and Virology of NASU. The affiliation of isolates to one or another species is confirmed by the high bootstrap value in the reconstruction of the 16S rRNA genes. Differential and diagnostic features of the studied strains coincide with the properties typical to bacteria of the genus Pseudomonas. Compared with the strain of Pseudomonas yamanorum HH1, which was isolated from soil samples collected in Isla de los Estados in Argentina, the isolated strains of Pseudomonas yamanorum are characterized by low amylase activity. They grow on starch ammonia agar but do not form a zone of starch hydrolysis. These strains are characterized by lysine decarboxylase activity. Strains of Pseudomonas arsenicoxidans 5A1N24 and 89-1T89 are not able to hydrolyze pyrrolidonyl beta-naphthylamide, in contrast to described in the literature Pseudomonas arsenic oxidans VC1. Strain differences within the species were detected, in particular the ability to reduce nitrates, hydrolyze pyrrolidonyl beta-naphthylamide, form an indole from the tryptophan, hydrolyze aliphatic thiols, lysine decarboxylase activity. Other strains differences were determined using ID 32GN kit and presented on the slide. There are the ability to assimilate rhamnose, dairy-bose, sodium acetate, potassium ketoglutamate, L-arabinose, and so on. Resistance to heavy metal compounds is an important feature of industrially promising microorganisms. A fairly common property of Antarctic microorganisms is resistance to antibiotics and heavy metal ions. This slide provides information on the presence of certain heavy metal ions in the soils and shoots of plants of Kind George Island, Galindez Island, and the Argentine Islands. In a research of Fabry et al., it is shown that copper, nickel, zinc, vanadium evenly distributed in the soils throughout the Phil's Peninsula of King George Island. The presence of heavy metal ions in Antarctic substrates is due to the processes of their bioweathering and human exposure. In the article of Fabry et al., the increased content of certain heavy metal ions compared with the previous studies conducted in these areas was revealed. This is associated with anthropogenic influences. In a study of changes in trace element enrichment factors in a glacier during 1950 to 2016, conducted by Leo et al., shows that the enrichment factor of arsenium, cadmium, copper, lead, and stibbium exceeds 10, which means the influence of non-crustal resources. These scientists have shown a relationship between their enrichment factor of arsenium and copper mining production in South America before the 90s. The decrease in this indicator correlates with the introduction of environmental regulations in 1994 to limit the arsenium emissions into atmosphere. As a result, the arsenium concentrations in the Antarctic ice sheet decreased significantly since the mid-90s. The authors argue that there is a correlation between the annual change in metal concentrations and the sea ice concentration. The aluminium, manganese, lead, and vanadium exhibited significant positive correlation with sea ice concentration in southern Indian Ocean, which may indicate the dominant source area of air masses to Dome A. The strong positive correlations are in the region of East Antarctic offshore area in the Indian Ocean sector, suggesting that trace element concentrations increase when sea ice concentration is high in this area. Strain Pseudomonas arsenic oxidans 89-1T89 was the most sensitive to copper chloride when grown on Tripton soy agar. The most resistant among the studied strains was Pseudomonas yamanorum 79-102. This strain grew in medium with 6 millimoles of copper chloride, 20 millimoles of ferrous sulfate, 50 micromoles of cadmium chloride, and 2 millimoles of potassium bichromate. Strains of Pseudomonas arsenic oxidans were more resistant to cobalt chloride and grew at 5 millimoles of this salt. Pseudomonas yamanorum IMV B7916 was the most resistant to potassium bichromate. For Pseudomonas yamanorum IMV B7916, the maximum permeable concentration of copper chloride is 4 millimoles, cobalt chloride 1, ferrous sulfate 15, and potassium dichromate 0.5 millimoles. For Pseudomonas yamanorum 79-102, the maximum permeable concentration of cobalt chloride is 1 millimole, ferrous sulfate 10, and potassium dichromate 0.5 millimoles. For Pseudomonas arsenic oxidans 5A, 1N, 24, the maximum permeable concentration of copper chloride is 4 millimole, cadmium chloride 0.1, cobalt chloride 1, ferrous sulfate 10, and potassium dichromate 0.5 millimoles. And for Pseudomonas arsenic oxidants 89-1T89, the maximum permeable concentration of copper chloride is 4 millimoles, cadmium chloride 0.1, cobalt chloride 1, ferrous sulfate 15, and potassium dichromate 0.5 millimoles. Despite the relatively low concentrations of heavy metal ions to which the obtained isolates are resistant, these concentrations are hundreds and thousands times higher than the maximum permeable concentration for water. Also important for biotechnological developments is the resistance of isolates to a large number of heavy metals with different mechanisms of biological influence. It is known that heavy metal ions affect the lipolytic activity of microorganisms. Because certain lipases are metalloenzymes, this effect must be considered. In particular, it is reported about lipase activity inhibition of Pseudomonas strains by ferrous, ferric, copper, and other metal ions. Lipolytic activity was determined by the ability of bacteria to form precipitation zone from calcium salt crystals of fatty acids around the colonies. Among the selected strains, the highest lipolytic activity was shown by the strain Pseudomonas arsenic oxidans 5A1N24. Ferrous ions at a concentration of 5 millimoles and copper ions at a concentration of 2 millimoles completely inhibited the growth of the studied strains on medium with twin 20. At lower concentrations, no significant effect on colony size and lipolytic activity of Pseudomonas yamanorum strains was detected. Among Pseudomonas arsenic oxidans strains, strain 89-1T89 is characterized by weak lipolytic activity, and under the influence of ferrous sulfate, a concentration of 0.5 and 1 millimole, colonies were smaller than in control. The size of the precipitation zone of calcium salts of fatty acids also decreased. Under the influence of 0.5 millimoles of copper chloride, slightly reduced the size of the colonies of these strains, but there was not significant difference in the diameter of the precipitation zone of calcium salts of fatty acids. At higher concentration of copper chloride, in the medium with twin 20 bacteria, Pseudomonas arsenic oxidans 89-1T89 didn't grow. Ferrous sulfate at a concentration of 0.5 and 1 millimole and copper chloride at a concentration of 0.5 and 1 millimole didn't affect the size of the colonies of the strain Pseudomonas arsenic oxidans 5A1N24, but decreased the diameter of the precipitation zone of calcium salts of fatty acids. The results of research concerning the influence of heavy metal ions on lipolytic activity can be useful for developing bacterial lipase production processes and also have theoretical significance for understanding the vital activity processes of Antarctic microorganisms. The synthesis of exopolysaccharides by psychrophilic bacteria demonstrates the adaptation of bacteria to low temperatures. Exopolysaccharides is a defense strategy by bacteria to keep itself from the external environment and for establishing biofilm communities on solid substrates. All studied strains produce exopolysaccharides at 6 and 22 Celsius degrees, but in different quantities. The highest content of exopolysaccharides was found during the cultivation of the strain Pseudomonas arsenic oxidans 5A1N24 at both temperatures. At a temperature of 6 Celsius degrees, it synthesizes 770 milligrams of exopolysaccharides per gram of biomass. The ability of exopolysaccharides to remove heavy metal ions from solutions, which is provided by the structure and composition of these exopolysaccharides, is mentioned in the literature. We assume this ability of exopolysaccharides of studied strains. The obtained isolates are different strains of Pseudomonas arsenic oxidans and Pseudomonas yamanorum. They differ in enzymatic activities and in the ability to assimilate certain organic compounds. They are characterized by resistance to copper chloride, ferrous sulfate, cobalt chloride, cadmium chloride, potassium bichromate, and are able to produce lipases during growth with copper chloride at a concentration from 0.5 to 1 millimole, and ferrous sulfate at the same concentrations. The strains Pseudomonas arsenicoxidans 5A, 1, and 24 is a promising producer of lipases and exopolysaccharides. This study was performed within the project Metabolic Activity, Physiological and Biochemical and Molecular Genetic Characteristics of Antarctic Metal-Resistant Strains of Microorganisms under the State Target Scientific and Technical Program of Research in Antarctica for 2011 to 2020. Thank you for your attention.
Thank you very much to you, Solomia, for the interesting presentation. And I invite you to look at the chat. You have questions there. Welcome, please. Can you see?
In your opinion, which projective adaptive characteristics of these are more prominent when the microbes are in the biofilm form and otherwise? I think that the characteristic to synthesize exopolysaccharides and also the heavy metal resistance are the most prominent in such time.
Okay. Are you finished? Yeah?
Yes.
Okay. Okay. Dear colleagues, have you any question? No? Thank you very much to Solomia. And we go to the next presentation. Pavlo Kovalenko. Yes. Okay, thank you. I see you. Welcome.
Hello everybody, my name is Pavlo Kovalenko and I present to your attention a rock analysis of mitochondrial DNA haplotypes in Belgica Antarctica populations. Belgica Antarctica is endemic to the Antarctic Peninsula. This species can be considered as a model organism for the studies of adaptation mechanisms and successful survival strategies in the harsh conditions of Antarctica. The elucidation of the above mechanism is provided inter alia by monitoring studies at various levels, from molecular to population. Analysis of the structure of Belgica Antarctica populations by molecular markers has been studied in few rocks and concerned 28 srDNA sequences and the mitochondrial cytochrome C oxidase subunit 1 gene. According to the sequence of this gene, Belgica Antarctica collected from 12 populations belong to 16 haplotypes. The haplotypes are clearly grouped in 4 haplogroups, A, B, C, and D. However, the real situation in populations of these species and its dynamics remain not enough studied. Imagos of Belgica Antarctica collected in March 2012 from 2 points at Cape Tuxen during the 16 Ukrainian Antarctic expedition were taken for mitochondrial DNA analysis. DNA isolation was performed using QIAM DNA microkit according to the manufacturer's instructions. Before DNA isolation, the samples were washed from the ethanol in which the material was fixed. Analysis of mitochondrial DNA was performed by sequencing a fragment of the cytochrome C oxidase subunit 1 gene. To do this, the fragments were amplified by PCR using universal primers. After amplifying these fragments, they were purified using a quick PCR purification kit and then sequenced on a sequence applied biosystems according to Sanji. The sequences of the obtained gene fragments were analyzed in Finch TV and the online program for multiple alignment cluster doubling. All 4 studied sequences were 711 bases pairs in length and were similar to the corresponding sequences of the cytochrome C-oxidase-subunit 1 gene of Belgica antarctica from gene bank. One sequence from Cape Taxon B is characterized by nucleotide substitution adenine instead of thymine in position 598. Such substitution was found neither for haplogroup D nor for other haplogroups in previous studies. Nevertheless, 3 sequences, 2 from Cape Taxon A and 1 from Cape Taxon B, are identical to the sequence of Belgica antarctica collected on the Berthelou Island and belong to haplogroup D. And it's not surprising, please look on the map. The studied populations near the academic Vernadsky station are in the middle of the distribution of haplogroup D according to previous studies. Thus, Belgica antarctica from the vicinity of academic Vernadsky station is characterized by haplogroup D mitochondrial DNA and the presence of 1 unique variant. Further studies are needed to establish the frequencies of mitochondrial DNA variants and their dynamics. Thank you for your attention. We will take into account all your remarks in the further work.
Okay. Thank you very much, Pablo, for the presentation. Colleagues, any questions?
I ask Pablo.
Okay. Please.
Pablo, can you read something about reasons for such cluster structure of cluster genetic structure of this Belgica antarctica? What reasons for this haplotype existing?
One of the reasons it also could be an area different area and of moment. As you can see in map,
okay,
there are some big areas which all haplogroup are distribution, but with this area there are some gaps, so it also could be a reason why there's populations have different haplogroups, because you know that mitochondrial DNA is the most known conserval sequences, so it also could be asked for your question.
So isolation, I understand.
Is isolation and huge territory of area gaps of if between the haplogroup A and haplogroup B is quite a big place. For example, and if we watch on substitution against a reference sequence in A with B haplogroups, there are so many differences.
Okay, thank you.
Thank you to both of you. Any questions, dear colleagues? If we have not more questions, Please, thank you very much to Pavlo Kovalenko. And I invite the next speaker, Igor Andreev. Have we?
Yes, I'm here.
Please.
Is it okay? Is my presentation?
Yes, everything okay.
Okay. Dear colleagues, I am very glad to take part in this conference and to have the opportunity to present here the data of molecular genetic analysis of Colopanthus quitensis populations from Argentine Islands region that was carried out in the Institute of Molecular Biology and Genetics of the National Academy of Sciences of Ukraine. Antarctic pearlwort or Colopanthus quitensis, along with Antarctic hairgrass, Ordeshamsia antarctica, is one of the two flowering plant species considered as native to the maritime Antarctic. This is the most wide-ranging species of the genus, occurring from the northwestern Antarctic Peninsula along the southern Andes Mountains up to locations northward to Mexico. In the Antarctic, the sites of Colopanthus quitensis occurrence are disjunct. They are located mainly on the Antarctic islands and the Antarctic Peninsula coastal ice-free areas, and often separated from one another by natural barriers, such as open sea, glaciers, or mountains that limit the exchange of gene pool among the individual populations. The species has been studied during the last decades as a model to investigate morphophysiological adaptations of plants to extreme environmental conditions. However, there are only a few genetic studies which can provide the data to address the issue of appearance and distribution of the species in the Western Antarctic Peninsula region. In this study, we used the IPBS markers to assess the genetic variation and genetic structure of Colopanthus quitensis populations in the Argentine Islands region of the maritime Antarctic. Few words about the IPBS markers. The IPBS method, what stands for interprimer binding sites, is based on the virtually universal presence of tRNA complement as a reverse transcriptase primer binding site in one terminal repeat retrotransposons. The IPBS technique has been introduced by Dr. Kalendar with colleagues as a powerful DNA fingerprinting technology which can be applied without the need of prior sequence knowledge. Furthermore, IPBS markers are highly reproducible due to the length of primers and the high stringency achieved by high annealing temperature, and they have already found application in clone identification, genetic diversity analysis, and phylogenetic studies. For molecular genetic analysis, a total of 92 samples were collected from 8 island and 3 coastal isolated populations, including Galindez, Skua, 8 Irizar, Bartelot, North Petermann, Darbo, and Lahil Islands, as well as Gerard Bay, Cape Tuxon, and Cape Perez. The number of the plants sampled from each population is indicated in brackets. For PCR-based molecular genetic analysis, We used 7 IP-based primers, which were earlier described by Calendar et al. in 2090, and applied for molecular genetic studies of Calabanthus cretensis from the maritime Antarctic by Polish colleagues. The number of DNA fragments generated in polymerase chain reaction for the total sample of the plants varied from 24 22 28 primer to 54 for 2085 primer. The percentage of polymorphic bands ranged from 40% for IPBS 2228 to 93.8 for the pair of IPBS 2076 and 2085 primers. This data shows that the used primers can generate highly polymorphic profiles of amplified products with the DNA of Calabanthus cretensis plants from the studied region. The next slide presents the electrophoretic profiles of PCR amplification products produced with 2224 IPBS primer. Totally 217 amplified bands were scored, among which 180 or 82.9% were polymorphic. The percentage of polymorphic bands in individual populations ranged from 18% for Cape Tucson to 46% for Galindez Island, with an average of 27.2%. The indices of genetic variability calculated from the PCR data for the total same were as follows. Shannon diversity index 0.113, unbased Naive gene diversity or expected heterozygosity 0.077, and the average Jacquard's pairwise genetic distances 0.386. A comparison of individual populations showed that some of them differ significantly, up to more than 2 times in terms of genetic diversity. The lowest values of genetic diversity measures were found in Cape Perez and Cape Tucson populations, and the highest values were found for Galindez and North Petermann Islands. The variation observed in the indices of genetic variability may be partially attributed to the different number of plant samples from these populations. However, it is observed also for the equal samples, and thus may reflect some differences in the size and history of populations or local environmental conditions that should be elucidated in further research. Analysis of molecular variants for Calabanthus cretensis plants from 11 populations from the Argentine Islands region showed that genetic differences between the individuals within populations accounted for 56% of the total genetic variation, while another 44% of the total variance was attributed to between population differences. This indicates a moderate differentiation among the studied populations and indirectly evidence their recent establishment from the same origins. A dendrogram of genetic relationships between populations based on the unweighted pair group method with arithmetic mean was constructed from the matrix of Ney's genetic distances, calculated based on the PCR analysis data. 2 distant groups of populations were reviewed. The first and the larger of which consist of several smaller subgroups, and the second includes the plants from Darbo Island and Cape Perez. It should be noted that the grouping is not completely consistent with geographical location of individual populations. In particular, the most southern population from Ohio Island clustered with the northern populations instead of its neighboring populations from Darbo Island and Cape Perez. The similar situation is observed for grouping of the populations in the first group. We suppose that this may be related to the history of the studied populations, particularly to the different times and ways in which the species had expanded to these islands. The analysis of the data using principal coordinate analysis method reviewed provide further insight in the genetic relationships among the populations in the region. We can see again the division of the samples into two groups. I'm sorry, I changed the numbers here and here. This is the first group and this is the second group. The one groups include samples from Darbo Island and Cape Perez and partially the samples from Berthelot Island. The second larger cluster includes the samples from other islands and 6 samples from Berthelot Island. It's interesting that Berthelot Island seems to be colonized by the individuals originated from different sources and can be seen as a crossroad of peerward dispersal. The analysis of 2 groups identified using principal coordinate analysis method showed that they are significantly differed in terms of the profiles of PCR amplified bands. For the first and the second groups, the number of private bands was 8 and 67, and the number of fixed private bands was 3 and 2 respectively. Furthermore, the 2-level analysis of molecular variants also showed the significant genetic differences between these groups, which account for 21% of the total variants. These results indicate the existence of 2 well-defined genetic groups of Colobanthes squatensis in the region. In conclusions, it can be said that the IPBS primers demonstrated their informativeness for studying the genetic variation of Colobanthes squatensis in Argentine Islands region. The levels of genetic diversity identified for Colobanthes squatensis populations from the Argentine Islands region are similar or slightly higher compared to ones established in previous genetic studies of the species from the other regions, including Argentine and Chile. And the results of principal coordinate analysis show the existence of 2 well-defined genetic groups of Colobanthes squatensis in the region. Taking into account relatively small distances between the studied locations, it seems that these groups were originated from 2 independent ancestral sources during the species expansion in the region after glaciation. A more detailed study of Colobanthes squatensis populations across the maritime Antarctic is needed to clearly identify the potential origins and the ways of dispersal of the species. Thank you for your attention.
Thank you, Igor, for very interesting presentation. Please, questions. One moment, I need to check in chat. We have not question in the chat. If no question, we can go to the next speaker. The next speaker is Dr. Oksana Porodnik. Please, you invite to presentation. Igor, you have some comments from Pete in the chat.
Yes, I answered for it in chat.
Okay, thank you.
Are you hear me?
Yes.
Thank you. Hello, everybody. Let me tell you about our biotechnological path of Antarctic research. In vitro clonal propagation of Calabanthus cuitensis from maritime Antarctica. Relevance and purpose. Calabanthus cuitensis is one of the 2 vascular plants of Antarctica whose adaptation to unfavorable conditions is maintained in particular by specific biologically active compounds. These compounds are able to provide protection of human skin against melanoma. Herewith, it is a high relevance to have a capacity to obtain the required amount of these plants' biomass in controlled laboratory conditions. The aim of the study is a developed method of clonal propagation of Antarctic paravolt, Calabanthus cuitensis in vitro. In this slide, you see a map of sample Antarctic paravolt were collected in Irzal Island, maritime Antarctic, by our colleges Prekrasna and Ivanets during austral summer, January-February 2019. Calabanthus samples take in this point. Next slide's methods of plants clonal propagation in vitro with author modification were applied. The first part of the carried out in the laboratory of Ukrainian Antarctic Station Vernadsky Research Base, and second part of the work was conducted in Department of Cell Population Genetics, Institute of Molecular Biology and Genetics of NES of Ukraine. In this slide, you see different state of in vitro clonal propagation, Calabanthus. A state, nature plant sampling in the Antarctic. B, sterilized plants on infalcon tubes. C, multiple shoots from noodle shoot segment in B5 Gamburg medium. D, in vitro clones growing for 3 weeks in growth chamber. And E, Calabanthus cuitensis grows in vitro in soil in a growth chamber. Next slide, result. Pass 1, the development workflow in the plant sterilization has following scheme. Washing plants under the tap in a washer, 5 minutes. Vortexing in solution, and the other, free planting in B5 Gamburg medium with 3% saccharose and 0.1 milligram of liters in NEAA in 15 milliliters falcon tubes. Cultivation at window still, illumination by natural daylight, photoperiod 12 by 12 at approximately 20 degrees Celsius. If bacterial infections was detected, plants were sterilized again. Result part 2. Calabanthus cuitensis was planted in 250 milliliters glass flasks with 40 milliliters of B5 Gamburg medium. 2. New growth conditions in our laboratory 2,500 to 3,000 luxes photoperiod of light darkness 16.8 and 13 and 14 degrees of Celsius for 2 months. 3. It roots plants formed from 2 to 12 shoots in 2 months. Plants were on shoots and plants in the fresh medium. Period of micropropagation reduced to 30 days. And 6. 41 initial seedling yielded 124 clones plant. And our index of propagation is 3. In conclusions, you see methods in vitro clonal propagations of Colobanthus cuitensis from natural habitat on Irizal Island, Maritime Antarctica was developed. In this slide, you see in photo 1, plant were flowering, and in next slide, you see many plant from one, and we take many flower from one genotypes from Irizar Island, and our works continue. And this year we take another samples from another place of Antarctica, and we continue these works. Biotechnology is very important because our work first start 10 years ago, and we have Antarctic collection from the Shamsi Antarctica. This plant is new for us. Thank you for attention, and please ask me your question.
Thank you, Oksana, for very interesting talk. Joint work of Institute of Molecular Biology in the National Antarctic Center. Some questions from our scientists. No, I will look in chat. No question.
Thank you.
No question in chat. Thank you, Oksana. Let me invite next speaker, Dr. Roman Ivannikov, ELIV group. Roman present?
Yes, I'm here.
Okay, go ahead.
Good afternoon, dear colleagues. I would like to share with you the results of our comparative study of biologically active compounds contents in Shamsi Antarctica samples.
Roman, I'm sorry, do you have presentation? We don't see it.
Just a moment. It's okay?
No.
No, Roman. We have no picture.
You should press share screen button in Zoom. Or you have...
Probably in Ukrainian.
Yes? Yeah, now.
Good, yes, we see.
Sorry.
Now you should do full screen. Yes, good, excellent.
Sorry. Let's go. Natural polyphenols are biologically active compounds widely used due to their low toxicity as well as pronounced antioxidant, anti-inflammatory, and antimicrobial properties. Synthesis of polyphenols in plants is known to increase under stress conditions. Vascular plants of Antarctica growing in extreme environmental conditions can be considered as a promising source of polyphenolic compounds with a wide range of biological action. Complex collection and limited plant material of Deschamps Antarctica, as well as the possibility of plant this due to meeting glaciers and coastline flooding, make advisable its culturing under artificial conditions. In the future, it will allow one to obtain in a controlled environment the required amount of high quality standardized rough material. The aim of the work was to study the compounds available in Deschamps Antarctica plants gathered in 6 various localities on Argentine Islands region, to introduce the plants into in vitro culture and to compare the composition and antioxidant properties of the extracts from the plants grown in situ and in vitro. The subject of the study may be interest from the practical point of view to produce effective antioxidant of plant origin. Deschamps Antarctica plants under study were collected while the season during 25th and previous Ukrainian Antarctic expedition from 10 locations at Galindis Island, 3 locations at the Lahil Island, 1 location of the Barslot, Gretioler, Darbru Islands, and Rasmussen Oasis, respectively, as well as were artificially cultivated from plants of the same localities. In the presentation, focus will be made on the data obtained for plants both in situ and their in vitro analogs from 6 locations of the following islands: Galindis, 4 localities, Darbru, 1 locality, and Lahil, 1 locality. The respective geographic coordinates are given in the table on this slide. The main tasks of the study were: 1. To investigate the composition of biologically active compounds of Deschamps Antarctica plants selected during the 25th and previous Ukrainian Antarctic expedition. To identify the biologically active compounds of Deschamps Antarctica plants grow under artificial conditions from seeds selected from the same natural localities. And 3, to perform the comparative study of the antioxidant properties of extracts and to evaluate their antioxidant activity. The following materials and methods were used for this study. To prepare the extracts and above-ground plants of Deschamps Antarctica, plant growth in situ or in vitro were used. Plants were frozen to -20 degrees Celsius and thoroughly ground and poured with methanol at a ratio of plant material to methanol of 1 gram per 10 milliliter. The extraction was performed by maceration for 24 hours. High-performance liquid chromatography method was used for identification and quantification of bioactive substance available in the extracts. Spectral data were recorded from 200 to 900 nanometers during the whole run. Qualitative analysis of extract composition was performed by means of matrix-assisted laser desorption ionization time of flight mass spectrometry. Mass spectra were recorded in positive ion extraction mode. DPPH assay was used for evaluation of the antioxidant anti-radical activity of plant extracts. On the slide, you can see the fragments of chromatograms for the extracts obtained from Deschamps and Antarctica plants growth in situ-A and in vitro B. The plants originate from Lahil Island. The main groups of phenolic compounds identified in the chromatograms were 1. Hydroxybenzoic acid, simple phenols and their derivatives. 2. Hydroxycinnamic acid and their derivatives. And 3. Luteolin glycoside and luteolin derivatives, apigenin derivatives, and other flavonoids. Here you can see the contents of various phenols in the extracts obtained from Deschamps Antarctica plants growth in situ and in vitro. According to HPLC results, the highest concentration in the in situ extracts is referred to various luteolin glycosides. The second most abundant flavonoid is apigenin glycoside. Also, in situ extracts include quite high amount of simple phenols, hydroxybenzoic acid and their derivatives, as well as hydroxycinnamic acid and their derivatives. The main feature of in vitro extract is the predominance of simple phenols and hydroxybenzoic acid and their derivatives, as well as a lower amount of flavonoids. Among the flavonoids, the most abundant compounds are luteolin glycosides as well. On this slide, you can see an example MALDI mass spectra of the extracts obtained from Deschamps Antarctica plants growth in situ A and in vitro B, as shown in this figure. The results of the analytions identification and assignment of the most probably compounds are present the following this table. The data agree well with above results of chromatographic study and provides additional specification of the extracts compounds. The most intensive peak in the mass spectrum of in situ extracts corresponds to MZ value of 449, which is characteristic of orientin, luteolin 8C glucoside. The spectrum also contains the signals that can be referred to luteolin and luteolin derivatives, such as luteolin trimethyl ether, isoswertijaponin, orientin 2O beta arabinopyranozine, and other. Thus, 7 of 17 signals can be referred to luteolin and luteolin derivatives, which 3 of those being among the most prominent in the mass spectrum. The spectrum also shows the presence of apigenin derivatives, MZ 579, as well as the other flavonoids and flavonoid derivatives. The data also confirm the availability of hydroxybenzoic acid MZ 138 and caffeic acid glucoside MZ 343. For extracts from the plants growth in vitro, the main 100% signal of the MALDI mass spectrum belongs to the ions referred to hydroxybenzoic acid MZ 38. The spectrum also includes 4 peaks, assignment to hydroxycinnamic acid derivatives, MZ222, 296, and other 6 sufficiently intensive signals of luteolin derivatives, and 2 lines corresponding to apigenin and apigenin derivatives, MZ447, 579. The data also shows the presence of several flavonoid derivatives, both in situ extract and in vitro extract to contain fatty acid, while in situ extract also includes coumarin. Based on comprehensive analysis of MALDI mass spectra, wide range of in situ and in vitro plant extracts from different islands, we have formulated the criterion of the difference between native and artificial Deschamps Antarctica plants, which can be expressed as the product of the relation of peak intensives of ions with MZ 449 to 595 and 593 to 595. To value of 0.2 is a reference point for the characterization of samples origins. All the in vitro samples had the value less than 0.2, while all the in situ samples studied demonstrate the value more than 0.2. The essence behind this criterion with respect to the biologically active substance produced by the plants as a response to influence of environmental factors of it's not yet fully comprehended and is a subject of future research. Among the antioxidants of natural origin phenolic compounds appear to have the best antioxidant anti-radical properties. The data obtained show a very high connect of polyphenols in all extracts studied. Therefore, the extracts can be expressed to possess significant antioxidant activity. Indeed, all the extracts showed high activity in the test reaction with DPPH radicals. The following figure shows that even the different extracts inhibit 50 to 90% of DPPH radicals during 30 to 60 minutes. It's worth to note that the data obtained to not trivial correction between the total phenols content and the activity of the extracts in the reaction. Perhaps it is because of the presence of various phenols with different anti-radical activity. If one compares the data for the extracts obtained from the same plants growth in situ and in vitro, can be concluded that in 2 cases in situ extracts have higher free radical scattering activity than in vitro extracts, figure A and figure B. At the same time, for plants, the in situ extracts and in vitro extracts reveal similar activity in DPPH radical inhibition. You can see figure C, F. Based to the results of our study, the following conclusion can be drawn. 1. The extracts of Deschamps Antarctica, both in situ and in vitro, were found contain 300 to 900 milligram per liter phenolic compounds with hydroxybenzoic acid, hydroxycinnamic acid, and flavonoids, as well as the derivatives of all compounds being the main groups of phenols identified. 2. Luteolin and luteolin glycosides were the most common compounds of flavonoids group, while hydroxybenzoic acid prevailed among phenolic low molecular weight compounds. The extracts from the same plants growth in situ and in vitro were found to have the similar composition with the different concentration of phenols of various groups being the main distinction between in situ extracts and in vitro extracts. In situ extracts include more flavonoid luteolin derivatives, while in vitro extracts contain higher amounts of phenolic acids. 3. Both in situ extracts and in vitro extracts show high activity in the reaction with DPPH radicals under standard test conditions. Tenfold dual extracts discovered 50 to 90% of DPPH radicals. In general, despite the distinction in the extracts composition, in vitro extracts were not significantly inferior to in situ extracts as radicals coverage. 4. Deschamps Antarctica plants are valuable source for extraction of natural phenolic antioxidants, and cultivating these plants in vitro is a promising way to produce this antioxidant at the lab scale level. The next step is to optimize the biotechnology procedure for growing the plants in vitro. I'm at a future promoting the synthesis of antioxidants, Deschamps and Antarctica plants. Of course, we are grateful to State Institution National Antarctic Scientific Center of the Ministry of Education of Science of Ukraine for support our studies. Thank you very much.
Thank you for you, Roman, and you have already question in chat. You can see it in the chat, but I can read. Dear Dr. Ivannikov, do these phenols exist in the plants in bound and free states? And if so, did you measure them separately? Their physiological roles might differ.
For second time, please write a question in chat. I will answer all of the question also in chat, if it's possible.
Of course, if it's more easy for you. Another questions? Colleagues, no questions in the, another questions in the chat. Okay, in this case, I invite to presentation Dr. Natalia Miryuta. Please.
I'm here.
Yes, go ahead.
Sorry. The report correlation model of adaptability, Deschamps Antarctica genotypes of different origin grown under in vitro condition for you. main objective of this research has been to construct adaptability correlation models of the Shamsi Antarctica plants from the collection genotypes obtained from seeds collected at different sites in Argentine island region and grown in vitro in Institute Molecular Biology and Genetics. Plant while adopting to adverse external condition had developed a number of adaptation mechanisms at level of molecular, cellular, and physiological responses, such as stress protein production, increased antioxidant synthesis, and accumulation of compatible solids. To take into account biodiversity and sometimes different directionality of adaptation mechanism, the application of methods to the complex consideration within the integral index proved to be an urgent task. The way we have chosen to season for a basic genetic characteristic influence on adaptive indices different hierarchy levels is through creating correlation models visualized in probability correlation shift graphs. Such models will allow to characterize the heterogeneity of the studied genotypes which should be taken into account the further studies. We use such methods of research. Genetic distances by ACCI and IRAP markers according to article Navrodskaya et al. 2017 and genome size based on the method of flow cytometry for genotypes that were used as basic indices of initial genetic heterogeneity for analyzed plant genotypes. To assess individual adaptability indices for the Shamsi Antarctica genotypes, measurements of the leaflet mathematic index and determination the flavonoid context by routine were used. To estimate probability correlations, the method of extreme grouping was used for pairwise comparison of serious differential differences of indices for each pair of genotypes. Within this method, when the increase-decrease of genotype differences in one index is accompanied by the increase-decrease in another one, corresponds to a positive probability connection value, while the increase-decrease of genotype differences in this index is accompanied by a decrease-increase in another one, corresponds to its negative value. Next three slides consist information about those data set. First line in this table presented name of the researched genotypes. Second column population consists population localization where the seed have been collected for the experimental plants genotypes production. Next column consists genome size, and last column consists of flavonoid content. It is interesting that flavonoid content was maximal in the Lehil 59 genotype. All these mostly of all these genotypes has diploid chromosomal set. This densitogram is located in low row, in low line of this slide, except Yelur 66 genotype, which has hypotriploid chromosomal set. This densitogram is localized in upper left corner of the slide. Okay. The next slide presented genetic distances by Jacquard between plants of different Deschamps Antarctica genotype calculated by results of ISSR, intersimple sequences repeats, and IRAP, retrotransposon amplified polymorphism analysis by article Navroitskaya et al. 2017. Next slide consists density functions based on leaflet. All these genotypes have density functions described by Gaussian models, but average value of genome of leaflet is different. First line consists genotypes with origin with Galindez Island origin. Second line consists genotypes with Great Yelur Island origin, and then Squaw Island, Rasmussen Point, and Winter, Durbo, Lehil Islands. The simple example of an algorithm for calculation united quality latent index for plant genotypes from set of different populations under standardized cultivation in vitro presented in this slide. To construct correlation models, we used part of this algorithm. First line consists source data sets of genome size or genetic distances. Leaflet and flavonoid content. Then source data set pairwise condition by index I, where I is genotype number, was consist in second line. Then we used method of extreme grouping of differences set pairs of source data by index in index I in studied genotypes, and then determination of each I genotype probability of matching into a positive or negative group. This line we used to construct correlation models. Correlation models of adaptability were constructed in in the form of bipartite-oriented graph based on probabilistic relationships between the pairwise differences of 4 major parameters: genome size, genetic distances, leaf length, and flavonoid content in the leaves, Podeshamsia Antarctica, genotypes, sets. The first step of constructing correlation models presented in this slide. Correlation models of the differences in genome size influences on differences leaf length and flavonoid content, possibly auxin-related metabolism indices for 11 genotypes, presented in a triangle graph, graphs of probability relations. Diploid genotypes with positive connections are outlined by ovals. Genotypes with chromosomal polymorphism with negative connections are outlined by rectangles, and diploid genotypes with some negative connection are outlined by pentagon. Next step of construct correlation models presented in this slide. We added to triangle graphs correlation models of differences in genetic distances, influence on differences of leaf length and flavonoid content for 8 genotypes. Internal graph is named genome size graph. External graph is named genetic distances graph. Diploid genotypes with most of positive connection outlined by ovals. Genotypes with chromosomal polymorphism and most of negative correlation and negative connection outlined by rectangles, and diploid genotypes with mostly of negative connection outlined by pentagon. The individual of the adoptive portrait of studied genotype in vitro cultivation is shown. Next, the influence of main genetic characteristic, genome size, and genetic distances on the adaptability and diasis leaf length and flavonoid content depended on auxin-related metabolism is shown. Among the studied genotypes, we distinguished 4 different variants by correlation models. The first group contains diploid genotypes of the same origin, YLUR62 and YLUR67, which have mostly positive connection, except for 1 negative in the graph genetic distances. The second group contains genotype YLUR66 and RBO12 with chromosomal polymorphism that have negative relationships in the genome size graph and between graphs. The third group contains SCOA22 and Rasmussen 35, which are characterized by presence of only positive and neutral connections in both graphs. The fourth group contains genotypes Winter 1 and Hill 49, characterized by 2 positive and 1 negative relationships in the genome size graph, and negative relationships in the genetic distances graph and between the graphs. The genotype individually and the grouping according to the peculiarities of adaptability correlation models should be taken into account during experimental studies when these genotypes used as model plants, especially in experiments to study the productivity regulation, study the influence of various exogenous factors, and so on. Thank you for your attention.
Thank you, Natalia. We already had a small discussion in the chat about we have used this method for different life forms or for different kind of plant species, or this method only for the Shamsia life form. I already like to offer answers that we try to prepare method for universal methods, because life form or kind of species not principal thing in it, but of course work with this method not finished yet, so this only one of the stage of our work. And another questions? If no questions, I thank our speaker Natalia Miruta and invite next speaker Twardowska Mariana. Mariana.
Yes.
Okay, go ahead, Mariana.
Can you see my presentation?
No.
I don't see the chat now.
Нема презентації.
To close presentation.
Наташа, виключіть мікрофон, будь ласка.
Can you see my presentation? No?
No, Mariana. We have not the presentation on the screen.
Mariana, do you need help? You need to find share screen. Демонстрация экрана.
Да.
Нажимаешь демонстрация экрана, что далее?
Entire screen. Share.
Можешь выбрать только окно, но можешь выбрать entire screen.
Не вижу.
Я выбираю entire screen.
И что дальше происходит? Дальше надо нажать согласиться, совместное использование. Да, есть.
Есть?
Есть, да. Теперь можно переключиться на презентацию.
Є тепер? Видно?
Так, є.
Good day, my name is Mariana Twardowska. I would like to introduce you my presentation. Comparative analysis of the content of biologically active compounds in intact plants and tissue culture of the Shamsia Antarctica. The ability of plants to accumulate biological active substances is used in biotechnology and pharmacology to obtain phenolic compounds, flavonoid, antioxidant, alkaloid, saponin, and other substances. Most of these metabolites are usually synthesized in response to abiotic stress, which can be caused by low or high temperature, limited access to water and nutrients, long period of darkness in winter, and high level ultraviolet radiation in summer. Accordingly, in plants growing under suboptimal or extreme conditions, the content of secondary metabolites may be increased due to permanent stress. One such species that has adapted to the environment condition of Antarctica is an extremophile plant, the Shamsia antarctica, which may be promising in terms of studying the biological activity of its secondary metabolites. It is known that the Shamsia antarctica accumulates a number of biological active substances, in particular flavonoids, ornithine, luteolin, and isosvartiaponin, phenolic compounds that show anti-tumor activity, inhibit melanoma cell proliferation, as well as inhibit the growth of colorectal carcinoma and its metastasis to the liver. In studying this species, previous researchers, this compound were found in extract from the Shamsia antarctica wild grown plants or plants subjected to 4 degrees Celsius for 72 hours, but they were not detected in plant grown in vitro at 13. They also proposed tablets and granules for the prevention and treatment of people prone to cancer, the active substances of which in the Shamsia antarctica luteolin extract. Antarctina obtained from extract of the Shamsia antarctica. These compounds can be used as an adjunct to cancer vaccines. In our work, we analyzed quantitative and qualitative content of phenolic compounds and flavonoids in plants with different number of chromosomes under different growth conditions: nature plants, plants cultured in vitro, regenerated plants, plants grown in a growth chamber, as well as tissue culture, the Shamsia antarctica. The biochemical analysis showed that aerial parts of the Shamsia antarctica plant, green color in this slide, has a high content of phenolic compounds and flavonoids compared to the roots. Only in the R30 genotype was the amount of these compounds slightly higher in the roots. In view of the results obtained, the aerial part of the plants was further taken for biochemical analysis. It was found that the content of flavonoid in nature plants was in the range of 16.6 to 25.3 milligram, in plants culture in vitro slightly lower, 2.3 to 21.3 milligrams. The largest amount of phenolic compounds was found in the wild plant of Great Yalur Island, plant from Great Yalur Island, and in Yalur 66 plant with a hypotriploid karyotype grown in a growth chamber. Among plants cultured in vitro, the highest content of phenolic substances was found in plant of Dar12 genotype. Regenerated plant did not differ significantly in the content of biological active substances from the original plant in vitro. The amount of phenolic compounds was similar in the Shamsia antarctica plant under different growing conditions. However, when transferring plant from aseptic condition to a growth chamber, a significant decrease in flavonoid content was observed. For biochemical analysis, we took various chromosome forms of the Shamsia antarctica, which we found previously. diploid with B chromosomes, diploid and hypotriploid with Robertson translocation. No significant differences in the number of phenolic compounds and flavonoids for different chromosome form of the Shamsia antarctica were found. In this slide, you can see results of a biochemical analysis of the Shamsia antarctica tissue culture showed that the content of phenolic compounds in kali of different origin was up to 5 times lower compared to the content of these substances in the explant donor plants. Only in tissue culture of root origin of genotype L57, the amount of phenolic compounds was twice as high as in the root of the initial in vitro plants. The content of flavonoid in tissue culture was 1.2 to 4 times lower compared to aseptic plants and in tissue culture of genotype Dar13. It was 18 times lower. In the roots of plants of genotype R-T, the content of flavonoids was the highest, whereas in the tissue culture obtained from it, the flavonoids amount was to order of magnitude lower. No significant changes in the content of phenolic compounds and flavonoids were observed in culture of this cell line with different period of subculture, 10 and 19 passages, Galindes Callus. It is known that the synthesis of secondary metabolites in vitro culture is influenced by such factors as the initial plant genotype, culture duration, mineral and carbohydrate composition of the nutrient medium, the concentration of phytohormone. It should be noted that all studies, tissue culture, Galindes genotype, were cultures on the Gamborg-Evalay medium supplemented with 2 mg dichlorphenoxyacetic acid and 1 mg benzylaminopurin, whereas the other were culture of the Murashige-Skoog medium supplemented with 1 dichlorphenoxyacetic acid and 1 mg kinetin. Therefore, it can be assumed that the highest content of flavonoids in kali JD12-2A among all tissue culture may be associated both with the composition of the nutrient medium and with the influence of the genotype. The qualitative analysis of extracts by HPLC revealed 5 substances that presented in similar ratios in the samples of wild and in vitro grown plants. One of the substances was identified as orientin or luteolin glycoside. The 5 basic substances, including orientin, could not be detected in the analyzed tissue culture B. Instead, they found 3 other more polar compounds, one of which is presented in a smaller amount in the Shamsia antarctica. Thus, in tissue culture, there is a shift in the biosynthesis of biological active compounds towards the formation of more polar metabolite. Kali of different origins contained mostly the same set of substances, but in different quantitative ratios, which did not depend on the type of source explant used for callus formation. In this slide, you can see the content of orientin in the leaf tissue of the Shamsia antarctica plant range from 2 to 4.8 milligram. Wild grown plants from different localities, white shading in this picture, differ significantly in the content of these substances. Plant collected from Rasmussen oasis and Rasmussen oasis and Great Yallour Island contained twice as much orientin as plant from Galindas and Darbo Island. Among plants grown in vitro, the content of orientin was highest in regenerated plant, while in other its range from 1 to 2.5 milligram. In tissue culture, under isolated growth condition, the range of synthesized compounds may expand and substances that are not typical of an intact plant may appear. The results by HPLC analysis of tissue culture Deschamps Antarctica indicate a similar phenomenon occurring. Those Deschamps Antarctica plants contained significantly more biological active compounds than tissue culture. The content of phenolic compounds and flavonoid in plants culture in vitro is generally comparable to than in wild plants, with the exception of some genotype. No significant differences in the number of phenolic compounds and flavonoids for different chromosome form of the Deschamps Antarctica were found. The qualitative analysis of extract by HPLC revealed 5 substances that are present in similar ration in the samples of wild and in vitro grown plants. One of substances was identified as orientin or luteolin 8C glycoside. The main substances has not been identified so far. It is shown that the transition of cells to undifferentiated growth is accompanied by a decrease in the content of biological active substances and significant change in their qualitative composition. In tissue culture, there is a shift in the biosynthesis of biological active substances towards the formation of more polar metabolites. All this gives grounds to consider the in vitro culture of the Deschamps Antarctica as a potential source for obtained appropriate biological active substances. Thank you for attention.
Thank you for your presentation, Mariana. Are some questions? I don't see.
No, see.
Okay. Okay, and I cannot see any questions in the chat. So in this case, our life science session came to the end. And a little bit about our future sessions, future things. In accordance, we are waiting for you tomorrow. In accordance with the conference program, the life science section will start at 7 o'clock universal time, Kyiv time, universal time 10 o'clock. Tomorrow. Sorry. Tomorrow we will have 5 oral presentation, 19 poster presentation, and after all, and 2 p.m. universal time round table discussion on the topic development of possible cooperation in life science. We are waiting for all of you at the session and in the round table. See you tomorrow. Goodbye. Be healthy.
Thank you.